Good morning, everyone. So welcome to day 2 of Ourcon 2021. So I'm logged into Gathertown and I'm seeing some familiar faces. So welcome, it's nice to see people up bright and early today, this morning. So welcome to my session 5 indeed. So this is proteomics, lipidomics, metabolites and metabolic pathways. Just to remind you, so I would put something in the chat. The group chat is now working, so please do put questions in for our speakers. I hear they are raring to go. So again, please put some questions in our chat. We are fully working. Okay, so first of all, I would like to introduce our keynote lecture speaker. So our first speaker today is Helen Cooper from the University of Birmingham. So I'd like to welcome Helen. Helen, can you hear me?
I can hear you.
Oh,
yes.
Good morning. How are you today?
Good morning. Fine, thank you.
That's great.
So I'm trying to work out how to share my screen.
Oh, okay. So take your time. Please don't rush. Whenever you're ready, and we're looking forward to hearing your talk today. If you move your mouse, Helen, to the bottom of the screen, you should see a button that says toggle screen sharing.
Yeah, I can see that, and I've clicked on it, but I can't see my presentation.
Okay. So you might have to open and... Can you toggle on and off? So if you go to the toggle, go on and off, it might reappear again. Sometimes it's a bit tricky. Yeah, and it is on a delay sometimes. Any joy? Oh.
Can you see now?
I can see the screen changing, so I can see me with you. Yes,
but
if you
not
go
the
to presentation up, perfect. You've got it.
Right.
We can go.
Okay.
So you need to go back to the presentation. That's
right.
It.
Here we go. Here we go. So you can see this now, right?
I can see that. We're back on track.
Thank you. Thank you.
Over to you.
Right. So, well, good morning, everybody. Thanks very much for the invitation to speak today. It's a pleasure to be here, albeit virtually. So today I'm going to talk about our recent work in native ambient mass spectrometry. And so I thought I'd start by just articulating our overall goal for our work, which is to combine native mass spectrometry with mass spectrometry imaging in order to obtain both spatial information and structural information in a single experiment. Now, of course, I don't have to introduce mass spectrometry imaging to anybody here, but I thought I would give a brief introduction to native mass spectrometry. Okay, so when you electrospray protein samples from denaturing solvents, so comprising organic solvents and acids, the result is unfolded proteins, and any complexes that were present are dissociated. If, however, you electrospray from native-like solvents, so these are usually ammonium acetate based, the proteins will remain folded and the complexes will remain intact. And so when we look at the mass spectra with the denaturing solvents, we see, let me get my laser pointer up. Okay, so we have higher charge, lower M/Z species, whereas with the native-like solvents, we see proteins with lower charges and higher M/Z. And from this, we can determine stoichiometry of any protein assemblies or protein ligand complexes. And if native mass spectrometry is combined with ion mobility spectrometry, we can determine collision cross-sections. And I mean, the reason that we're interested at all in native mass spectrometry is that many diseases are associated with misfolded proteins or irregular protein ligand binding. Okay, so to date we've used 2 ion sources for native mass spectrometry imaging, and we've used LISA and we've used nanodesi. And in fact, if I'd been giving this talk 18 months ago, I would have talked exclusively about LISA, so liquid extraction surface analysis. So this makes use of a robotic pipette. It's the Advin Triverse Inanimate. And so a few microliters of solvent are aspirated from a solvent well before we get liquid microjunction sampling of the tissue section in a defined location. And then the sample is introduced to the mass spectrometer by nanoelectrospray. Okay, so it is possible to perform native mass spectrometry imaging with LISA, and these are some examples. These are obtained on the Eclipse instrument. So we've got, this is heart fatty acid binding protein, regicalcin, and the RID-A homotrimer. Okay, but what's immediately apparent from these images is that the resolution of LISA is very low. Okay, so the pixel size here is 1 millimeter by 1 millimeter, and that's something that we get asked about a lot. And so about 2 years ago, we started working on nanodesi, offers a higher spatial resolution than LISA. So, I mean, nanodesi is similar to LISA in that it makes use of liquid junction sampling. But unlike LISA, the solvent is continuously flowing through 2 capillaries. Okay, so whereas LISA samples discrete locations with a discrete volume of solvent, nanodesi is a raster technique, and that is, now let me get this video working, demonstrated in this video here. Okay, so that shows the sampling taking place. Okay, so, oh, now I can't move on. There we go. Get my laser pointer back again. Okay, so if we take a closer look at the nanodesi source, and I should say that nanodesi is not currently available commercially. So this is a home-built source. It's built by Ollie Hale, who's a postdoc in the group. Ollie's got a poster later on today, so I'd encourage you to go and have a chat with him. Okay, so if we take a look here, so the solvent flows down this capillary, up this capillary. Here is the liquid junction, and in this example, the diameter of the liquid junction is about 90 microns. But the point really is that nanodesi is very flexible. It's possible to tune the diameter of the liquid junction by varying the capillary size and the flow rate. So a good thing about nanodesi is that the tissue stays relatively intact post-analysis. So on the left here, we've got an optical image taken post-analysis of, so after we'd imaged a rat kidney. And the imaging stopped just short of this left-hand side of the section. So we can compare the sampled and unsampled tissue. And so you can see that so we've got blood vessel here. So while the blood's been washed away, removed by the nanodesi, the actual structure of the tissue appears to be intact. And so on the right here, we've got a section of human liver, which was H&E stained post-analysis. Okay, so you can see where the nanodesi is tracked across the sample. But if we look closely in, you can see the nuclei remain intact. And just to sort of further emphasize that point, so here we've got 2 adjacent sections of human liver. So these are from an individual with NASH, which is a severe form of non-alcoholic fatty liver disease. And the tissue is characterised by this scarring, which you can see surrounding these regenerative nodules. Okay, so as I say, we've got two adjacent sections. So the top section hasn't been sampled. The bottom section has. Okay, you can see where the sampling's taken place. If we zoom in, again, you can see that you can see where the nanodesi is tracked across the sample, but it's still quite clear where the regenerative nodules are. Okay, so so far we've published 2 papers on nanodesi, both of those earlier this year. And so what I'd like to do is take you through some of the data from those articles before moving on to some more recent unpublished data. Okay, so in this work we were imaging rat kidney. Again, this data is from the Orbitrap eclipse. And here I'm showing some mass spectra from various regions of the kidney. And what you can see is that they are information rich, so we're detecting lots of proteins. In terms of protein complexes, so in the cortex we can see the RID-A homotrimer, so that's one that we've detected previously with Lisa. I showed that earlier. In the blood vessel we've got the hemoglobin dimer, and again that is a species that we've detected previously with Lisa. But if you look here in the renal pelvis, we've got this S100A6 dimer, so it's about 20 kilodaltons, and that's one that we hadn't detected before. So we had to identify it by performing tandem mass spec on a serial section. Okay, sorry, I went forward 2 then. Let's find the slide. Yes, okay. So these are those data. Okay, so the precursor appears to be an 8+ dimer overlaid on the 4+ monomer. And that was confirmed by proton transfer charge reduction, PTCR. Okay, so we see the 7+ and 5+ dimer. So we do have both the dimer and the monomer there. And then when we performed HCD, again, we saw dissociation to the subunit. So we've got the 5+ and 3+ subunits, as well as a number of sequence fragments. And we can search those sequence fragments against a protein database using ProCyte, and that confirms the identity of the protein. So we know it's the S100A6. Okay, so then in the second paper, we combined nanodesi with travelling wave eye mobility spectrometry. So this was work done on the SYNAPT G2S. And the reasons for doing this were twofold. So firstly, we wanted to improve image specificity. And secondly, we wanted to be able to determine collision cross-sections. So this slide has got 2 examples of improved specificity. So at the top here, we've got the arrival time distribution obtained for beta-thymosin 4. So this is a very low abundant protein. Okay, so we've got 2 peaks labelled with the red and green arrows. Now, if we look at the total mass spectrum, which is shown in this panel here, we can detect the peak, so we can see that beta-thymosin peak, but if we look at the image, we can't really discern any distribution. But now if we introduce arrival time filtering, okay, so here we've got just the data underneath that peak labelled with the red arrow. We can see that in the mass spectrum we improve the signal to noise, but also we're now able to see in the image that the protein is found in the medulla. I should say this was kidney, so this is the medulla of the kidney. And the green, the data under the green arrow is just adding noise. Okay, at the bottom is another example. So this time we've got kidney fatty acid binding protein. And if we look at the total spectrum and the image without any arrival time filtering, we can see that the protein is distributed around the cortex but we can improve the image specificity by including that arrival time filtering. Okay, so here we've got the image generated when we apply the arrival time filtering. Okay, so as I mentioned, the other reason for including travelling wave ion mobility was to determine collision cross-sections. And these are just 4 examples of proteins less than 10 kilodaltons where we've done that. So all of the collision cross-sections that were determined by nanodesi were in good agreement with those that we determined by LISA, and also with literature values where they exist. Okay, and this is some examples above 10 kilodaltons, and again, there's good agreement between the collision cross-sections determined by nanodesi and those determined by LISA, and again, literature values where they exist. Okay, so my slides are moving themselves on automatically, so I'll try and stop that. Okay, so the data I've just shown were obtained on the SYNAPT G2S, but everything I'm going to show you from now on is from our Orbitrap Eclipse. So as well as performing nanodesi in positive mode, we've also performed it with negative mode. Okay, so here we've got some images from rat kidney. So we've got the heart fatty acid binding protein, which despite its name is found in the kidney, in the medulla. We've got the S100A6 dimer in the renal pelvis, and the RID-A homotrimer in the cortex. And the top we've got the positive mode images, and in the bottom the negative mode images. You can see that there's good agreement between the two. The real benefit, though, of using negative mode is the ability to identify some ligands, and that's something I'm going to come on to in a moment. Okay, so here we've got some unpublished data from rat brain. Okay, and the images here are for monomeric proteins of between about 12 and 15 kilodaltons. And at the top, I've got some LISA images, and below are nanodesi images of adjacent sections. And I'm showing these really to illustrate the point that although the LISA images, they are coarse grain, they are low resolution, it's nevertheless good agreement in terms of spatial distribution between the LISA images and the nanodesi images. Okay, so I guess the point there is even though it's low resolution, don't write off LISA. But I am going to stick with nanodesi for now. Okay, so while we're looking at the data from the rat brain, I'd like to further illustrate the point about the flexibility of nanodesi that I was making earlier. So these are 3 images from the rat brain data set. The pixel size here is 200 microns by 200 microns. And here we've got an adjacent section. Okay, so this has been partially imaged with a finer capillary, so you can see where it's been imaged here. Okay, so the pixel sizes here are 100 micron by 100 micron, and even though we're down at these pixel sizes, we've still got plenty of signal in the mass spectra. Okay, so the proteins imaged here are ARF3, beta-synuclein, and carbonic anhydrase. But it's really important to note that we didn't know the identity of these proteins beforehand. So all of the proteins I'm going to talk about, they've been identified by in situ top-down mass spectrometry on adjacent sections. Okay, and to do that, we use one of 3 methods. Okay, so we either use LISA or nanodesi, or we combine LISA extraction with nanoelectrospray from pooled glass tips. Okay, so the LISA extraction occurs, and rather than directly introducing to the mass spectrometer, we collect the sample and then load it into these tips. Okay, so we use the image from the adjacent section to inform where we're going to sample the tissue, and the choice of which method we use tends to be down to how abundant the protein is. Okay, so in this example then, this image was obtained for a 7+ iron with master charge 3018, and it appears to have an interesting distribution. It's found primarily in the basal ganglia, and so we wanted to identify it. And to do that, we performed HCD, so this was by Lisa, okay, and then searched the fragments against the protein database, again using ProCyte, and the protein was identified as ARF3. Okay, so that's the ADP ribosylation factor 3. Okay. This data showed that the protein was myristylated at the N-terminus. So that's all great, except that there's a mass difference between the measured mass and the theoretical mass of the protein, even with that modification. So a difference of 443 Daltons. And the other curious thing in the HCD spectrum was the presence of these peaks here. Okay, so these appear to be neutral losses from the precursor ion, but the mass shifts don't correlate with 443 Daltons. Okay, so a literature research revealed that R3 binds guanosine diphosphate, so GDP, and that's got a mass of 443 Daltons. So that was really exciting because it suggests that what we've imaged here is an intact endogenous protein ligand complex. But to be really confident of that, we needed to identify the ligand. And the problem, of course, is GDP is acidic, and we're working in positive mode. And so we switched to negative mode. Okay, so this is a line scan from the brain, and this peak here corresponds to the 7- ion of this putative R3 GDP complex. So then we performed HCD of that 7- ion. So this is the high master charge region, and we detect two 6- ions, and they correspond to the loss of 362 and 442 Daltons. And if we look at the low master charge region, we detect those peaks at 442, which corresponds to GDP, and at 362, which is the GDP with the loss of phosphate. Okay, and then the final confirmation is to select this ion and perform MS3, so a further round of HCD. Okay, and this is the spectrum we get, and that confirms that it is indeed GDP. So just to recap then, so we've extracted an intact protein ligand complex from brain tissue, and then we've used tandem mass spectrometry to confirm both the identity of the protein and the identity of the ligand. Okay, and I've got another example here. So this image is 8+ ions of master charge 3100. When HCD was performed in positive mode, there were no, this is with the nanodesi, there were no backbone fragments identified. Okay, but we did see this rather unusual neutral loss region, okay, which is familiar from the ARF3 analysis. When HCD was performed after Lisa with the pulled tips, we got one backbone fragment, which suggested that this was GTPase RAN, which is another GDP binding protein. Now, of course, a single fragment does not give high confidence in a protein assignment, but the fragment was at an aspartic acid. Now, this is a preferred cleavage site in native top-down mass spectrometry. So although we've only got one fragment, we can get additional confidence from the fact that it is at aspartic acid. But again, we switched to negative mode. Okay. And so in the low master charge region, again, we've got these peaks corresponding to the GDP anion, master charge 442, and the GDP with the loss of the phosphate. And again, performed second round of HCD, so MS3, and the fragmentation pattern confirmed that we do indeed have the GDP band. But there's still a mass difference between the measured mass and the calculated mass, corresponding to 22 Daltons, but this can be explained by the fact that GTPase RAN binds magnesium. So here we've got a protein that we've identified with both a ligand and a metal bound. And we observed some other metal bound proteins. So here we've got an image of 5+ ions, master charge 2383. This shows the protein to be present in the cerebellum. We performed HCD, and again searched the fragments against the protein database, and that confirmed that we've got parvalbumin alpha. Now this is a calcium binding protein, and there's a mass difference between the measured and calculated mass of 76 Daltons, and that corresponds to 2 calcium ions. And then if we look back at the HCD spectrum, you can see that a number of the fragments actually retained the calcium ions, so giving us further confirmation of this. And then another example of metal-bound protein is carbonic anhydrase 2. So this is a zinc-binding protein. Again, so HCD confirms the identity of the protein, and the presence of the metal is confirmed both by the mass difference in the precursor ion, but also the fact that some of the fragments also retained the zinc ion. Okay, so as I should say before I move on that these data that I've shown from the rat brain with the ligand-bound and the metal-bound proteins were undertaken by Emma Sisley, and Emma's got a flash presentation this afternoon, so I'd encourage you to go and see that and talk to Emma to get more information. Okay, so as well as identifying protein ligand complexes, we're also interested in identifying protein assemblies. And one of the benefits, as I've just shown, of the Orbitrap Eclipse is this facility to perform multiple rounds of tandem mass spectrometry. So if I just use the example of the RID-A homotrimer, because I keep mentioning it, in terms of identifying protein assemblies, we can take the precursor ion, fragment them by HCD. So in this case, we've got the 10+ trimer, and then it's dissociated to the 5+ monomer and the 5+ dimer. And then we can select the monomer subunit for a further round of HCD, giving us the sequence fragments and allowing us to identify the protein. So we can calculate both the protein stoichiometry and the identity of the protein. And what we've been doing recently is trying to push the limits in terms of molecular weight of these assemblies. Okay, so this is a section of kidney, and this is the image of RID-A. So that's got a molecular weight of 42.6 kilodaltons, and that's the highest mass complex that we've imaged so far. Okay, but by optimizing source optics and the pressures in the various regions in the instrument, we can push that limit. So we've got 61 kilodalton homodimer, 67 kilodaltons, 73 kilodaltons, all the way through to 94 kilodaltons. And if we take a closer look at this, okay, so this corresponds to the magnesium-bound alpha-enolase homodimer. Okay, so that is, or the stoichiometry is determined by HCD of the 19+ charge state. Okay, so we see the dimer dissociating into the monomer subunits. Okay, so we got up to 94 kilodaltons. It's not just a rat kidney, so we can detect protein assemblies from human liver as well. Okay, so this is the NASH tissue that I mentioned earlier, so characterised by the scarring and these regenerative nodules. And this is an image of 14 plus ions of a 51 kilodalton dimer, and you can see that it appears to be present in the regenerative nodules. So HCD confirmed that it was a dimer. Okay, so here we've actually, the 15 plus has been dissociated into the subunits, and then if we increase the energy of the HCD, we see the sequence fragments that allows us to confirm the identity as this glutathione S-transferase A1. Okay, so we've got a number of proteins which we have yet to image, but nevertheless have confirmed their identity by nanodesi. Okay, so this example here is a homotetramer from kidney. Okay, it's the GPX1 homotetramer. It's 89 kilodaltons. Okay, so this is the full scan spectrum. You can see the tetramer as well as the RID-A trimer. So yeah, the 20 plus precursor was isolated for HCD. Okay, so we see the dissociation to the subunits. Subunit mass is 22 kilodaltons. And then another example, this time from liver. This is the OTC homotrimer. So here we finally breached that 100 kilodalton barrier. Okay, so we can deconvolute using PTCR. Okay, and then if we use HCD, we can dissociate to the subunits. The subunit mass is 36 kilodaltons. So this is great. All the proteins that I've discussed so far are soluble proteins. And so our most recent challenge then has been to apply native nanodesi to membrane proteins. So membrane proteins are notoriously difficult to study, but they're of great interest. So they constitute about a third of the proteome. The targets are about half of currently used drugs and about 70% of drugs in development. But the main challenge in terms of their analysis is their poor solubility. Okay, so this is typically addressed in native mass spectrometry by including detergent in the solvent. And so the effect of this is to draw the membrane protein out of the lipid membrane and into a micelle. And then on electrospray, the detergent molecules are removed, leaving the bare ion. Okay, so our goal then is to use nanodesi for in situ analysis of a membrane protein. Okay, so the membrane protein that we focused on is aquaporin 0. So this is 113 kilodalton tetramer, and each subunit comprises 6 transmembrane helices. So this is properly embedded into the membrane, so it's a proper challenge for us. Okay, so here we've got the nanodesi mass spectrum. This is obtained from a section of sheep's eye lens. The solvent contained 2 times the critical micelle concentration of this C8E4 detergent. And we can clearly detect the 15, 16 and 17 plus charge states of the tetramer. Stoichiometry is confirmed by HCD. Okay, so we are able to use nanodesi to extract the intact tetrameric membrane protein from the tissue. And so lastly, and most excitingly, we've imaged a sheep eye lens. Okay, so using nanodesi, using the 2 times CMC solvent, 2 times CMC detergent containing solvent. So this is a full scan spectrum. Okay, so we can see the aquaporin membrane protein, the tetramer in the 15, 16, 17 plus charge states. And we've also got these two species. Now these are heterotetramers of crystallines that are abundant in the lens. Okay, so perform imaging. Okay, so these are top two images are from the crystalline heterotetramers, and the bottom image is from the membrane protein tetramer. Okay, so these are equatorial sections. So looking into the eye, it's a bit spooky because it feels like the sheep is looking at you. But just to reiterate, so this is the first demonstration of imaging of a multimeric membrane protein in tissue. Okay. So just to summarise then, we've been applying nanodesi to the analysis of intact protein assemblies directly from tissue, before performing the imaging and the top-down experiments with same source, no sample preparation, in situ identification of ligands as well as the proteins themselves, and as I just said, first mass spectrometry imaging and analysis of an intact membrane protein assembly. Okay, and I'll just finish up by thanking the people who did the work. So Ollie Hale, who, as I mentioned, has got a poster this afternoon, so do go and talk to him. Emma Sisley, who's got a flash oral this afternoon. Emma's done all the work with the brain. And James Hughes, who's been working on the liver tissue. And I'd like to thank Richard Goodwin from AstraZeneca and Trish Lalore from the Liver Labs at Birmingham, who provide us with samples, and Ian Stiles, who helps us with all things computational, and our funders as well. And thank you for listening.
Wow, thank you very much. I have to say those nano-DESI images are absolutely beautiful. From the AR-F3 work right down to the difficult customers, which really are the membrane proteins. I agree. So I've got time for a couple of questions. So I do have a question for you, Helen, in the chat. Peter Marshall, Helen, do you have any idea of the concentration of the proteins that you are seeing? That's a difficult question, I know.
That's a really difficult question. That's a really difficult question. I'm sure we're seeing the more abundant proteins, and we're just trying to dig down and down.
Yeah, I completely agree. I do have another one. Okay, so Francesco says, thanks, great presentation. What is the smallest pixel size nano-DESI can reach without losing excessive intact protein signal? Are you planning to perform UVPD instead of HCD fragmentation?
So the smallest pixel sizes that we have shown are the 100 by 100 micron. So in the slide that I showed earlier. Of course, the problem of reduce, I mean, nano-DESI, of course, can have smaller pixel sizes than that. But for us, the issue is abundance. So I don't know yet how low we can go, but the issue will be signal intensity. And UVPD, well, we haven't got UVPD capability, so not anytime soon.
Okay, thank you. It is really challenging. So I know I've got to move on to the next speaker. There are a couple of more questions in the chat from Sadia and Kevin Shea. So hopefully they can have a chat with you later, because they are really good questions. But all I can say is thank you very much for a fascinating talk. Thank you, Helen.
Thank you.
No problem. So can we then move on to our next speaker? So Shibayoti Lahira from Munich. Good morning. How are you?
Good morning.
Good
morning.
I'm
good. How are you?
I'm fine, thank you. Whenever you're ready, we're looking forward to hearing your talk on in situ protein profiling.
Okay. Can you see my screen?
Not as yet. If you go right down to the bottom where the toggle is, sometimes there's a little bit of a delay.
Now? No.
Not as yet.
Okay, let me try once again.
Okay, so if you hover right on the toggle button right at the bottom, and then it will give you an option to turn it on, and then you can open your presentation.
Yeah, I think you should be able to see now.
Oh, yeah, I can see it now. That's perfect.
Perfect. That's very nice.
In your own time.
Okay. So, hi, good morning, everyone. So, from a really cold Munich. So, I am Shibo, and that's, I guess, how you should call me to make it more convenient for all of us. Well, as my title suggests today, I try to take you through a systematic process and method that we have developed recently in the lab, which enables in situ protein profiling to assess complex disease etiologies. And of course, like many of us here, the motivation comes from the fascinating milieu of the different cell types or the heterogeneity that you observe within the tissues, and which are now being increasingly implemented in both the pathogenesis and optimal management of diseases. And of course, since the proteins are the workhorses of the cells, and they are the ones that affect all the molecular processes going on inside the tissues, pathological states, they do demand investigation of the different proteins, or they demand investigation of the cellular heterogeneity at the protein level in order to clarify the interplay between cell type and functional state. So a very relevant and important case in this respect is the male infertility. So male infertility is quite a problem all over the world, as you can see, because they contribute to around 50% or even more at some places to the total infertile couples, which amounts to around 50 million worldwide. And more importantly, because of many social issues and other factors, male infertility is very limited studied in a limited way, and there are very, very less number of investigations that leads to this. There are a few chromosomal anomalies that are found for male infertility, like Klinefelter syndrome or Y chromosome deletions or other genetic mutations, but they account for only a very minor percentage of all the cases that are encountered in the entire plant. However, there had been many anatomical or physiological phenotypes that has been associated with male infertility, but the underlying molecular mechanisms and other stuff, they are completely unknown, and which has led to most of the cases, like the majority of the male infertility cases, to be classified as idiopathic cases and therefore subject to empirical treatment and with very, very limited success. So considering this background, it is obvious that the systematic understanding of spermatogenesis, the main process going on behind successful fertilization, is imperative. And spermatogenesis, as you can see, is a very complex process involving a lot of different kinds of cells like spermatogonium, spermatocytes, spermatids, Sertoli cells, involved in a wide variety of differentiation and proliferation cellular processes. And proteomic studies, mainly to address infertility, has so far mostly been focused on the sperm and the seminal fluids, thereby addressing the end point of the whole complex process of spermatogenesis instead of the process itself. However, the structure of the testicular tissue here provides us to do in situ protein profiling very well, because as you can see that all the processes involved in spermatogenesis actually starts from the periphery of the tubule and ends up in the lumen. So if you can profile proteins in situ, this should really give you a good chance of understanding spermatogenesis at the molecular level. So with this background, our initial study for understanding spermatogenesis and infertility was involved with a mouse model called Arom+. This transgenic mouse model basically overexpresses aromatase enzyme, that converts testosterone in to estradiol, thereby rendering the subject infertile. And these mice, the testes of these mice are basically characterized by activated macrophages, Leydig cell hyperplasia, and severe inflammation, thereby making them a very good phenocopy of the human case. So keeping this in mind, of course, the tools that we have applied are the very basic ones and are the very basic ones that we have at our disposal. So we sectioned the tissues, the testes from the mice at 12 micron thickness, and then performed, say, imaging mass spectrometry on sections of peptides that were generated in situ by digesting with trypsin using a standard protocol provided by the manufacturers. And then we did peptide analysis in a Brukker RapidFlex platform at 25 micron spatial resolution. And the serial sections were used for deep tissue shotgun proteomics, where we extracted, again, by standard protocols, the proteins and did an LC-MS analysis in an Orbitrap instrument. Now, data sets generated from these two complementary techniques were actually integrated then by an in-house built software called ImShot, which eventually led to identification of the proteins within the tissues. So, as a first glance of the results, you can see here that we could record spectrum from the RapidFlex platform with reasonable accuracy. As you can see, these are some 5 representative masses in both cases that spans the entire mass axis. And on the y-axis, you see the difference in between the centroid masses and the observed masses within the pixels across the whole tissue or across the whole measured area. And you see the variation is very well within the acceptable limits. And indeed, these peptides do define and mark the different seminiferous tubules that you find in these tissues. As you can see that these peptide clusters basically distinguishes the different tubules based on the proteomic composition within one tissue, and it also shows that this proteomic composition is severely reduced, or this diversity is severely reduced in the disease state. And additional clusters show the interstitial spaces in the wild type, the peritubular spaces which surrounds the tubules and performs other important functions. And very interestingly, there was another cluster that localized to the interstitial space of these diseased testes, thereby emphasizing the importance of the interstitial spaces in this particular process of infertility. So now the data integration, of course, because what we saw in the earlier slide was just the clusters, and of course we know that using these basic platforms, it is not possible to have a systematic and direct identification of the peptides here. Now, for this, we have created an in-house built software called ImShot, which uses the spatial clusters from the IMS datasets to generate the mass list as input. Now, you must be wondering that why do I use spatial clusters instead of spectra or any other thing? That's because there existing challenge of de-isotoping peptide spectra in imaging mass spec dataset. As you can see in this particular example here, that peaks at the isotopic positions of one particular peptide has entirely different distribution patterns. Now, you wouldn't expect that. You would expect that if these peaks are the isotopic peaks of the parent peptide, they would have identical distribution as this. So this means that these peaks basically arise from the different peptides which has a similar mass and is masking the isotopes in this position. Now, how to resolve this? And here it comes the spatial clustering. So we have used SkillsLab software to do the spatial clustering, but you can use any other kind of software that gives you such clusters. So when you basically segregate the entire spectrum into these different clusters, then it is ensured that the isotopic peaks, basically at the isotopic positions of the parent peptide, they do have identical distribution, meaning that these peaks now are coming from the isotopes of the parent peptide and not from other distinct peptides. So following this, you can basically apply a de-isotoping standard algorithm, which is taken care by a separate module in our software. And then you can create a monoisotopic mass list from the imaging mass spec dataset, which is then compared with the identified peptides that are obtained from the complementary LCMS study by a user-defined tolerance and try to identify them. Now, of course, life would have been much easier if you would have identified here directly and got a one-to-one mapping. But this is, of course, not the case, as you can imagine, because of the different resolution, mass resolution capacities of the RapidFlex platform and any orbiter platform. Usually you find mass of one peptide actually annotated to multiple proteins. In our case, in this data set, we found around 65% of the masses of the peptides that we generated here had these multiple annotations. So now, in order to therefore rescue these peptides from their identity crisis, we tried to do a ranking by introducing a scoring method, which we call the MLP, or the most likely peptide scoring method, that takes care of the intensity of the mean intensity of the peptide that is obtained in the LCMS data set, and the log fold change and the p-value in this equation. So the mean intensity, so if you have a higher mean intensity, this means that this would be like a more abundant peptide. And this also emphasizes the concept of peptide detection in multi-imaging mass spec, where given two or more peptides of similar masses, it would be more likely that the instrument detects the peptide that's coming from the most abundant proteins, because there are no separation techniques, be it pre or post ionization. So introducing that factor along with this fold change factor, which actually signifies the discriminatory ability of that particular peptide between the healthy and the disease state, that how different and how much can it distinguish. And in addition, we have this p-value from the volcano plot that is generated from the LCMS data that imparts the statistical significance of this particular peptide. Now, of course, you can realize from this equation and the description of the parameters that higher the score of this equation, so which means that higher the mean intensity, higher the log fold change, and lower the p-value, it is more likely that this particular peptide originates from that particular protein which has the highest MLP score. And this is exemplified in this particular example here by this brown box, where this particular mass is assigned to this particular peptide. Now, based on this scoring, what did we see, and what could we see rather in this particular model of infertility? And of course, the software also has this separate module to deal with this part of the algorithm. So as you observed before, in addition to this loss of proteomic heterogeneity between the wild type and the aromatase model, we see that actually there are processes involving different proteins in the wild type that are related to the late stages of spermatogenesis, which are severely downregulated in this particular case of infertility. And also, there are certain other processes which are very much going on within the cells that are highly active and therefore energy demanding. And if you take this special interstitial cluster within the disease model and do a pathway analysis, you can see that this is actually involved in a lot of extracellular matrix regulation through some creatine sulfate degradation and collagen biosynthesis and other modifying enzymes. Now, this corroborates very well with the previous findings that there is a very strongly observed fibrosis within the interstitial spaces and the tubular walls in this particular case of infertility. Further, we tried to validate a very small subset of the proteins that we have identified in this way, where we see a severe downregulation of the chaperone that is involved in proper folding of actin and therefore is very instrumental in the cell division processes or the proliferation processes that are part of the spermatogenesis. And a mild regulation of the collagen, which was also corroborating previous findings. And we found a novel factor, which is called the mimicin, that are involved in this creatine sulfate degradation and metabolism pathway that are highly upregulated in the disease state. We have actually we have actually identified and figured out and corroborated many other previous findings also, but because of time constraint, I would refrain from mentioning them, and instead I would refer you to our very recent publication in the Life Science Alliance, where you can have many more information. So based on this background, we thought that how or if we can actually translate this workflow or protocol into the patient samples, going a step further and going to a step nearer to clinic. And of course, for the patient samples, we needed to deal with FFPE tissues, and therefore we had to introduce several processes or several modifications to our workflow that ensures optimal protein extraction or peptide extraction from these tissues in both the imaging mass spec and LCMS analysis. And of course, I'm not explaining the details and the significance of FFPE tissue because this audience needs none. So therefore, I will quickly show you just the very preliminary results that we have obtained from our initial pilot study. So one group of patients biopsies that we obtained were the mixed atrophy, where this is characterized by normal tubules and atrophied tubules within the same section. And our workflow could actually find the peptide cluster defining the normal tubules, and the identification on them showed that they have very relevant cellular processes going on within these tubules as compared to the other cluster that distinguish the atrophied tubules and were enriched in testicular inflammation and autoimmune reaction. The second set of patients that we had were suffering from spermatogenic errors. So spermatogenesis was arrested at one particular stage, and they showed that they have one class of tubules which had only Sertoli cells, as you can see, mostly in the periphery, and there another cluster which had a mixed morphology because of the disease state. And here also we could find clusters that are distinguishing one from the other, and this disease set of tubules are basically enriched in platelet degranulation and transporter function, like for immune response activities. And the Sertoli cells, the cluster that defined the Sertoli cells, were very much enriched in peritubular regulation. Now, if you remember, the location of the Sertoli cells that I showed you initially, they are the cells that are very close to the periphery of the testicular tubule, thereby making them very much prone or vulnerable to peritubular regulation. And therefore, I would assume that this is what we would expect here in this case. So to summarize, I think I showed you a rapid and confident screening method that we use to identify proteins and processes in situ. And of course, we see different processes like proteomic heterogeneity and other processes that are differentially regulated in this particular form of infertility. And of course, we see a very strong effect of differential regulation of the tubules and the interstitial spaces according to the sample that we are analyzing. And of course, a very good outcome of this project was this data integration workflow that's quite general, user-friendly, and we have developed a desktop app for this. And this will be the app, the software, along with all the codes and all the documentation will be available publicly very, very soon. Until then, or even after that, if you want to have the software, you want to collaborate, discuss, and test the software, please feel free to mail Wasim or myself. Wasim is the bioinformatician who wrote the codes. We would be happy to share our stuff with you. So following that, of course, I think very strongly that this workflow has got a very good potential to be applied to patient biopsies and distinguish heterogeneous morphology with an aim to understand the molecular basis of different cases of idiopathic pathology. And of course, the future application would involve large patient cohorts, software extension, like incorporation of the various features in the software and other diseases for this particular projects. Which brings me to the most important slide. I'm very grateful to my entire lab for the wonderful environment that we have created here. Clara and my group has done and also is doing the translation from the mouse model to the patient samples, and we'll go on with that. Wasim is the computer engineer or the bioinformatician who is writing the codes and the software. Ignazi is our chief officer at the mass spec facility who takes care of measurements of the LCMS measurements of the samples. The aromatase models were generated in Mati's lab at the University of Turku. Arthur is the main person here at BMC with whom I collaborate in this particular project of male infertility. And Frank is the director of the Andrology Institute in Munich, from whom we are receiving the patient samples and collaborating very strongly. And of course, I thank all my fundings and thank you very much for listening and your attention. And I'm open to questions.
Thank you very much. It's really always interesting to consider health and disease, but I also like the overlapping transition phases. It's really important and what's going on there. So we're nearly to time, but we have a couple of quick questions, if it's okay. So I've got a question. So a question about the tissues that I think somebody's missed. Was it cryo or fresh? And have you observed several peptides from your identified proteins confirming your approach?
Yeah, so for the mouse model, the tissues were fresh frozen. And for the patient samples that I showed the transition of the workflow, the tissues were FFPE.
Mm-hmm.
And what was the second question? Confirmation of peptides that I observed in my identification.
Have you observed several peptides from your identified proteins confirming your approach?
Well, so in this particular case, I would say that we observed not many. We observed a couple of proteins which had multiple peptides. However, there are two more proteins which had independently been validated by a different lab very recently that we found in our identification process. And of course, we are also in the process of like increasing the high throughputness of this particular protocol so that we are able to observe more peptides. So right now, not many. The few that I showed, and the few that we have observed in other labs. This is not a very high percentage.
It's always work in progress, isn't it? Thank you. Thank you for a wonderful talk. Thank you very much. So I'd like to welcome our next speaker, if we can. So we have Nuria Slickhouse from Rotterdam. Nuria, can you hear me?
Yes, I can hear you. Can you hear me?
I can hear you loud and clear. Whenever
Good.
you're ready, we're looking forward to hearing your talk.
Yes.
That's perfect. I can see that.
Okay, perfect.
Over to you.
Thanks. So good morning, everyone. My name is Nuria. First, would like to thank all the organizers from Ourcon to allowing us to talk about our research, which is in collaboration with Waters. So today I will speak about high-resolution DESI-MSI of lipids in advanced human carotid plaque. So first, some background information about atherosclerosis. So atherosclerosis is an arterial disease where the artery is narrowing due to plaque buildup. And this plaque formation and growth is the result of the accumulation of lipids into the vessel wall. But also inflammation plays a big role by the accumulation of inflammatory cells, such as macrophages and monocytes, and also T cells and foam cells. And in more advanced stages of plaque progression, cell death occurs, and then the plaque develops necrotic areas. And in these advanced stages, there's a high risk of plaque rupture, which can lead to a TIA or stroke, which is still one of the most prevalent causes of death and disability worldwide. So the current treatment of atherosclerosis in the carotid artery is to surgically remove the plaque when it's getting too big, and this is done by performing a carotid endarterectomy. But of course, with every surgery comes a risk, and right now also for every 6 patients that undergo the surgery, only 1 stroke is prevented. So it's far from an ideal treatment. So this is what the plaque, the vulnerable plaque looks like. So typically a healthy artery has, let me put my pointer on, has 3 layers, so adventitia, media, and intima. And in the vulnerable plaque type, there is thickening of the intima layer, and there develops a large, thick, lipid-rich necrotic core, which is covered by a very thin fibrous cap. And then in this core, a lot of inflammatory cells accumulate. There are also leaky microvessels. Of course, there are the cholesterol crystals and also calcifications. So this altogether forms a metabolic process which induces inflammation on its own. However, besides this lipid-rich necrotic vulnerable plaque type, there are also plaques with the same morphology, but a very different tissue type. And these plaques have a much thicker fibrous cap and less necrosis. So this makes the plaque less likely to rupture and cause an event. So therefore you can question if removing these plaques is really necessary. And currently the patient risk stratification remains poor. And because we think that lipids play a prominent role in the disease pathology of atherosclerosis, we would like to investigate the role of lipids in the different stages of atherosclerosis. So we investigate the plaque lipid content and distribution in human carotid plaque to elucidate the vulnerable plaque biology, to correlate those findings to plaque and plasma lipidomics, and eventually to aid in the research for biomarkers for patient risk stratification. So for this study, we collected 14 human carotid plaque samples from patients that underwent a carotid endarterectomy. And these plaques were cut in 3 millimeter blocks and embedded in 10% gelatin, and then cryosectioned. So in collaboration with Waters, these samples were measured by DESI. On the consecutive sections, we performed histology. So besides the staining, which stains the lipids in red, as you see here, we also performed CD68 staining, which colors, and we performed alizarine staining, which colors the calcifications in the plaque in red. So a short explanation about DESI, although I think most of you know, but DESI is an ambient MSI technique which works with a fine spray of charged microdroplets, and those first droplets wet the tissue surface and extract the analytes from the tissue sample. And then the droplets that follow kind of act as projectiles, causing the desorption of these secondary droplets with the analyte to go into your mass spectrometer. So on this slide, that's used for the measurement, and that can be optimized for optimal measurement. And we used 100 micron pixel size for the complete data set for high throughput measurements, but also measured a subset of the data with a 20 micron pixel size for more detailed images. So the DESI MSI measurements were executed on the new Waters DESI XS source. And this source has several major improvements compared to the previous DESI source. So if you haven't seen the lunch talk yesterday, I will quickly go over them. So the most important improvements are the newly designed sprayer, which has a more robust design and is more user-friendly with less optimization. And it also has a reduced spray diameter for increased imaging resolution. And the other big improvement is that the standard MS inlet was replaced by a heated transfer line, which improves transmission and especially in the negative ion mode. And for more detailed information, you can also still visit Mark's poster number 38. So then with DESI MSI, we detected around 550 lipids in positive ion mode and 650 lipids in negative ion mode. And in earlier research, we found that lipids within the same class correlate to each other. So this slide shows some example images of the different lipid classes we detected in both ionization modes, and how these patterns of the different lipid classes are specific, and how they are also different between the advanced and moderate plaque type in some cases. So now we have detected these hundreds of different lipids, difficult to analyze all by eye. So therefore we performed dimensionality reduction by unsupervised spatial clustering analysis called non-negative matrix factorization to extract the major spectral patterns. So the input was 418 mZ of lipids in positive ion mode that were present in at least half of the sample size. And we got out 8 components that separated the lipid classes. So here you see those 8 components. And as you can see, every component is one, or in two cases, two different lipid classes. So next we compared these images from the NMF to histology. And we found a co-localization of NMF component 3, which are the phosphatidylcholines, to co-locate with a CD68 immunostain, which stains the macrophages. So as I said before, macrophages are an important driver of inflammation. So it's interesting to know lipids they co-locate with to understand the inflammatory mechanism in the plaque. And because it's difficult to see for you on this slide, I will zoom in on the next slide. So here you can see a zoom in of the plaque area with macrophage infiltration. So here you see the complete image and the zooms. Next to it, you see the MSI image. So this is a 20 micron pixel MSI image of phosphatidylcholine 32.0 of these same areas. And this co-localization can be explained by literature where there's a study that describes that when macrophages are loaded with LDL lipoprotein particles, which are the major cholesterol transporters in human plasma, the levels of phosphatidylcholine significantly increased. So then we also found that NMF component 2, which consists of sphingomyelins, co-located with calcifications in the plaque. So here you see an overview of all the plaques and their histological image, but I will zoom in again on the next slide. So here you see the images of the alizarines. Areas are the calcified regions of the plaque. And on the right, there's a zoom in of sphingomyelin 34.1, which co-localizes exactly with these red regions in the histological image. So when we look for a biological explanation, this can be sphingomyelin-mediated arterial calcification. So this is a mechanism where the increase of oxidation, and that increases the sphingomyelin content in the cell membranes, and subsequently the interaction between the membrane and the ionic calcium increases, so there's a higher risk of plaque calcification. So then for negative ionization, we'll do dimensionality reduction by NMF analysis to extract the major spectral patterns. And this time the input was 606 lipids and we got out these 8 components. So in negative mode, lipid identification is still ongoing. But, we did find a large difference for NMF component 1 between samples. And component 1 presumably consists plaques. And as you can see on the right, there are histologically very similar samples. So they are both very lipid rich, as you can see in the O-red-O staining. They have similar grades of calcification in the plaque as well, and similar grades of macrophage infiltration. However, this component 1 is completely absent in one sample, while it's very intensely present in the other sample. So this proves the high heterogeneity of the samples, and also that this cannot always be captured by histological analysis only. So then our next question is, what do we do with the findings of these mass spec imaging studies, and how can we translate this knowledge to the patient care in the future eventually? So one option is that is currently studied in our group is to use this information for non-invasive optical spectroscopy of plaque lipids, and that can be used for in vivo imaging later on. So in this way, lipids we find to be correlated to vulnerable plaque type can be detected in the patient for risk stratification purposes. And the other possibility is to find a correlation between the plaque lipids and plasma lipids to discover the possible biomarkers that can determine plaque risk. So ideally, this could be that you have a certain lipid profile A that corresponds to stable plaque and a lipid profile B that corresponds to unstable. Differences in the plasma lipids of patients with plaque lipid profile A and B as well. So to answer this last question, we have set up a pilot study for which we included 18 patients that had a carotid endarterectomy. And we collected from these patients also patient information, such as the risk factors, the medication use, and the laboratory data. And we are from these patients. So actually 14 of these patients were the same as I showed you before in the imaging study. So we have DESI-MSI data from these plaques as well. And then from all plaques, we also took a 3 millimeter block that we didn't embed in cryosection, but we homogenized the sample and performed a lipid extraction on the tissue homogenate. So then we had the tissue homogenate and the plasma sample mesh platform from SciEx, which can identify and quantify a lot of different lipid species. And we got out about 750 lipid species in both plaque and plasma of the same patient. So this data we can compare to each other, but also correlate to the DESI-MSI results that we will have. So to conclude, dental plaque lipid signature using DESI mass spectrometry imaging and NMF clustering revealed patterns that correspond to classical histology, but also identified the plaque heterogeneity that is not captured by histology. So what is currently ongoing is that we need to identify the lipids, especially in negative ionization mode. We would also like to quantify the analysis between histology and MSI imaging to plaque and plasma lipidomics. So I would like to thank the whole Department of Biomedical Engineering and especially these people. And of course, I would also like to thank my co-authors, especially Mark and Emmanuel from Waters, and thank you for your attention. So if you have any questions, I'm happy to answer them now.
Thank you once again for an inspiring talk. Thank you, Nuria. So if you do have any questions, please pop them in the chat. I've already got a question by WhatsApp, actually. And the question for you is, so with your new DESI ion source, how quickly could optimization be performed for a specific sample type? So I guess what they mean is, one, have you tried to optimize in any other vessels or any other areas of the heart or any other tissues? And what type of sort of time scale are you looking at to optimize such beautiful images on a different tissue?
Yes, well, actually, Mark Towers measured the samples for us on the DESI, but we do have a lot of experience already with the, we measured before the carotid samples by MALDI as well. And now Mark performed the DESI on these samples. So the nice thing is, I think, which makes it easier for those samples is that we can measure the calcified regions in the plaque as well. Because with MALDI, the laser kind of burns away these calcified regions. So that is, I think, really nice with DESI that works. But for optimization, I think it was really fast. It was working really well.
That's really good and reassuring to hear for those that want to branch out into a different area of imaging. So thank you. So we've got a question in the chat from Marco. Marco says, nice work, Nuria. Maybe I've missed it, but I have a question. Did you perform immunohistochemistry of CD68 on fresh frozen tissue? The staining looks great.
Yeah, so thanks. The staining was performed on the embedded tissue. So we have the embedded blocks and the consecutive cryosections, and then we first remove the gelatin and then perform the stainings.
Guess that answers your question, Marco. Thank you. All I want to say again is thank you for your lovely talk. Each talk seems to offer so much work and interesting work going on currently. So thank you once again.
Thank you.
You're welcome. So with that, can we move to our last speaker? We have Kevin Shea. Kevin, are you in Nashville, Tennessee right now?
I am. Can you hear me?
I can. So I was just going to say good morning because I believe it's 20 past 4 in the morning. Is that correct?
That's correct. Yeah.
My goodness, you look quite fresh and perky, to be honest. So thank you for joining us.
Sure, it's my pleasure. I just have to find the correct file here.
That's not a problem.
To share.
In your own time. So Kevin is joining us from Vanderbilt University, and he's going to talk to us about age-related changes in glycolipid profiles in human lenses.
Okay,
and
can you see my screen?
I can.
Great. A laser pointer. All right. Well, I want to thank the organizing committee for giving me this opportunity to present our work on lens lipid imaging. And I also want to congratulate Malcolm and his team for putting together a great conference under difficult circumstances. It's been truly outstanding. The presentations have been great. So what I want to tell you about this morning are some lipid imaging of human lenses, and specifically some glycolipid images. So I probably don't need to tell you, but the lens sits in the front part of the eye and serves to focus light onto the retina in the posterior part of the eye. And if you take a cutaway view of the lens, there are two major cell types: the anterior epithelial cell layer, shown here in purple, so light would be coming in from the top in this particular view, and then these elongated cells, which are called fiber cells. And the fiber cells differentiate from the epithelial cells in the equatorial region. And upon differentiation, the fiber cells lose all their organelles, so there's no light scatter in the central axis where light moves through the lens. And what this means is that there is no new protein or, for today's talk, lipid synthesis in the older, fully differentiated fiber cells. In fact, these cells in the very center of your lens are as old as you are. They were formed in utero. And so we've been looking at the lens for, well, the last 30 years or so, because it provides a wonderful model tissue to look at protein aging, and for today's talk, again, lipid aging. So the proteins and lipids in the outer shell or the outer cortex of the lens are relatively newly synthesized. And as I said, the proteins in the center of the lens are as old as the organism is. And when we take a cutaway view of a three-dimensional lens, we can cut the lens in equatorial sections, so you get this honeycomb pattern of fiber cell cross sections, these elongated hexagons, or we can cut the lens in axial direction and get these elongated fiber cells. I think for most of the presentation today, I'm going to be showing you equatorial sections where, again, the older molecules are in the center of the lens and the younger molecules are in the outside cortex, the outer cortex. Another important point I want to make at this point is that multiple groups have studied enzymatic activity, and there appears to be very little, if any, metabolic activity in the very center of the lens. So we attribute most of the changes that we see as a function of age to be non-enzymatic processes, and that'll become important later. So early on, when we started imaging mass spectrometry in my laboratory, we were looking at lens proteins. Some of you may know Gus Gray. He's at the University of Auckland. He spent his postdoc in my lab, and he imaged lens proteins. And here I'm showing age-related effects on proteins. So for alpha A crystalline, one of the major crystalline proteins, it's around 19 kilodaltons. In a 7-year-old lens, this is the full-length alpha crystalline in the upper panel. It's most abundant in the outer cortex, the young fiber cells, but it's present throughout the entirety of the lens. And you can start to see some truncation products, some C-terminal truncation products build in the oldest fiber cells of the center lens, even in a 7-year-old lens. As we move to older lenses, you can see that the center of the lens starts to be depleted in the intact protein, and the smallest fragments accumulate in the center of the lens. Now, when you get to be my age, plus or minus a few years, I'll let you figure out whether it's plus or minus. There's not so much prevalence of intact, unmodified alpha A crystalline in the center of the lens, hence the dark color in the center. Most of the intact, unmodified proteins in the youngest fiber cells of the outer cortex. And even one of the major truncation products is further degraded. And at the bottom panel, you can see the center is filled with many different truncation products. I'm just showing one here. And then there's not much change as you get older. So you see the very, very similar pattern of small truncation products accumulating in the oldest fiber cells of the lens nucleus or core. So we asked ourselves, what's happening to the lens lipids? And so we looked at 6 different human lenses in 2 different sets of experiments. We cut frozen lenses, 10 micron thick. We applied different matrices, either by sublimation or spraying. For today's talk, I'm just showing the negative ion mode since we're interested in glycolipids. We used an FTICR instrument for the first study and then followed this up more recently with the timsTOF instrument. Spatial resolution is not critical. These lenses are typically about a centimeter in diameter, so 60 to 150 micron spatial resolution is what we typically use. And so the first point I want to make is that what we see is that lens lipids have distinct distributions within the lens. So in the left, I'm showing a cartoon diagram of lens regions where the outer cortex is shown in blue, the nucleus or core is shown in red, and then there's a barrier region shown in green. I won't go into the details of what this barrier means, but it basically is a region where, as we age, materials no longer penetrate to the center of the lens. And by materials, I mean water and nutrients. So I use this cartoon diagram to show that for 3 different lipids shown here on the right, they have very distinct spatial distributions. We have a lactosyl ceramide that appears in the core. We'll come back to this in a minute. And then some PE ceramides that are differentially distributed in the barrier and outer cortex regions. So this may confer different properties of the membranes in these different lens regions, which is of great interest to lens biochemists. Now, of course, we are not the first group to look at lipids in the lens or lipid aging in the lens. My good friends and collaborators in Australia, Roger Trescott, Steve Blanksby, Todd Mitchell, looked at lens lipid content using LCMS technologies, and they showed that as a function of age, total phosphatidylcholine, phosphatidylethanolamine content decreases with age. However, as highlighted by this red box here, the panel on the right, they found that total ceramide content increased with age, and they isolated just the lens nuclei from these donor lenses. So the age of the material is roughly the age of the donor. And so they didn't have a good explanation for this increase in ceramide content. They speculated that this was perhaps degradation of ceramide-containing lipids. Now, many of you know Shane Ellis from his days in Ron Heeren's lab, but I knew Shane when he was doing lens biochemistry, and he did some in Australia, and now he's back at his home institution in Wollongong. He used DESI imaging mass spectrometry to look at lens lipids, and in this example, he's showed for a 41-year-old donor lens, these sphingomyelin lipids, two different forms, were predominantly found in the cortical or outer part of the lens. And we see a similar distribution in our MALDI imaging, shown down here for this sphingomyelin lipid, and roughly the same 40-year-old lens, in this case 48, the lipid is predominantly found in the cortical region and not so much in the core. So there's general agreement with what Shane published. Now, Shane published a variety of different lipids, but I'm just going to focus on this one right here for the moment. This ceramide phosphate is predominantly, again, in the outer cortex, and we see an abundance of this also in the outer cortical region of a 40-year-old lens. And in Shane's publication, notice this panel here at master charge 540. Here's a ceramide that's concentrated in the core or the nucleus of the lens, which is consistent with what this group found by LCMS in previous work. So when we looked at our initial study of lens lipids, we found a ceramide shown here at master charge 648 that's predominantly in the center of the lens, although it looks to be throughout the lens. And it's more prevalent in the oldest lens, 78-year-old lens. And we were looking for lipids that were accumulating with age and in the core, and we found this pattern shown here for a number of different signals. We identified this particular signal at master charge 862 as a lactosyl ceramide, accumulates in the center of the lens with age. And then there were a couple of other signals that at the time we didn't know what the structures were, but we noticed that the mass difference corresponded to an acetyl hexose and a hexose, respectively. So we speculated that these might be gangliosides. And the structure of which is shown here, I'm sure most of you are familiar with this, but the ganglioside structure consists of a sphingosine backbone, and then is modified through very specific enzymes to attach glucose, galactose, a sialic acid form here off the galactose, and acetyl galactosamine, and another galactose. So that's the general structure. As I said, the enzymes for making these gangliosides in cells are well worked out. And so there are different forms. So I'm going to focus on the gangliosides GM3 and GM1 for this talk. You can see that there's the ceramide backbone with the various sugars and the sialic acid in the green. And so these gangliosides in the red box are what we're going to focus on for the data I'm going to present. So now we move to our more recent study, the timsTOF study, where we looked at three lenses of differing age. And so for this ganglioside, GM1, the structure in symbolic form is shown on the right. We see accumulation with age in the cortical region, not so much in the core of the lens. And with the timsTOF instrument, we're able to do MS-MS directly from the tissue. We get a very nice tandem mass spectrum that confirms the structure of this particular GM1 ganglioside. Here is a different GM1 with a different fatty acid composition. And we see a very similar pattern, not so abundant in the younger lenses compared to the older lens, and again, accumulation in the cortical, not the nuclear region of the lens. Now here's a slightly shorter or smaller ganglioside, GM3, so it's missing those last two sugar moieties. And again, we see accumulation in the older lens, although it's slightly more abundant in the younger lenses compared to the GM1 lipids, but again, not so much in the core, a little bit in the core. The tandem mass spectrum shown here confirms the structure of this particular ganglioside. Here's another GM3. This one, interestingly, appears to be more in the very outer fiber cells, the outer cortex. And the difference between the previous GM3 and this one is a single double bond. So the previous one was 18:0. And so here we have 18:1. It has a slightly different distribution than the other GM3. So what is in the core? So we found two different lactosyl ceramides. Again, the symbolic structure shown here, that it seemed to be accumulating in the core, the central core of the oldest lenses. Again, the tandem mass spectrum shown here directly from the tissue confirms the structure of this particular lipid. We also found that these GA1 gangliosides, where actually these gangliosides are missing their sialic acid, the neuraminic acid moiety, and so they just have the sugars here. And so these also accumulate in the very center or core of the oldest lenses, in all lenses, but there's more abundance in the oldest lenses. So we speculate now that, again, there's not much metabolic activity in the center part of the lens. So we speculate that these gangliosides, these larger gangliosides, are degrading non-enzymatically over decades of life to form these smaller gangliosides and lactosyl ceramides in the center of the lens. And here's the tandem mass spectra for both of these GA1 forms. So here's the summary of what I think might be going on is the largest GM1 that we've observed, shown here, can break down by losing the sialic acid form to these GA gangliosides, and then can lose the terminal sugars to form the GM3, again, non-enzymatically. So in conclusion, there are two major takeaways from this talk I hope that you get, is that there are distinct spatial differences in different lipids, not just gangliosides, but many other lipids that appear in the outer cortex, the inner cortex, and the core, and that this lipid distribution changes with age. Again, lots of lipids change with age. I just focused on the gangliosides today, and we speculate that this is due to non-enzymatic degradation of these gangliosides. So the future questions are, and the implications of this work are, first of all, what is the young lens composition of the gangliosides? The larger ones didn't seem to be very abundant compared to the older lenses. And so what gangliosides are present and made in the youngest lenses? And then, of course, what are the functional consequences? Gangliosides are known to be involved in lipid raft formation, which can affect membrane protein function. They can also affect lipid membrane flexibility. And so do these lipids play a role in presbyopia? That is, the lens becomes stiffer and we need reading glasses after we're, say, 40, 45 years old or older. And there's also older work that has shown gangliosides present in cataractous lenses. So do these lipids play a role in cataract formation? All interesting questions that need to be further investigation. So I want to thank my co-authors, Amir Hossein and Jared Fincher, who were postdocs in the lab, who generated all the data I showed you, the donors and the National Disease Research Interchange for providing the lenses, funding by NIH, and support by the Mass Spectrometry Research Center at Vanderbilt. Thank you for your attention, and I'm happy to answer any questions.
Thank you so much, Kevin. It's really inspiring talk. I do have a couple of questions, actually. So a question from myself. I do apologise. I missed this. Were your samples the ganglioside synthesis study from deceased individuals or enucleation patients?
Yeah, that's a very good question. They are from deceased individuals. Cataract surgery today, 99% of them use phacoemulsification, so they grind up the tissue so you lose all the spatial information. So it's hard to get fully enucleated lenses.
Yeah, so I just wondered how that would affect, you know, lipid metabolite synthesis. I don't know. So that was just my question. So, okay, thank you. I do have a question. So, Kevin, are you looking in diseases such as cataracts and glaucoma? Oh, right. Yeah, and they've just put, oh, she's partly answered the question now, so that's fine. It was just something else, if you don't mind. Okay, so talking about any pre-treatments for tissues. So obviously when you're looking at lipids, you want to keep everything as is. In many terms, it's very different, isn't it, to looking at proteins and peptides and so on. But do you do any pre-treatments to the tissue for analysis?
Yeah, that's a good question. When we first started the work, we looked at washing with ammonium formate, try to get it, shift everything to the protonated form for positive ions. But we found that not washing provided a more reproducible signal, and the washing did not increase our sensitivity at all. So we just went with no washing at all. So these are frozen lenses. I failed to say that. These are frozen and unwashed.
Frozen, unwashed. No, thank you. That's really, really good work. Don't think we have any more questions coming through or on the text. So all I can say is, again, thank you for contribution. Thank you for getting up at a ridiculous hour. It's really appreciated. Thank you. Thank you very much.
My pleasure. Thank you.
No problem. So just before we close our session, before break, I just need to give, one shameless plug. If you go to poster 44, I repeat, poster 44, Malcolm and I have a poster there. We're doing a part 2 to the Imaging Methods and Protocols book. It will be due out probably this time next year. If you're interested in contributing either a Methods and Protocols chapter or a discursive chapter of some sort, then please drop us an email. All the details are on poster 44. Thank you very much.
Hello and welcome to the single and subcellular imaging session. I am Stefania Manetta. I am a PhD candidate at Imperial College London, and I'm also a PhD representative at MSIS. Our first keynote speaker is Theodore Alexandrov from EMBL. So yes, let's welcome Theodore. Hello, Theodore. Welcome. Nice to see you virtually.
Hello. Yes, can you hear me?
Yes, everything is fine.
Okay.
So whenever you're ready, you can share your presentation.
Cool. All right. So I'm actually surprised very pleasantly that it works. So thanks for the introduction. Hello, everyone. Really nice to reconnect with all of you in this virtual setting. It feels almost real. So, and I'm very excited and I'm very thankful to the organizers to present the work that we're doing. Can you see my screen?
Yes, perfect. We can see it. Thank you.
Perfect. Great. Okay. So what I will talk about today is how we transitioned or how we enhanced our capacities from doing spatial metabolomics to actually do single cell metabolomics. And here I would like to introduce and acknowledge the efforts and of the whole team who contributed here. And by the way, two members of this team, of our team, is actually here in either the audience or somewhere in this virtual gather town. So please, if you have questions, do not hesitate, and quite the opposite, please do approach them, in particular Mons Akilov. And Mons, he is an imaging mass spectrometrist in our team, and he's the person behind the mass spec and to make really mass spec produce perform the best. And the second person also in the audience and today is Lachlan Stewart. And Lachlan is our senior software developer and also data scientist behind Metaspace. So if you have any questions, then do not hesitate absolutely to reach out to him. And I must say that a bunch of new features that I'll quickly announce today that we introduced in Metaspace over the past 2 years, they actually were spearheaded by Lachlan, who was motivated by talking to many of you at the previous, the last real, not virtual Ourcon. So this is actually how it goes. We listen, we try to understand what you need, and then we expand software that we also give back to you. So, but let me introduce our team briefly. So we combine scientists in biology, in omics. We definitely use mass spectrometry for detection, and here there are several people who are working on various aspects, instrumental and computational aspects of mass spec. We have several computer scientists who try to actually figure out the ways how to best use the data for this. And then also we have the software developers, software engineers who implement software, in particular Metaspace, that some of you may know. So let me now present the topic, the research focus of our team that got crystallized over the past years and also over the COVID time with our recent developments of single-cell metabolomics. So what we are currently extremely interested is to develop tools, methods, experimental and computational methods, which help reveal metabolic cell state. So by now, in particular with the development and applications of single-cell RNA sequencing, we know so much about cell types, we know about their differentiation, but we also start understanding that it's not only cell type which defines the function of the cell, but it's actually this state, in kind of transcriptional state. But clearly, we are also very interested in non-genetic factors. And here we come to this question, which is elusive and also dynamic. And so far, there is not many methods to really elucidate this metabolic cell state. And we know by now also that this cell state is affected by other cells. I mean, they can be cells which surround it in the, say, tumor, in the tumor microenvironment, or cells which interact, but they also can be distant cells which produce some sort of chemokines or cytokines. So it's also influenced by the environment. And here we're talking about microenvironment as, again, tumor microenvironment, but also, global environment. What does effect we have coming from pollution, from the environment we live in, and from all the chemical interactions we have with the space and also other human beings that, and here definitely diet, but also microbiome comes into play, of course. And then definitely we are very much interested in how pathogens, how they can reprogram the state and how they maybe can trigger it for their benefits, and how therapy can potentially influence the metabolic cell state to increase efficacy, to reduce the number of non-responders. So long story short, here spatial and single-cell metabolomics comes as very handy technology, because if it's spatial, it really provides the context. And here you can definitely appreciate all the advantages imaging mass spec brings to the table. But also it's single cell, it helps reveal and dissect the single cell heterogeneity. And metabolomics here as the powerful technology which provides readouts closest to the phenotype, in particular to probe non-genetic factors. We can not only detect the host or cell metabolism, but we can also detect drugs. And this is where definitely pharma and those of you working on drug discovery development are interested in. So our target applications for spatial single-cell metabolomics, the two topics which interest and excite us the most are cancer and in particular tumor microenvironment. And here we're slowly coming to this area, not only working with clinical data and biopsies, patient biopsies, but also ourselves or with collaborators, but also working with tissue constructs such as organoids. And the second topic where I think single-cell metabolomics will have the major impact is immunometabolism, or understanding how immune cells, how they reprogram their metabolism, but also what kind of requirements, metabolic requirements they have, and how can we influence here. And here you see, for example, an image from our collaborator, Erica Pierce and colleagues, where they show this immunotherapy, anti-PD-1, anti-PD-L1, and now it's known that even like to improve the efficacy of this immunotherapy, there are a lot of questions about metabolism that we need to answer. So this is like our major applications that we are looking forward to work on. So the technology we have in-house, and our tools are not only for this technology, but this is what experimental setup that we're running is AP small d 5 with 5 micrometer pixel size, coupled with the Orbitrap. And this allows to do very high spatial resolution, yet high mass resolving power and accuracy. So, and this is the setup we have, and I'm showing it here also to bring it, kind of provide the segue for the second part of the talk, which will be about single cell metabolomics, is 5 to 10 micrometer pixel size. And as you know, we can detect a bunch of molecules, metabolites, and so on, but definitely generates a lot of data, and this is where we are coming into play. So my talk will be split or arranged into two parts. The first part will be focused on metaspace, because despite, with single-cell metabolomics, you will see we're very excited, and this becoming the major topic for us. Metaspace and in general, imaging mass spectrometry is the foundational technology for us, and we keep pushing it, we keep improving it. That's why let me first go through the metaspace part before I'm switching gears to single-cell metabolomics. So, so here I want to take a step back a little bit and review the computational progress which has happened in the field over the past 20 years with the contribution of a lot of groups. And what's also interesting is not only computational groups who contributed, but also experimental groups, which out of necessity, they brought up a lot of important questions, and they developed a lot of solutions for this, starting from signal processing, where there is definitely questions, normalization, centroiding, and so on, then visualization, color mapping, like visualization per se, but also overlaying and all enhancements you can think of. Statistical analysis is absolutely necessary for any sort of biomedical questions. And here there is not only the questions of segmentation, but also how can we search for markers in the spatial data. So what we are interested in, and we're interested in for the most of our time over the past 7 years, is molecular identification. As you might know, here we have developed to metaspace, but also there is all other approaches that definitely exist beside metaspace. And what we think is the next biggest step that is being performed already, but will be the focus of attention, is actually how to make sense out of molecular data and statistical data that is coming from imaging mass spectrometry. And here definitely there will be questions of validation, in particular molecular validation, but also biological and biomedical validation and interpretation on the molecular level, doing enrichment analysis, pathway analysis, and linking all this data also to other omics. So, and still, interestingly enough, there is lots of open questions. And for anyone computational scientist in this field, I mean, there is so much still to be done. And so one is like, we rely so much on math accuracy because in particular before ion mobility is getting more and more increasingly introduced, we're relying basically on math accuracy. So how can we make it better? So then also, how can we not only visualizing now, there are many and more very nice software tools for visualization for getting images, but how, once I visualize, how can I share these results with my collaborators within the group, besides group, how can I share it with pathologists? These questions are still open. And so in statistical analysis also is very exciting to witness the tremendous progress we all achieved in this direction. And there are so many really cool software packages for this, and many of them, they are actually presented at this conference. But there are still questions and brought up not only by me, but also many others. So how can we deal with batch effects? Because we know that mass spec, in particular imaging mass spec, is slow technology. So from one slide to another slide, from one day, a month to another month. So we introduce batch effects, and how can we compensate for them? And then if we're talking about, as yesterday was this panel discussion, inter-lab studies, how can we compensate for batch effects introduced not only within one mass spec, but also between the mass specs and between the labs? This is definitely, to get down to molecular data, there is still some work, reproducible, and there is still some work to do. So in molecular identification, since we know this topic probably the most, of other topics that we're dealing with. So which molecules can we detect? And some of these questions are still open. Where they are located, in particular if we have different edits, they produce different images, and how to estimate the confidence, and how can we find molecules with a higher confidence from the data that we're given. And finally, so definitely there is a still open question about pathway analysis, but linked to genes, proteins, and ultimately mechanisms. So, and this is kind of like where we stand as the community. And as you might know, we focus mainly on molecular identification. And here I'll show the slide which I showed previously. So what's the big deal? What has attracted us in this topic? And not only that the data set is big, it really requires powerful computing and some sophisticated algorithms, but also we want to search for more and more molecules. And as you will see, we are in Metaspace, we're introducing additional molecular databases, trying to cover even space of microbial metabolites. And this space is like the number of molecules we want to screen for is actually much larger. Now we know it. And there is still the question, like if I have a molecule of interest, I know molecular weight, I know the common adduct. So in fact, this molecule can still produce through the combinatorial tree or hierarchical tree of possible ionization pathways. So it can produce many MOZ values, and some of these MOZ values, they can have different images. And if I can still resolve it for one molecule, I need to do it efficiently for many, and then I need to account for the presence of many, because again, in particular, before ion mobility was introduced, we're dealing with extremely complex mixtures, as complex as it can be in analytical chemistry. And we need to account that all the signals can be produced from my molecule of interest, but also others. So knowing how the molecules produce signals is extremely important. Deconvolving is extremely important in dealing with this in the context of uncertainty, so-called dark matter, which produces a lot of signals which we do not still know how to associate them with molecules. This is still a big challenge. So Metaspace, this is the software that we have developed. Again, here, shout out to the current developers. Many people have contributed our past. So Lachlan Stewart here is in the audience. Please do reach out to him. But also Lucas, our front-end developer, and Sergey, our back-end developer. And what Metaspace can do now, anyone can submit data there. So upload them in IMZ email centroided format, and in roughly 10 minutes, you will get the molecules. You will get email that the process is finished. You can have a look. And just in the browser, you can browse your results, you can visualize them, you can sort, filter, and you can also share, and you can also publish it. So what's really, really cool, and I think this is probably the biggest impact of Metaspace, not even providing you the capacity to create this image, find molecules in your data, but to really share a lot of results and data publicly. And so, I don't know, something like 7 years ago, there were just a few, really handful of public data sets available. And now in Metaspace, there is more than 5,000 publicly available data sets. So anyone who is interested in method development can go there and find some data sets for themselves and use it for absolutely open with just acknowledgements of the submitters for their purposes. And I see already that this is switching completely the ways how computer scientists work. So I think this we have achieved here as a community a lot. And we are also paving the way. I mean, I see in many other communities and fields, they're reaching out to us and they're like, how could you do it? So I think that here we're also paving the ways for some other omics. And definitely this project is purely academic, it's nonprofit. And so we are really fortunate to get supported by European Commission, by National Institute of Health, and also lately we were fortunate to explore some new technologies together with IBM and the European Project Cloud Button. So if you don't know how Metaspace looks like, please go to metaspace2020.eu, in particular in virtual setting. Now you can just open another tab and go there. And so if you come there, so there is many different options. There is some introduction here, but you can go to projects. You can pick a project like here, where we are showcasing data sets from Matthew in previous year and sharing previously on Genentech, which they shared with the whole community. And now we can see this data sets of whole body sections, which beautifully demonstrate the power of imaging spectrometry of getting untargeted metabolomics analysis with high spatial resolution. So over the past years, Metaspace really helped many scientists throughout the globe. We have processed more than 58,000 submissions. And again, what's really, really cool about it, that 30% of them, they went public. So, and it is used now by more than 80 labs, more than 80 registered users. There are more than 500 projects. So this is sort of the folders that someone created. And what I'm most happy about is that we are really witnessing now the scientific impact, where Metaspace was mentioned or used in more than 100 publications as of, I think, September. So, and the number of data is growing. It's what's really cool that COVID didn't really impact the field negatively. We see even an increase of submissions during COVID times. So here I also want to give a huge shout out to those labs who contributed most of public data. I believe that they really set the stage not only for us as the community of imaging spectrometry, but for pushing this community forward, because we need computer scientists, we need data scientists, and they need data. So if we share the data publicly, we are actually attracting and reinforcing our field by large. So here I want to give shout out to Chris Anderton lab from PNNL, Zoltan Takash from ICL, Kumar Sharma from UTHSA, Max Planck, Bremen, Manuel Libicke, Christian Janfeld, Copenhagen, then also Liebe at Berlin University Austin, and many, many others. So it's really great to witness how they actually share their results publicly. And a lot of these data sets, they are really reference data sets, they're the best quality. So we worked hard also over the COVID time, and then here I would like to quickly highlight the latest features we added. So now you can share IMS emails if you want to share them with your collaborators. It's very easy to do. You can publish your data, and this is very important for supporting the consistency of the data in the future. Once you create, you publish, you put it in the paper, the data will be frozen, and we'll make sure that this data will be stored the best way, and these links will really become permanent links. So you can overlay multiple images. I'm illustrating it here from dataset from sharing. And then you can also use custom databases if you have molecules of interest. They can be also private. And we have, together with collaborators, we have added new public datasets, in particular database for N-glycans together with Dusan Velikovic and Chris Anderton from PNL. And there is more things coming up soon. So there will be features on visualization, adding TIC, like on the fly. You can compare and visualize multiple data sets. For those who are interested to really get down to the bottom of their data, so you will have an option to browse your IMS emails, and also introducing regions of interest for either statistical analysis, or if you want to share it with or get this information from your pathologists. So with this, I would like to slowly wrap up the first part of my talk about Metaspace, and I would like to kind of review where we stand since we developed bioinformatics methods. We implemented it for cloud computing, we made it available to everyone, and now there is this growing by itself knowledge base of data sets that anyone can use. And I think we're still at the beginning of this point where we started developing methods for based on artificial intelligence, and we are also looking forward how others can make use of this knowledge base and these capacities. So take-home messages. So metabolism is absolutely essential to study, and what's really cool, for many properties I didn't go into, it demands to be done in situ on the single cell level. And we all know, and we actually did the publication on this, that imaging mass spectrometry already a few years ago was the major technology for spatial metabolomics, and it just continues to increase its strong position there. And so we are really happy and fortunate to contribute to this field by providing you an open, free cloud platform for spatial metabolomics. With this, let me switch gears and let me highlight our work on single cell metabolomics. This is pretty recent. So the publication went out just a couple of months ago. The preprint was 2 years ago. But what happened over this past 4 years is that single cell metabolomics became the major direction in our group. Almost every newcomer who's coming works on single cell metabolomics, either applications or development. And we really believe that this is the major direction also for imaging mass spectrometry. So for those who don't know, who didn't witness, who didn't heard about single cell revolution, it is really happening. So in other omics, so 2017 and 18 were the years of single cell transcriptomics and genomics, but mainly single cell transcriptomics. There were special issues on this, and 2018 single cell transcriptomics was the breakthrough of the year. So single cell metabolomics is catching up. Here you see different single cell omics technologies. Yellow is metabolomics. You see how 2019, 2 years ago, it was kind of catching up here. What's interesting, all these technologies, they grow exponential scale. So we are, of course, there were many people before us who started developing single cell metabolomics. And there are many people also in the audience who do develop them now. And this is based on microsampling, based on multi-imaging, and various other technologies. So what we wanted to, specific challenges we wanted to address with other developments is to increase throughput, is to remove the need for cell isolation, and to be able to perform it on the regular imaging mass spectrometer that many of you have in your labs. So the method that we have developed called SpaceM. So the method was pioneered by Luca Rapes, PhD student, currently postdoc in Barcelona and CRG. And for everything, I refer to this Nature Methods paper. There is like a lot of supplementary material if you're interested to go really answer detailed questions there. But how it works, an overview. So in a nutshell, we take cells, we culture or plate them on a glass, then we do microscopy, we fix them, we do microscopy to find where they are, how they look, what's their fluorescent phenotype. Then we do imaging mass spec, we run data through Metaspace. Then we do image analysis, and this is the step that Luca invented, how to overlay it with a subcellular precision, image mass spec and microscopy. And once we do also normalization, we can assign normalized signals or intensities of metabolites to individual cells. We're getting what we call spatial molecular metrics, where for every cell, we have intensities of metabolites, lipids, fatty acids, drugs, like anything that you can detect with imaging mass spec. And here we're talking about multi-imaging. So then we get fluorescence readouts and integrated over cell, or it can be even subcellular fluorescence readouts. We get morphometric properties. We also get spatial features, which cells are next to each other if we're interested in cell-to-cell direction. So we can detect, basically, it's very interesting, like detection of cells is very, very similar to tissues. There are a few aspects, but basically the sensitivity, the numbers are very similar. And with this technology, we can experimentally analyze at least 1,000 cells per hour. So this is just to illustrate the idea that Luca came up with how to overlay this data. So what he did back then and what we do now in every experiment, we do microscopy once again, post-MALDI. And here we can detect the sparks or craters left by the laser. And here you're looking at data set, which is 4 years old. You see craters were pretty irregular. Oh, okay, my PowerPoint closed, but no worries, I'll restart. Can you still hear me? I hope so. Interesting.
Hi, Theodore. We can still hear
Hi.
you. Is everything okay?
That's perfect. Yes, my PowerPoint is now, I'm restarting it.
Okay, perfect.
I'll get back to it, no worries.
Yeah, let me know when you're ready.
Okay, yeah. No worries, it's getting back. And is my video as blurry as I see it of myself, or you don't see my video at all? How does this work?
No, we cannot see a video.
Okay, this may be even better. Okay, no worries. I'm getting.
Okay, yeah, now we can see your screen again.
Okay, nice.
Yeah,
okay,
it's fine.
Okay, so how much time do I have? I have 10 more minutes, right?
I think so, yeah. Last 5 for the question.
Okay, wonderful. Cool. So where I stopped? Oh, I stopped at this ablation marks. Yep.
Exactly.
Cool, thanks. Yeah, here you see ablation marks from our previous system, which was APS-MALDI 10. Ablation marks were bigger. The motor stage was not as precise as this right now, but still, it already, like, doing this additional microscopy really allowed us to match what we normally think as pixels, but actually these ablated areas with individual cells. And there is also definitely normalization behind this. If you're interested, check out the paper or ask me questions afterwards. Long story short, what was very important, not only to detect it, but evaluate single cell precision. We evaluated the image analysis procedure that we can really overlay these ablation marks with microscopy cell outlines with sub-micrometer, actually much better precision. This was good, but we still wanted to check whether we do have single cell data. So for this, another PhD student, Sergio, from our team, he developed an approach where he co-cultured 2 different cell types, and every of these cell types, they would be expressed in fluorescent protein, here RFP or GFP, red or green. These are cells from mouse fibroblasts or HeLa, human cancer cell line. So, and he also, he kind of devised it all that to make it spatially heterogeneous. We have cases like this where one cell of one type surrounded by other cells of another type, and then we ran SpaceM on this co-culture system, and we were able to show that SpaceM predict the cell type with very high accuracy. So it means we, first of all, we're getting biologically relevant profiles, metabolic profiles, which are informative of the cell type, but also what's even more interesting, we can resolve cases like this of individual cells surrounded by other cell types. So we do have single cell resolution. So now I would like to show you the results that we have achieved together with collaborators on applying SpaceM method to the problem of NASH or non-alcoholic steatohepatitis. NASH was introduced earlier today already. So this is active field of research, not only in imaging aspect, but in general, because there is, till very recently, there was no actually approved drug. There was just recent approval. There is still a lot of things to develop here. And NASH is this inflammatory stage on the transition of liver from healthy state towards liver cancer. And what's very interesting here that it is happening on the effect of fatty acids or diet, but also pro-inflammatory cytokines. So our collaborators, Mira Stadler and Matthias Heichenwälder from German Cancer Research Center here in Heidelberg, they have developed this in vitro cell culture model of human hepatocytes, differentiated, and then they use them with fatty acids and pro-inflammatory cytokines. And you can see how some cells, they accumulate these lipid droplets upon inflammation and others do not, or do it very mildly. So there is a strong cell-to-cell heterogeneity here. First, what we did, we took the cells, we put them into SpaceM, and we created metabolic profiles from every cell, and we looked at it. And for looking, we used the method called UMAP. It is sort of modern PCA analog, where if the cells are close to each other on this UMAP plot, they have similar profiles. So since we have also microscopy data, we mapped the fluorescence information here for every cell, how much lipids it has here, shown here in red. And you see that we have a distinct subpopulation of cells which have higher lipid content. And if we break it down, now we start in imaging mass spec, we start pulling markers from imaging mass spec like this triglyceride 480 and mapping intensities of this triglyceride, and we clearly see that it has high intensities in this subpopulation. So then again, we did more like summarization analysis. We found those lipids which correspond most to lipid droplet accumulation based on fluorescent. For example, this marker I've shown you previously, but also either diglyceride and other molecules. And we did enrichment analysis, finding what are the properties of these lipids here, which are mainly accumulated with lipid droplets, compared to other lipids here. So, and we clearly see the accumulation of neutral lipids, in particular triglycerides. And also shout out here, so this enrichment analysis, it is lipid ontology enrichment, which was done with a tool lipidontology.com, I believe, or .org. Just Google lipid ontology or Lion. So this is work of Martijn Molenaar, who's currently in our team, who developed it in previous lab at Utrecht. It's a really nice tool for lipid enrichment. So a bit more complicated analysis, but a really cool one, at least I think so. So this combines several conditions: control cells, fatty acids, inflammatory state with pro-inflammatory cytokine, and anti-inflammatory treatment. And we can already see that cells do change here upon the treatment, but it's impossible actually to devise how do they change, in particular upon the strong heterogeneity we see here. If we do SpaceM and we create single cell profiles which integrate 700 metabolites lipids per cell, we have a lot of cells here. Every cell is a dot. They also are orange here, so the cells close to each other, they have similar profiles. We color code them by the condition, and we clearly see that SpaceM is able to separate the conditions treated from untreated, but also inside treated cells we see differences. So, but what is even more interesting, if we do clustering, and if we cluster it in 3 groups, then we have control cells, and these cells, they actually, here treated ones, they cluster in 2 groups. One, mainly dominated by inflammatory state, the model of Nash. But we also have some cells which are actually induced only with fatty acids. We also have some treated hepatocytes which still reside in this state. So that means what's very interesting to witness here is that we're not separating just the conditions, but we really see subpopulations. Some of the hepatocytes induced by fatty acids, they actually already have inflammatory profiles, which are normally associated with the action of cytokines. So there is a lot to discover here, and in particular now we can also, by matching the differences, we can kind of interpret the action of every other treatment here. So we did the markers, and here we found the trajectory. We plotted intensity of all markers along the trajectory. You see some molecules here, lipids, they are high intensity in control hepatocytes. You see some that really rise up in this trajectory in this steatotic state. We did also validation in vitro, in vivo in mouse, Western diet. What's really cool is to see this is bulk lipidomics of homogenated livers in vitro. this is our cell model. You can appreciate how well we have reproduction of this single cell data in the bulk. And also, if we look at analysis even of data coming from plasma of patients, those markers and those ones were reported in the context of NASH for human patients. So this is really cool and shows the relevance of single cell model and single cell methods as well. I would like to come to the summary of my second part of my talk and reinforce the message and that single cell revolution is absolutely happening. And here, I think we as a community, we as method developers, but also as application scientists in imaging mass spectrometry, we have a unique tool to really probe single cells and their metabolomes and lipidomes. So we contributed to this emerging field by developing a method, SpaceM, so which works for cell cultures and has a number of advantages, and it can discriminate cell types, but what's even more important, it can discover and characterize metabolic cell state. Much more subtle and elusive concept. And also by overlaying it with microscopy, we thus link phenotype and metabolism. I would like to take 2 moments to throw some of the idea or our hope where we want to go with single cell metabolomics, and maybe invite some of you who are interested to work together on this journey. So I think what we're witnessing right now in single cell omics is a very interesting time point where single cell RNA-seq has been developed to the reference technology, very powerful and so on. And what's also interesting is introduction of AI methods, machine learning methods to really decouple, deconvolve, interpret this data. But AI also, it works the best when there is a lot of data available. And here also another direction of work was introduction of all these atlases throughout the globe, in particular in the US, which provides a lot of data to train AI on, in particular using single cell RNA sequencing. However, so these 3 directions, okay, it's not helping me. Okay, let me just continue with my voice because it's anyway
yes.
wrapping up slides.
We cannot,
yes.
Yeah, we cannot see it, unfortunately.
But you can hear me, right?
Yeah, we can hear you.
Okay, cool. Yeah, okay. So I'll just repeat, I'll just say what is there on the slide. So basically, there is single cell RNA-seq, there is AI, and there is atlases. However, single cell RNA-seq is very, very expensive. And producing atlas, this is hundreds of thousands of dollar or euro costs. AI need this data. So how can we, with a single cell metabolomics, can contribute? And here, cell metabolomics comes at absolutely unique position because it is the fastest and cheapest of any possible likely single cell omics which can be derived, because it doesn't rely on sequencing, it doesn't need any separation, it can do things on the fly. So I think we have a very bright future here to contribute to this single cell revolution with a high throughput single cell analysis, in particular in applications where this is required. So with this, I would like to wrap my talk, and I would like to thank everyone who contributed here, in particular single cell team, developments of Luca Rapes, Sergio Triana. Shout out to developers and Metaspace, in particular Lachlan and Mons, who are in the audience. Please reach out to them. Our collaborators at Temple, German Cancer Research Center, everyone else who helped us on this journey. And we also will be expanding our capacities, but also our team will have multiple positions to be open soon. So if you're interested, please do reach out to us. And thank you very much for bearing with me and bearing with a PowerPoint, which was not very helpful today. Thank you very much.
Thank you very much, Theodore. It was a great talk. We have time for one question, and there are already a couple in the chat, so you can take some time to see them afterwards. I actually have a question regarding the sample preparation. So you said that you're fixating the cells for the single cell analysis. Do you see any contamination or decreased sensitivity by doing this?
We do see some changes, of course, because what we do fixation, this protocol also published, this is PFA 4% for 10 minutes. So this is pretty common fixation in cell biology. However, what it actually does, it opens up membranes so that there will be cross-linked, the proteins inside and metabolic enzymes will be cross-linked. So you can assume that there is some leakage, in particular of some hydrophilic metabolites going on at this point. So
yeah, this is why I asked. Yeah.
Exactly. So we evaluated it. We also compared it with the freeze drying. So we do see some differences. So one should be careful about this. Most of the differences was not black and white, kind of complete disappearance, but we did some differences. Yeah.
Okay, so it doesn't, because you showed a nice image of the lipid droplet in inflated cells.
Yeah.
So it would affect that, I guess, at least a little bit, the fixation and the sample preparation. So what I'm saying is that potentially you're losing other cells that have a lipid droplets because of that. I
would assume, and our experience says that we're not losing cells. Lipids are least affected, probably. But if you want to do, in particular, quantification, like not really fold changes, but it's absolute quantification on small molecules, yes, you should be very well aware of this. And you should devise your experiment so that you can be aware of this aspect as well. Absolutely. Yeah.
Okay, thank you very much for your talk. Also, check the chat, because I think a lot of people are very keen on seeing your work towards peptides as well, not just metabolites. So check the chat and you can reply to them there. Thank you. Theodore.
Totally. I'll respond to everyone on chat. Please ask things for the questions, and also if you have more questions, just post me on chat or just approach me. I'll be lurking around in my avatar view. Thanks, everyone. Enjoy the conference. Very nice to reconnect with everyone. Cheers.
Thank you. So our next speaker is Laura Capolupo from EPFL. So welcome, Laura.
Hello, can you hear me?
Yes, we can hear you. So whenever you're ready, you can share your screen and your presentation.
Can you see it?
Yes, it's perfect.
Okay, perfect. So first of all, thank for giving me this opportunity to present. And second, I'm really happy to present after Theodore's talk, because we are kind of presenting the same thing. And I really want to convince you that single-cell lipidomics and single-cell metabolomics can be really helpful to understand some biological questions and some biological processes that are not easily understandable by standard techniques. So, as you know from also Theodore's talks, cells are part of communities where they contribute to collective phenotypes by showing different behaviors and characteristics. So cells are heterogeneous, and this cellular heterogeneity can drive important biological processes. So, and this can be easily hidden by pooled cell data assay. So in the past years, with the advent of a lot of single-cell omics methods, and with the advent of all the cell atlases, we started to understand a bit more about cellular heterogeneity. But not much is still known about lipids and whether lipids can have a role in cell fate and cell state determination. So whether lipids contribute to determination of cell states. And as a model, we decided to use fibroblasts, because human fibroblasts, they are plastic cells that can exist in multiple cell states. So resident fibroblasts can get activated to myofibroblasts, fibrolytic fibroblasts, and inflammatory fibroblasts. And the proportion of these cells are really important in the maintenance of the architecture of the skin, where you have epidermis, dermis, and hypodermis, and you have different kinds of fibroblasts in the reticular and papillary dermis according to their function. And again, while a lot is known about how proteins and growth factor can drive, how they drive fibroblast heterogeneity, it's not known whether lipids are involved. And to answer this question, what we decided to use was single-cell lipidomics by MALDI-MSI. So we used an approach similar to what Theodore showed before. And the first, after we, with high spatial resolution MALDI-MSI, the first thing we did once we get our images was a single pixel analysis, where we considered each pixel and all the M over Z molecule, all the M over Z in each pixel as independent vector to perform principal component analysis. And what we found is that the 90% of the variability of our cells was made by the first 8 principal component. And once we put back in our mass images the first principal component, what we noticed is that basically there are some lipids or some molecules that are mostly expressed in the inner part of the cells, while others are mostly expressed in the outer part of the cells. And when we looked at the loadings of the lipids expressed in these 2 parts, we actually found in the inner part mostly glycerolipids, and in the outer part mostly sphingolipids. But what was really interesting for us were these 2 principal components, 5 and 6, where we found actually that cells are different according to their lipid composition. And once we looked at the loadings of this principal component, we actually found glycosphingolipids being the most variable lipids. So we found that there are 2 major variation axes in lipid composition in fibroblasts. One is intracellular and one is intercellular. But while the intracellular lipid difference in lipid composition is quite well known, so it's well known that glycerolipids are mostly associated with the inner part of the cells and sphingolipids to the plasma membrane, this cell-to-cell variability was quite a new thing for us. So we wanted to go deeper into this discovery. And what we did was to use a workflow where our optical images or fluorescent images were used to segment mass images and to obtain single-cell lipid data that then were normalized and used for further analysis. So our first question was really to understand whether all the lipids are cell-to-cell variable or just some of them. And to answer this question, we use the coefficient of variation. And once we plot lipids from the most variable to the least variable, we actually found, again, that sphingolipids are the most variable lipid in single cell. But at this point, what was interesting to understand was whether we have co-expression of metabolic pathway of these lipids. So whether lipids co-vary in the same cells or they are not covariant. And to do that, we used the Pearson correlation. And what we did was to connect lipids that were similar among them in this network. And what we found is that, again, sphingolipids, the high variable sphingolipids, form these discrete covariation groups that are connected among them, but not with the other lipids. So it seems there is a metabolic expression of these lipids in single cell. And if you look at the sphingolipid biosynthetic pathway, this is a simplified version. These are all the lipids that seems to be hypervariable in our multi-MSI analysis. And as you noticed before, we have several kinds of ceramide and sphingomyelin and so on. This because lipids can have different acyl chain. And what happens if you take mass images of lipids with same head group but different acyl chain is what you get is this kind of really homogeneous picture. But then when you take lipids with different head group but the same acyl chain, then you get this kind of really heterogeneous picture where it seems that fibroblasts exist in specific subpopulation according to their lipid composition. And also, it's notable that sphingolipid variability is mostly accounted by variation in the head group processing into complex sphingolipids. So as I said, this fibroblast seems to exist in cell subpopulation. So what we did was to use standard approaches that you can use in single-cell RNA sequencing, where we clustered cells according to their lipid composition. And what we found actually is that cells are actually different and they can be clustered according to sphingolipids. Like if you look at the ceramide, it's mostly present in this cluster 1 and so on. So at this point, the question was actually whether we could validate these findings or we can only trust the MSI data. And we were actually lucky because glycosphingolipids that are the most variable lipids in our data are a receptor for bacterial toxins. Specifically, GM1 is cholera toxin recognized the glycosphingolipid GM1, Shiga toxin 1A the lipid GB3, and Shiga toxin 2E the lipid GB4, but also to some extent the GB3. So we stained the cells with normal approaches, with flow cytometry and fluorescent images, and again a fluorescence staining. And again, what we found is that fibroblasts exist in these cell subpopulations, so they are quite heterogeneous. Then, to definitely confirm this, we also did a correlative fluorescence and MALDI-MSI acquisition. And what we've noticed actually is that there is a very good correlation between MALDI and toxin staining. And what we concluded at this point is that, yes, fibroblasts exist in a specific sphingolipid configuration that we called from now on, that I will call from now on lipotypes. So it seems at this point that these lipids are heterogeneous, but is there any correlation between lipotype and cytotype? This was the main question of our work. And so to answer this question, we did single-cell RNA sequencing data. And what we found, of course, is, as you can find in literature, the fibroblasts are really heterogeneous. But we were interested in understand where our lipotype can fit in this transcriptional landscape. So we fact sorted for different populations according to their lipid composition. And we did bulk RNA sequencing to basically create a metagene for each lipotype that then we mapped back into the UMAP space. And what we found is that actually there is a good correlation between lipotype and cytotype, with cholera toxin positive cells or GM1 positive cells being a more fibrogenic population, and Shiga2, Shiga1 positive cells, so GB3, GB4 positive cells are mostly associated with basal and proliferative cell state. So we have this good correlation. But this is true in vitro. Is this true also in vivo, so in the skin? So our lipotype can populate distinct region of the skin. So what we did, first of all, was to take genes for papillary and reticular. We took papillary and reticular signature that are the two different layers of the papillary dermis and the reticular dermis, and we mapped them into the UMAP space again. And what we found actually is that the cholera toxin positive cells are mostly reticular, while the Shiga2 positive cells are mostly papillary. Then toxin stained our some skin section, human skin section, and again we actually found that cholera toxin positive cells mostly correlate with the reticular dermis, while the Shiga2 positive cell mostly correlate with the papillary dermis. And this can be really important when you think about skin cancer or wound healing, because fibroblasts can get activated once you have a skin cancer or a lesion. So what we did was to take some squamous cell carcinoma cancer from 3 different patients, and we again toxin stained them. And what we found is that the fibroblasts surrounding the tumors that are actually called cancer-associated fibroblasts are mostly only cholera toxin positive. So it seems that cholera toxin positive cells or GM1 positive cells are the ones that get activated and become cancer-associated fibroblasts. And this is the same thing that happens if we isolate cancer-associated fibroblasts from patients. So what you can see is again that the CAF are mostly cholera toxin positive compared to the healthy fibroblasts from the same patient. So at this point, we know that there is this correlation, and this correlation is true both in vitro and in vivo. But what happens if we change the lipid composition of the cells? Because we wanted to know whether lipids are involved in the cell fate decision. So what we did was to deplete all the cells by sphingolipids by using this drug called fumonizing B1. And we did again single-cell RNA sequencing. And what we found is that actually there's a very strong shift between control and FB1 treated cells from a more fibrogenic to a more fibrolytic population. And if you look again at the papillary and reticular signature of the skin, you can clearly see that sphingolipid depletion generates a shift from a more fibrogenic or reticular population to a more fibrolytic or papillary state. And of course, the fumaricin B1 depleted cells by all the sphingolipids. So we wanted to understand actually which was the real lipid doing the job. And to do that, we prepared human fibroblast overexpressing glycosphingolipid enzymes. In particular, we overexpressed GB4 synthase to get GB4 positive cells, and we overexpressed GM3 synthase to get choriotoxin or GM1 positive cells. So at this point, we actually could change the lipotype of these cells, and we could do, again, single-cell RNA sequencing to check whether lipids actually change the state of the cells. And what we saw is that in the control cells, you still have two different cell states, a more fibrogenic and a more basal or fibrolytic one. In the GB4 synthase overexpressing cells, or the GB4 positive cells, are mostly fibrolytic, while the GM3 synthase overexpressing cells are mostly fibrogenic. And if you look also at the signature of the papillary and reticular, signature, again, you can see that the overexpression of two different glycosphingolipid enzymes have opposite effect on cell state, with the GM3 synthase overexpressing cells being more fibrogenic and reticular, and the GB4 synthase overexpressing cells being more papillary and fibrolytic. So at this point, we know that there is this correlation and that lipids causally influence cell state, but which is the signaling pathway that is triggered by the change in the lipid composition of the cells. And what we notice is that most of the changes in our single-cell RNA sequencing data were mostly associated to the TGF beta and FGF signaling pathways that are the most important signaling pathways activated in fibroblasts and in skin. And what we found is that sphingolipid deprivation actually either promotes the FGF signaling or decrease or suppresses the TGF beta signaling pathway. We looked at two specific protein for TGF beta, ACTA2, and the FGF signaling pathway MMP1 in our overexpressing cells, and we actually see that there is this change. So with the GB4 synthase overexpressing cells that are, as you remember, the more papillary and fibrolytic one being mostly MMP1 positive, so they seem to activate the FGF signaling, while the GM3 synthase overexpressing cells that are the more fibrogenic or reticular one are mostly ACTA2 positive, so they actually suppress FGF signaling pathway and promotes the TGF beta signaling. So this means that the overexpression of two competing glycosphingolipid synthetic enzymes can have opposite effect on cell state and also on cell signaling. So just to wrap up and conclude, what we understood is that some lipid metabolic pathways are cell-to-cell variable, that cell-to-cell lipid variability is sufficient to define discrete lipotypes that correspond to cell state, that lipid composition causes cells to respond differently to identical stimuli, leading to distinct transcriptional states, that this is non-transcriptional. I'm sorry I didn't have the time to show all the data, and that lipotype configuration are reflected both in vitro and in human skin, and this might be important in tissue patterning. And finally, I hope that we demonstrated the potential of single-cell lipidomics to reveal biological phenomena that cannot be studied using a classical lipidomics or single-cell transcriptomics approach. And with that, I just want to acknowledge all the people involved in this project in the past years, and of course, all of you for the attention. And I'm happy to take questions.
Hi. Thank you very much for your talk, Laura. It was really interesting. And yeah, so you've done this experiments only in fibroblasts, right?
Yeah.
Any plans to expand this methodology to other cell types and potentially correlate interactions with fibroblasts over these pathways that you found to be activated?
Yeah, okay. So first of all, I need to say that lipids are actually heterogeneous in a lot of cell types. We know that by the use of our toxins, but this is of course only related to the glycosphingolipid at this point, but also other lipid configuration that can exist. Second of all, yes, we are trying to expand our methodology. The main disadvantage is, of course, spatial resolution, again, because fibroblasts are big cells, easy to analyze. But maybe with space M, we could try to get something better also for other cell types.
Okay, yeah, good luck with this. I think we have a question from the audience. So it's great talk. Is cell-to-cell lipid variability somehow correlated to cell communication?
Well, we didn't find any anything that tells us that this is related to cell communication. But I mean, glycosphingolipids are on the plasma membrane, and they're glycans, so they are used to communicate with cells, but we didn't find anything to prove this.
Okay. Okay, and at this point, I would like to thank Laura again and all of you for staying with us in this session. And also to remind to the MSIS members that 12 to 12:30, we will have the MSIS general meeting. So members of the society should have gotten a separate email with the Zoom link to join. And yes, thank you very much. And yeah, we will continue afterwards. Thank you.
Hello everyone and welcome back to Lecture Theatre 1. My name is Robert Bradshaw. I'm a lecturer and researcher at Sheffield Hallam University, and it is my pleasure to introduce our lunchtime seminar, which today will be given to you by Brukker.
Yeah, thank you. Can you hear me?
We can.
Okay, great. Then let me share my screen.
Okay, we have that on screen now.
Hello.
Yep, we can hear you. Good to go.
Yeah, perfect. Can you see my screen?
Yes, we can.
Perfect. So it's all okay to start?
Yeah, go ahead.
Perfect. So first of all, thank you very much for this opportunity today. As you can see, I'm having a call at the moment, so I'll try to speak as loud as possible. So today's talk is about an application without using the multitude Tinto Flex platform for the assessment of the distribution of isobaric androgen in target tissues. And in its case, we use a murine xenograph from prostate cancer models using chemical derivatization approaches. So the main topics of today's talk is to talk about the androbiology in prostate cancer, so why it's so important to measure these androgens in tissue and also in the circulation, but obviously we want to focus on tissues. And then the isobaric challenge, what we call in the field regarding the androgen mass spectrometry imaging challenge that we face. Some things about teams and multitude, but this was pretty well covered by the previous presentation. And then the method development that we done, and obviously this was a proof of concept study, and then we'll discuss the findings and the final remarks of the study. So something to the biology of prostate cancer. So we all know that castrate-resistant prostate cancer is CRPC, is a more lethal form of prostate cancer. These androgens, the active androgens like testosterone and DHT, obviously they use DHEA as a precursor in a human model. This has been shown that using androgen deprivation therapy to deplete this level of active androgens like testosterone and DHT improves the disease, but it hasn't been found and shown in the literature that after the androgen deprivation therapy, the retention of this active androgen signaling was retained even in tissue and the circulation. And so the researchers were hypothesizing of the presence of the different androgen metabolic pathway, or they call de novo pathway, from different precursor sources. So the study of endocrinology, but was pointed out by Lavrier lab in Canada, is basically the field to study the ability to maintain these testosterone and DHT active androgens level in tissues, but study what's happening at tissue level. So that was the endocrinology basically studies. So unraveling this tissue-specific metabolic pathway to see why after the androgen deprivation therapy, some of the patients still have high level of testosterone and DHT. So unraveling this tissue-specific metabolic pathway to see what happened at the tissue level, at the leach into the circulation to keep this testosterone and DHT level low, basically could be a key to the development of a new therapeutic target for this more lethal form of prostate cancer. So regarding mass spectrometry imaging, so we call the isobaric challenge, we all know that androgens have poor ionization efficiency in mass spectrometry. I mean, in particular in electrospray and MALDI, and they have really low endogenous level in tissues and circulation. So we're talking about some nanomolar. These compounds are really potent biologically. And then there's different derivatization strategies to enhance the signal intensity and also improve the signal to noise haven't been used in the past by different groups for a long time. But the isobaric interference remain an issue. I mean, these are the two examples that today's talk. So testosterone, which is an active ligand, what we call an active androgen, and DHEA, which is an inactive androgen and also the precursor. So the only difference in this molecule structurally is replacing a ketone in the C3 steroid core ketone to the OH in the DHEA, and also here in the carbon-21, the OH is basically turning to ketone. So the different derivatization strategy that have been used in the past for different groups and researchers, they use some what we call charge derivatization reactions like a Girard, T and P, and then some really charged reaction like all of these here. But the problem remains that after the derivatization, all the collision-induced dissociation and different dissociation mechanisms, they end up in the same daughter fragments. So it's impossible to separate this by using collision-induced dissociation or fragmentation-based approaches. And also, in terms of the collision cross-section in gas phase, these two molecules are really close to each other in terms of the structurally. So the tiny difference in an OH and a ketone won't be enough to do a proper separation by collision cross-section. So adding a derivatization reagent will change the conformation on the native testosterone and DHEA that will allow us to use the ion mobility to separate this derivative using the collision cross-section in gas phase. So our derivatization strategy was to use a charged moiety first, using the Girard. They are well known that works really well in MALDI and electrospray. And also we added really charged moiety, which is this case of dancyl hydrazine, as you can see here. So that's the Girard, basically adding a positive charge. And that's the dancyl hydrazine, which basically is a really ionizable tertiary amine in here. So both reagents cause conformational changes to the androgen steroid core. So hopefully there is potential there to be separated by the collision cross-section using ion mobility. And you can see in the right-hand side, these are the off-tissue spectra of testosterone and DHEA. So you can see that the only difference is the site of derivatization. So one is on the ketone on the carbon-21, and the other one is on the ketone on the carbon-4 in the steroid core. So that's the only difference. As you can see, the spectrum, both protonated mass and sodium adult, are completely isobaric. On the other hand, the dancyl hydrazine derivatives, you can see on the testosterone, there's a protonated mass and the sodium adduct. And the DHEA, the only difference is they are forming a radical cation, and there's isobaric sodium cluster. You can say that, yeah, this can be separated by mass using high resolution, but the problem we are facing is then you have an interfering contribution from the isotope C13 from the DHEA coming to testosterone. So unless you're using super high resolution, it's really challenging to separate this by using high resolution. So having a platform that can separate these based on the collision cross-section using ion mobility is going to be ideal. So as I said, for the mass spectrometry imaging platform, we use the MALDI TIMTOF Flex, so which the MALDI provide a signal enhancement for really charged species by the MALDI post-ionization process, and also the isobaric species can be separated used by the collision cross-section in gas phase, and also have the high mass resolution using the latest TOF technology. So I won't explain how the MALDI TIMTOF work because this has been addressed by a previous presentation quite well. So this is just to point out the platform we use. In terms of the workflow, as I said before, as a tissue, we use Helen mouse xenograph, dosed with DHEA. The reason why we dose the mice with DHEA was the mice cannot produce DHEA, that is being produced by the human adrenal gland. So in order to mimic what's going to happen in a human model, we use DHEA, obviously, and it could be some precursor of the synthesis of the testosterone and DHT. So regarding the workflow, is more like the standard workflow in mass spectrometry imaging. So the sample collection, the embedding and tissue sectioning. And obviously, we done previously on tissue chemical derivatization using GR and dancing hydrazine, and then the matrix application using CATA and DHPA. And obviously, we run this using the TintoFlex. So preliminary data of tissue show here the GRT DHT mix. So you can see that testosterone and DHEA were successfully resolved by collision cross-section, and either the protonated, the pseudoprotonated mass, which is the charged moiety in this case, and also the sodium cluster. And also using dancing hydrazine as a derivatization reagent, we're able to separate this based on the collision cross sections. You can see the protonated mass, the radical cation, and also as the sodium cluster. You can see here that some of the protonated mass, and some of, in this case, on the testosterone, there are more than one signal. That could be due to the trans isometric structure that they are now appear after the derivatization reaction that put the carbon nitrogen double bond into the sterile moiety. So you will have this 6 isomeric trans isometric structure here as well. Regarding the multitude, we done an assessment using the 2 derivatization reagents. So one in using the GR, as you can see there, testosterone and DHA GRT, using MALDI-enable and MALDI-2 disable. So, so there is no much of improvement in terms of signal intensity, and that is due to these are already charged species, so they don't really benefit for a post-ionization process. But and on the other hand, the dancing derivatives that they are readily charged species, so the signal was enhanced by 20 times by using the post-ionization by MALDI-2. And then we correlate this on the imaging mode. So you can see the downstream DHEA and downstream testosterone, they were enhanced the signal, you could see before in enough tissue. So even in imaging mode, the signal was enhanced by 5 to 10 times. And then, we ran our samples on the tissue xenograph model, and for the first time, we were able to have the distribution of these two isobaric hydrogens as a GR. We choose the GR derivatization reagent because they have a better separation as a sodium cluster in terms of the 2 species, as you can see on the left right side. And then we analyzed our tissue in question. We could see there were co-localization of both androgen observed in the tissue, which is quite good information in terms of the biology. And also on top of that, we done H&E staining to perform which cells that were involved in the de novo this pathway that do produce the active testosterone again. So what we found that these 2 androgens are co-localizing each other. It was basically saying that it could be one of the precursor or the other one. And that will explain why in the androgen deprivation therapy sometimes cannot work in some of the patients, that they were able to retain these signals and then created the active ligands by the DHEA precursor, or it could be further studies that we need to do in different models that could be also coming from cholesterol. So the remarks of this project, that the mass spectrometry imaging with on-tissue chemical derivatization is a powerful new tool to study the distribution of this derivatized isobaric androgen within tissues. So for the first time here, we have demonstrated the ability of visualization of testosterone and DHEA, so two biological relevant androgens, because one is active, one is inactive, and it's been really hard in the field to separate these two isobaric androgens in the past. So also we have demonstrated the utility of this method of detecting endogenous concentration within the mutant xenograft tissue, and this is a key model for prostate cancer intrakinology. So as I said before, the testosterone and DHEA are a really, really low concentration. So not only we're able to separate this by the collision cross-section, and all we're able to detect these low-level endogenous androgens. And obviously, this opens up different avenues and offer different opportunities to maybe target different steroids like glucocorticoids, or even the low-level estrogens as well, using different derivatization reagent approaches to see if that conformational changes is enough to separate these potential steroids by the collision cross-sections. That otherwise it's been difficult unless you're doing liquid chromatography or other techniques. So I want to acknowledge some of the people that were involved in this study, like Logan McKay and Faye Grushank from CIRCAMS at University of Edinburgh, and also Sen Solrich and Klaus Dreiswer from the Institute of Hygiene in Moscow University, who provide the equipment and the expertise to run the samples, and also Brian Haynes from the Center of Proteomics of Leiden University in the Netherlands, Carl Smith from the Mag Lab in Tallahassee, Florida, and obviously Brukker and Annika News for input and support. So thank you very much for this presentation, and I will take any question you may have about this presentation. Thanks for all.
Thank you very much for that presentation. I think you really did show off there the power of TIMS. I don't think we've got enough time for questions, but I'm sure people can ask you questions if they would like to contact you separately. For our audience that are watching this, you might have noticed that Janina appeared, then disappeared. It's because our people that are coming in from Brukker today are signing in via the same link. So we're going to try and get Janina back now, because when one person joins, it kicks the other person out. So hopefully we'll be able to get Janina back for her presentation. And then we can continue with Brukker's lunchtime seminar.
Okay. That's perfect.
Thank you.
Thank you very much.
Hi, Janina. Welcome back. Sorry, we cut you off a bit short there, didn't we?
I'm sorry. Yeah, sorry for the confusion. I think the problem was that we got the same credentials to log in.
Indeed. That's okay. At least you're back now and you can deliver your presentation to us.
Yeah. Okay, let me share my screen.
Thank you.
Okay, you should be able to see it now.
Yes, we can see that if you just put it into a presenter view mode.
Yes. Okay, good. Yeah, so then...
Hi, Janina. Welcome back.
Thanks.
Sorry, we cut you off a bit short there, didn't we?
That's okay. I'm sorry. Yeah, I'm sorry because I'm still having...
Janina, could you please turn off the stream audio because we can hear it in the background?
Yes.
Back now and you can deliver your presentation to us.
Okay.
Thank you.
Now it should be fine.
That's great.
Okay, so thanks for joining us at the lunch seminar, and thanks Diego for the presentation. I would like to give you now a few insights about what is new in spatial omics and MALDI imaging from a Brukker perspective. And you have heard about MALDI-2 now a lot, so I just would like to remind you that you can increase the sensitivity with MALDI-2 for many compounds in imaging. So here you see our software and a typical MALDI-2 lipid imaging spectrum. You see many compounds in the low mass range. And when you compare this to the regular MALDI-1 spectrum, then you see that they are mainly the lipid bulks present. So there's a lot more going on here with MALDI-2. And in our software, you just have to switch on the post-ionization to enable MALDI-2. Yeah, but we as a vendor, we do not only want to provide you with instrumentation, but we would like to make your research life a bit easier. And that's why we would like to offer you as well our consumables and software. The Flex matrix here stands with its purity for robust results. We have the IntelliSlides that enable automated workflows, and I would like to show you this in the next slide. We have also a user-friendly software such as Skills Lab and Metaboscape that help you with interpretation of your results. Yeah, here's a Skills Lab workflow shown, which is now available for the timsTOF Flex and already there for the RAPI Flex. It's enabled by our IntelliSlides. And what you need to do this workflow is a prepared slide, a scanned image of the slide before matrix acquisition, and then the software is doing mainly the rest for you. It selects the sample image based on the barcode. It scans the reference fiducials, these little engraved fiducials here for automated teaching. The software does the target profile calculation, it does automatic laser focus tuning, and automatic M/Z calibration, and also detects automatically the tissue. Then, after tuning the laser power, you're ready for acquisition. And this is how it looks like in our software. This is the acquisition software again. Here you have to load your sample and the geometry of the slide adapter. You can then select an acquisition method from our default application methods. Start Flex imaging, then a new wizard pops up where you can select the Skills Autopilot workflow. Here you can then select the slide position and then type in a name for your imaging run and select the directory. Finally, set the raster width that you would like to. And while you are typing these things in the acquisition software, the barcode is read and the IntelliSlides are scanned for the automatic teaching, for the scanning of the fiducials. Then you can afterwards do a performance check like target profile determination, focus adjustment, and MZ calibration. And again, in the acquisition software, the profile determination takes place. And here you see now the laser focus adjustment, which is performed on a matrix. and then you do automatically the M/Z calibration here. After this is finished, you just go back to Flex Imaging, finish the process, and the software will then automatically detect the outline of the sections. You can adjust eventually the laser power and start your run. And all this is quite convenient and nice, and you do not have to interfere that much with the software anymore. So do not introduce that many errors. Yeah, but we envision for the timsTOF Flex instrument the spatial omics workflow. You have heard about this from yesterday from Corinna already. Since we have the MALDI capability and the ESI capability, we can do multi-guided spatial omics and define regions of interest based on the molecular content of your section in a contextual manner, and excise these little areas and do laser capture microdissection and omics experiment from the little sample amount. And since the instrument is very sensitive, this works really nice. And when you're interested in that, please read our application note, LCMS 166. You can find it on the Brukker webpage. Go to mass spectrometry and then literature room and just type in the name. And I would like to make you aware that we will soon have also the skills region mapper to support the spatial omics workflows. Here you can now use a skills lab to define regions of interest, and the coordinates of these regions can be transferred through this region mapper tool to your LMD device. And here we support Leica LMDs and the Zeiss Palm Microbeam. When you're interested in that, please talk to Corinna and to Tobias Boskamp. They are both here at the conference. Another software which supports the spatial omics workflow is Metaboscape. We use the software to automatically annotate lipids and metabolites from imaging runs. In this case here, we have done beforehand a lipidomics experiment, and we have created a list of annotations from the same sample, from a homogenate of the sample. And the annotations are then based on retention time, exact mass, MS information, and last but not least, the mobility values, so the CCS values. And this is really nice because when you do then the imaging experiment, you can use this list, this lipidomics annotation list, let's say, to annotate the compounds in your MALDI imaging experiment. And here you have then not only the exact mass, but also the CCS information. And also this is summarized here in an annotation, an application note. But what you can also do is on tissue, MS directly on this instrument from tissue and exploiting the MALDI functionality. Here in this example, I'm showing you now two lipids that are very nearby in the MS domain, but we can nicely separate them in the TIMS domain. And if you would like to isolate these or one of the compounds, normally a pool would not be able to isolate these two compounds separately. But since you have now the mobility information, you can extract the MS spectra of each of the compounds and use then the MS information in Metaboscape to annotate these compounds. And then you can plot the images again back in Skills Lab, and you see in this case they are even differently distributed in the brain. So this is a quite nice example here. Then I would like to tell you shortly some of the news that are implemented in Skills Lab 2022A, which is available soon for all Skills Lab Pro customers. We have here now the feature table, so formerly known as peak table. Then the feature table, we have also now not only the M/Z, but also the mobility information included. And all the statistical tools can now work with these feature tables, including the mobility information. And last but not least, I would like to draw your attention to the QPath integration with Skills Lab. QPath is an open source pathology tool. And here you can use Skills Lab to annotate regions based on your H&E stain, for example, and then load these regions back into Skills Lab. And when you're interested in that, please watch our tutorial video on YouTube. You can just type in QPath and skills and you will find it. Yeah, that's what I wanted to tell you. And I would like to thank you now for your attention. Yeah, and please visit us at our booth.
Thank you very much, Janina, and thank you for dealing with our technological issues that we had at the very start. So thank you for the presentation.
Thank you. You're welcome.
If anyone has any questions for Brukker, I'm sure you can contact them separately. But in the interest of time, we now need to move on because our next session after lunch is starting. Okay, so I'd like to introduce myself. My name is Dr. Robert Bradshaw. I'm a lecturer in analytical science and researcher at Sheffield Hallam University, and I will be chairing this session for you on our short oral presentations. We have 7 really exciting and very different presentations today. Our first speaker is Maria Anderson, who is joining us from NTNU. Welcome.
Hi, do you hear me okay?
We can.
Good, then I will start sharing my screen. Do you see the presentation right now?
Yes, we can see that.
Okay. And how about, is it in the right mode?
Just bear with me 1 second. Yes, you've got it up now.
Yes. Okay,
go ahead.
Then I will just start right away. Yes, thanks. So what I'm presenting for you is the results from a storage study where we wanted to identify the most storage condition for metabolite detection with multi-MSI on prostate tissue. Regarding this group of analytes, there are 2 main challenges. One is that small polar metabolites tend to delocalize very easily on top of tissue sections. Secondly, they are sensitive to metabolic degradation. So, for example, ATP hydrolyzes very quickly into ADP and AMP if not stored correctly. Further, I am a member of the prostaomics group where we do a lot of work of multi-omics profiling of prostate cancer tissue. This is a logistically very challenging project with many different analyses included. So for this, we needed to know how long we could store these tissue sections for. Tested 3 different storage conditions. One was regular freezing at -80. The other one was vacuum drying, vacuum packing, and storage at room temperature. And the last one was combining this vacuum processing with freezing at -80. This setup included 4 different samples, 3 storage time, of which we have analyzed 2 so far, and 3 technical replicates for each parameter. All sections were covered by NEDC matrix and measured with MALDI-TOF in negative ion mode. We investigated analyte delocalization by looking at the small polar metabolites, citrate and taurine. If you look carefully, we can observe that both vacuum storing methods had showed a much more refined localization to the different tissue compartments and therefore performed better than just freezing. Looking at the ATP levels, both the frozen and vacuum frozen method had similar high levels of ATP and less of ADP and AMP. In contrast, slides stored at vacuum and room temperature had depleted ATP levels, which seems to have degraded into ADP and AMP. Some metabolites appear to have been more stable than others. For example, citrate showed no big changes for neither storage time or condition. Similar for glucose, whereas taurine levels was not affected by storage method, but may be affected by storage time. To conclude, vacuum storage at -80 is the winner, and in fact we have already used this storage method for our large sample set. In this project, we'll continue to measure the last time point sections and also investigate for identified metabolites and lipids. Thanks.
Thank you very much for that presentation, and quite clear you showed some big differences between the different storage conditions there, which is very interesting. I'm just looking at the chat and I can't see any questions, so I'll ask one myself. You mentioned that you're using an NEDC matrix. I don't think I've heard about that before. Can you tell me a bit more about it, please?
Yeah, I can't remember what it's for. I'm sorry for that. But it's become a very popular matrix in negative ion mode. I started out with 9AA, but we found that NEDC was gave more sensitive detection of small metabolites.
Fantastic. Yes, I can't see any questions in the chat, so I think in that case, I'd just like to thank you for your presentation, and we'll move on to our next speaker. Thank you very much.
Thank you.
Okay, everyone, so our next speaker is Emma Sisley. She is joining us from the University of Birmingham. Welcome.
Hi. Can you hear me okay?
I can hear you perfectly.
Lovely. In that case, I shall share my screen.
Thank you.
Can we see that okay?
Yes, perfect. Thank you.
Lovely. Okay. Hi,
Go ahead when you're
I'm
ready.
going to be talking about mass spectrometry imaging and identification of ligand-bound and metal-bound native proteins. Our group is interested in combining the benefits of native mass spectrometry with mass spectrometry imaging to visualize the spatial distribution of proteins and their interacting partners. Here I present native imaging and identification of endogenous protein ligand and protein metal complexes directly from tissue. These complexes were spatially mapped using nanodesi, which is a liquid microjunction-based sampling technique, and identified by top-down experiments. 21 kilodalton protein of interest is shown here. It is observed in the cortex, hippocampus, basal ganglia, and thalamus of the rat brain. A native top-down fragmentation spectrum was acquired using HCD, which identified the protein as R3 with an N-terminal myristylation. However, there is a difference of 443 daltons between the measured and theoretical mass of R3. Upon reading the literature, it was found that R3 binds to GDP, which has a mass of 443 daltons. To confirm the identity of GDP, an MS3 experiment was performed. However, using nanodesi, we switched into negative mode and isolated the 7- ion of R3 with GDP. Performing MS2, we observed 2 6- peaks and 2 corresponding singly charged peaks at 442 and 362, assigned as GDP and GDP minus a phosphoryl group. The 442 peak was isolated and MS3 performed to obtain the spectrum shown. The results confirm the identity of the ligand as GDP. The fragments labelled in red correspond to the shown cleavages on the GDP structure. So we were also able to identify and image distribution of metal-bound proteins. Here we can see parvalbumin alpha bound with 2 calcium ions distributed in the cerebellum, and carbonic anhydrase bound with a zinc ion with a stronger signal in the corpus callosum. So in conclusion, we've been able to identify proteins with distinct spatial distributions. We've used top-down fragmentation to identify these proteins, including metal-bound proteins. And we've also been able to implement MS3 with nanodesi to identify GDP bound to R3, as well as GTPase RAM, which is a protein I didn't talk about here. So with that, I'd just like to say thank you to all the people involved in this project, especially Helen Cooper, Oliver Hale and James Hughes, as well as thank you to our funders and AstraZeneca for providing us with the tissue we used in these experiments.
Thank you very much, Emma, for that presentation. Really nice applications of nanodesi there. I'm just looking to my chat again and I can't see any questions. So again, I'll ask you one myself. I don't think I might have missed it. I don't think you mentioned the resolution, lateral resolution you're working with nanodesi in those images you showed. And how long would it take for you to analyse one of the samples that you were showing as well?
So the resolution that I showed our images at today were at 200 micrometers. And then the time it takes obviously depends on the size of your tissue. So to do the rat brain, it tends to take most of the day, it's probably about 4 to 6 hours at the moment.
Okay, thank you very much. Again, I'm looking to the chat and I think that there aren't any that are coming through for us. So I guess we'll leave you there, Emma, and say thank you very much for your presentation.
Okay, thank you.
Okay, our next presenter is Michael Taylor. He is joining us from Pacific Northwest National Lab.
Hi,
Michael.
Can people hear me?
Yes, we can hear you. If you'd just like to share your presentation.
Just bear with me for a second.
No problem.
Hello.
Hi there. Sorry, I'm just listening in my ear to the instructions. You can move your mouse to the bottom of the screen and you should see...
Not sure if anyone can hear me.
A toggle which says screen sharing. Right at the bottom of your screen.
Hello.
Hello, can you hear me? Okay, I think the computer is currently muted. Can anyone let him know his computer's muted? I don't think he can hear me at the moment.
Okay, you can hear me.
I'm getting a thumbs up. Yeah.
I'll just get started.
Okay.
Okay. Okay. Screen sharing. Okay.
There we go.
Hi, I'm Dr. Michael Taylor from PNNL. So my talk is about the LACI source we've been developing. So LACI uses a near IR laser to ablate water in situ, along with neutrals, which are then post-ionized. So it's a very powerful ambient analysis method that can sample tissues in their native state. So this is the diagram of the LACI source. So there's two analysis modes, high single cell analysis and in multi-order imaging. So the key take home here is that we have a camera that can ID cells and tissues, and also a dual laser microscope lens to perform LACI or microscopy. So this is a slide showing single cell analysis mode on onion epidermal cells. So we can focus the laser to a spot size of 40 microns. We then take a mosaic image of the tissue. We can then identify cell centers for targeted ablation and then ablate a series of cells sequentially. So in our first paper on this system, we combined our source with a drift tube eye mobility system to measure accurate mass and CCS, i.e. the size of a molecule. And the right figure shows a spectra from a single onion cell with corresponding metabolite-rich mobilogram containing species, including cellotrios. So by screening multiple cells in high throughput, we can measure metabolite variance across the cell population and calculate metabolic noise. And we can see here that the metabolic noise for the detected is higher than the technical noise of the detector, and populations of cells can be identified. So our source is highly adaptable to other detectors, shown here on an Orbitrap. So here we're screening a 200 cell set in high mass resolution using optical selection. So we can identify sugar species as well as secondary metabolites, including malate and malic acid in high throughput, along with their variants. So as I mentioned, we can perform MSI using our system. So in this study, we imaged a leaf from a nerve plant from the pigmented regions. And you can see here that the metabolites correspond to physical structures within the leaf. So this is the highest lateral resolution to date reported for a LACI imaging study at 40 microns with no oversampling. And finally, we're developing a fluorescence train into our system for multimodal imaging. So here you can see optical images here from GFP labeled rhizobia within soybean nodules. And you can clearly see, using our camera system, you can see the infection zones where the labeled bacteria are populated. So thank you for listening to just one of the exciting developments we're working on at PNNL. And I'd like to thank my PNNL team and our collaborators at George Washington University, not forgetting the DOE for funding this project. So thank you very much.
Thank you, Michael. Can you hear me is my first question. I don't think Michael can hear us, everyone. So in that case, I think we're going to have to move on to the next presentation. Maybe he's muted, unfortunately.
So I think I'm having a bit of trouble with the microphone on my end. I don't think I can.
Yeah, I don't think we can ask any questions. That's okay. We'll have to move on to our next presenter. And anyone that's got any questions for Michael Taylor can send them through to him, I'm sure. Okay, I would like to introduce then our speaker. We have Michael Woolman from the University of Health Network. Hi, Michael.
Hello.
We can hear you. Would you like to open up your presentation?
Yeah. Okay. Can you see that?
Yes, that's great.
Okay. Okay, so good morning or afternoon, depending where you are. My name is Michael Woolman, and I'll be talking to you about picosecond infrared laser mass spectrometry for rapid determination of pediatric brain tumor pathology, fairly rapidly. So the CNS tumors, I'm mainly looking at the three most common: medulloblastoma, ependymoma, hepatocytoma. And beyond that, these are the genomic subgroups, which have more recently been, being studied. These subgroups have distinct clinical outcomes, but critically, they're not all interoperatively. So it takes DNA analysis to identify these, and in most of these tumor cases, you don't get preoperative biopsies. So generally, surgeons are treating all of these genetic subgroups the same way. We aim to get information intraoperatively using a picosecond infrared laser system. You've probably seen similar technologies around the place. We have a little laser. We pointed at the tissue, ablate a bit of tissue, and the plume of material, ideally these intact tissue molecules, are sucked into the mass spectrometer for analysis. This can be done in vivo or directly outside the operating room. Either way will be considerably faster than the current standard of care. And as these subgroups aren't identifiable morphologically, you have to do it that way. So I disable that, apparently not. The first thing I've done was build a brain tumor model. So this is just a PSA-LDA of the main subgroups, tumor types, the ependymoma, medulloblastoma, and hepatocytoma. As these are easily separable, it's not surprising we've got high sensitivity and specificity for this. We then expanded this to include the four main subgroups of medulloblastoma. So these are the four currently most clinically relevant ones anyway, as well as the four ependymoma subgroups. So medulloblastoma subgroups, we had enough to build the model as well as validate it. So we've got 98% sensitivity, 99% specificity. The ependymomas, we had enough points from two of the subgroups to build, but not enough to do proper validation as of yet. Although, given the separation that we're seeing in medulloblastoma, which is more tightly clustered, it seems very promising. Now, the imaging side of this comes from some of the misclassifications we were seeing in this model. We used some post-ablation H&E, and we'd seen that when we were getting misclassification, it was in areas with high stromal content or even health application. So we adapted a commercial DESI stage to hold our laser fiber and our collection tube, and this allows us to generate images kind of like what you see down here. We're still working on optimizing some of the conditions for this. The resolution right now is a little bit low, but as we improve things there, we can hopefully adjust our models and account for some of these heterogeneity effects. And that's a bit of my time. I'd like to thank my lab members and all of my collaborators, especially those at SickKids who supplied a lot of the tumor banks for this project. So the tumor samples for this project. Thank you.
Thank you very much, Michael. That was a really fascinating talk, and some of the numbers you were getting out for sensitivity and specificity are really amazing. I guess looking at the chat again, there's nothing coming through to me, so I'll ask you one myself. What would be the next steps in this research for you? Like, how can you take this further?
I think I'm still sharing my screen for some reason, but anyway.
If you click stop sharing, then it will take that off. That's it.
Anyway, next steps would be to so there's a couple of other subgroups of ependymoma that we haven't been able to access as of yet. I'd like to add to the model. We'd also like to do some more further validation on the ependymoma samples. But the main aspect in terms of the imaging side was really to basically bring some of the get profiles for some of the heterogeneity, like some of the healthy brain or just stroma and necrosis, things like that. Build them into the model so that we can hopefully reduce our classification rate even further.
Fantastic. Thank you very much, and thank you for your fascinating talk today.
Thank you.
Okay, I'd like to introduce our next speaker. We have Sebastian Bessler from the Institute of Hygiene with us today.
Hi, can you hear me?
I can hear you.
Perfect, then I will start screen sharing. Are you able to see the slides?
Yes, we can see it, thank you.
Okay, then I will start. Age-related macular degeneration is one of the most common causes for blindness in the developed world. There are two types of AMD. There's dry AMD and there's wet AMD. Wet AMD is characterized by neovascularization, meaning abnormal blood vessel growth, which could lead to hemorrhage. Our goal was to investigate lipids and metabolites in wet AMD at single-cell levels. For this, we used laser-induced choroidal neovascularization, which is a common mouse model for wet AMD. Here, rust membrane is ruptured using concentrated laser shots. This leads to neovascularization inside the retina. The eyes we used were harvested 7 days after laser treatment and cut into 10 micrometer thin sections onto ITO slides. We then used transmission mode MALDI-2 MSI coupled to an Orbitrap mass spectrometer to achieve 2 micrometer pixel sizes to be able to resolve even single-cell layers like the retinal pigment epithelium. As the matrix, we used dihydroxyacetophenone, which was applied via sublimation. Here you are able to see some of the resulting MALDI images. Using 2 micrometer pixels, we were able to nicely resolve all retina cell layers. We were also able to see some signal, like the signal in the middle, that were highly localized inside the CNV region. To further investigate these samples, we used MALDI data-dependent acquisition, where we used 2 micrometer step sizes in the X direction and 4 micrometer step sizes in the Y direction to essentially create 4x4 micrometer pixels with MS-MS data. For visualization and annotation, we used LipoStar MSI. Using the MS-MS data from the DDA measurements, we were able to automatically identify 43 unique lipids that ranged due to the MALDI-2 post-ionization over a broad range of lipid classes. But these annotations were often done only by one diagnostic fragment, and at least in positive ion mode, we were not, or most of the time, not able to identify side chains. This MS-MS data also let us identify the unknown signal as retinal palmitate, which, together with retinal stearate, localizes on both sides of the CNV region. These retinal esters are the storage form for retinol, meaning vitamin A, and ionize under MALDI-2 conditions as radical cations. We hypothesized that these retinal esters accumulate in RPE cells that were not destroyed during the laser treatment. Here, retinol is no longer consumed by the visual cycle, and because of this, its storage form accumulates. This could have negative impacts on the CNV process because retinol is known to exacerbate CNV. To conclude, we were able to measure mouse retina with laser-induced CNV at single-cell resolution using T-MALDI2-MSI. We were able to automatically identify 43 unique lipids using MALDI-DDA with an effective pixel size of 4 micrometers. And using the MSMS data acquired during DDA, we were able to identify signals inside the CNV region as retinal esters. Thank you much for your attention.
That was a really fantastic talk. Thank you, Sebastian. I'm again looking to my chat and I can't see any questions there, but I do have a couple for you. I found the MS and MSMS imaging together really fascinating, but my first simple question would be, at such a high resolution, does this give you absolutely ginormous data sets?
Yes and no. So the higher, or the Orbitrap data in itself isn't that big because the sample area we are able to measure is quite small. So we took around 3 pixels per second, and this measurement took around 10 to 18 hours. So the resulting image data is, I think, around 4 gigabytes. So not that big at all.
Yeah, not big in today's standards. I must say. And my other question was that I heard you mention that your sample preparation involves sublimation, but I missed the matrix that you were using.
DHAP, so dihydroxyacetophenone.
Lovely, thank you very much. I think that's all of my questions here. And oh, hang on, I can see a question in the chat from Tina.
Yep.
Was it 1 peak per MSMS pixel?
Yes, we used the top 1 experiment with an exclusion range or exclusion time. So always the highest peak was fragmented, and then it was excluded for 30 seconds.
Brilliant. Thank you very much, and thank you for your talk today.
Thank you.
Okay, we can move to our next speaker. We have Peter Marshall from GA. Hi, Peter.
Well, hello, and good afternoon, everybody. How's it all going? This is the scary point now to get something working. So let me know when you can see the screen. Is it entire screen or window?
Screen, please. Entire screen.
Entire screen. Okay, that doesn't allow me to share that one, but it's grayed out. Single window I can work. Shall I go with that
Try
one?
single window.
Can you see anything now?
That's great. We can see that.
Okay, so there's a top tip then. It's not entire screen. Okay. Well, first failure. So hopefully I can now manage to get the rest of my talk in the time allowed as well then. So, okay. Well, thank you. I'm going to spend the next few minutes talking about a wonderful collaboration with Jens and Klaus in Munster, looking at MALDI-2 on a wide range of compounds to see if we can get them to ionize much better. And it should advance. There we go. There's oftentimes when you haven't got sufficient sensitivity to detect your compounds. This is usually because of the low ionization efficiency of the drug, or actually the molecule itself, its properties that don't endure it to ionize equally. So we took 1,200 compounds and we looked at them on MALDI-2 to look at enhanced detection. They were 1,200 different chemical structures and also very different physical chemical properties. They all also had to be publishable. Made them up at 10 micromolar concentrations in methanol and spotted them on as 1 microliter droplets, and then used DHAP matrix. So here's a schematic of what happened. There's 1536 bottles made into solutions. Massive thanks to Annette Jena for their robot, so we could spot 60 compounds onto long rat liver homogenates, spraying with DHB, and a massive thanks to Hyundai for doing all the data processing. Brexit didn't stop us sending the samples to Jens, who ran them all on a Brukker timsTOF in both positive, negative mode, and also in MALDI-2 format. And here we are, we're looking at these particular ions and ion adducts. Here's a snapshot of the data for the positive mode, for example. So here's a protonated data on the left and the radical cations on the right. The blue circles are MALDI 2, the red circles MALDI 1. Signal intensity goes increasing down the middle. The scales are not the same because we'd never see any of the red ones on the protonated version. So here's the same sort of data, but not quite so clearly exhibiting it. But what we can see is some other features here that where we actually saw 78% compounds better in MALDI 2 and also 8.3% exclusively seen in MALDI 2 that we didn't see in MALDI 1, so making them visible. Physical properties, yeah, we saw some very complex interactions, of course you would, with 1,200 compounds involving the drug solubility and permeability. We're not sure we fully understand them all yet, but we're working on it, and hopefully we'll be able to publish it all very shortly. So lastly is the summary. So say we've got 78% compounds work better than MALDI 2. The compounds are too diverse to come up with some chemical information. And there's something happening in physical properties. But lastly, acknowledgement, massive thanks to Jens and Klaus, the guys at AJ, the team in GSK and Dave for providing all the compounds. And happy for that.
Thank you very much, Peter. That was a really nice talk. Quite clearly showing off how good MALDI 2 can be for ionisation of some of these difficult compounds. One of the things that I took from your talk was you showed those circular plots showing MALDI versus MALDI 2, and there was an awful lot of data in there. How difficult or not difficult is it to interpret that amount of data that you've generated?
It certainly takes a lot of time. The important thing was to actually have all of the mass data of the structures and the chemical formulae. But yeah, we're still working on a lot of it because, as I say, we've not just got the protonated data, we've got all the adult ions and stuff. So yeah, there's a lot of data we'll be processing. And Jens probably had the biggest job of actually sort of like looking and going to find all the ions associated with the masses in all of the data sets that he generated. So big thanks to Jens for that.
Fantastic. Well, thank you once again, Peter, for your talk. And in the interest of time, we'll move on. But some fascinating work there.
Thank
you. I do not believe we have the next speaker, everyone, in which case that brings us to the end of our short oral presentations. I believe there is now a short break before the next session at 2 o'clock. So come back at that time, everyone, with lots of questions in the chat. Thank you very much.
Hello, welcome back. We're at session 7, Data Processing Bioinformatics. And I'd like to welcome our first speaker, our keynote presentation, Melanie Foll from Northeastern University. If we can just bring up Melanie. Hello. Shall we check your volume? Your sound is good. And your slides, if you'd like to bring them up, we'll check everything's okay. That's wonderful. Thank you, Melanie. Over to you.
So thank you very much. It's a pleasure to be able to present here the latest updates of the MSI software Cardinal. My name is Melanie Foll. I'm a former postdoctoral fellow and now visiting scientist in the lab of Olga Witek at Northeastern University. I will start my talk with giving you a brief recap on mass spec imaging data and analysis, then continue to explain some of the Cardinal methods, and then also show with some use cases, so real case studies, how Cardinal can be used to analyze data starting from raw data until we get the final analysis results. So mass spectrometry imaging is used for diverse research questions, as we have already seen now in this conference a lot. So I will not go into any more details. I just would like to highlight that there are many different research areas that apply mass spec imaging, and that there are also many different research questions. As a quick recap, most multi-imaging techniques work very similarly, and I just depicted a multi-DOF device here. So we have a tissue section mounted onto a glass slide, and the laser of the multi-instrument is rastering over the tissue, acquiring one full mass spectrum at each position. And this is done for the whole tissue, generating many spectra, and they are actually labeled with their X and Y position, meaning that we can trace back their position in the tissue section and can then, for each analyte, plot the intensities in each spectrum, so at each position. Often we compare this to the optical image or an H&E stained image of the same slice to compare histological features of our mass spec imaging data with the optical images. The MSI data is often very large in size. So here's a representation as a data cube. We have the X and Y position on the tissue, and then in the Z dimension, we have many different masses that were measured. And when we look into one slice, we obtain the intensities of one analyte at each position, at each in all the spectra that were measured. And just to give some numbers, but you know this if you are doing mass spec imaging, we have often tens of thousands of spectra. We have, at least in the raw data, tens of thousands of MC values, and the typical file size is now several dozens of gigabyte in size. And in a typical experiment, there's not just a single file, but we have multiple tissues and therefore multiple files per experiment. And this means that we have a lot of data complexity and a large data size that requires specialized algorithms that differ from the classical mass spectrometry-based methods that are available. Another challenge is that we have a lot of metadata, for example, about the sample origin, or also sub-tissue annotations, for example, information where certain tissue areas, for example, tumor or healthy, are located, and they are then also mapped on top onto the spectral information so that they can be used later on for a correct data analysis. I depicted a few more challenges. There are even more, but I would like to focus on these. We have spatial dependence between our spectra, meaning that the neighboring spectra have closer molecular content compared to spectra further away. We use a diverse range of mass spectrometers and measure a diverse range of analytes, and having in mind a diverse set of research questions, and making this data very complex in the spatial, but also in the spectral dimension, and requiring very individual analysis approaches. Also, to advance the field, reproducibility is a key aspect that is often overlooked in this field, and this can be generated by sharing the raw data and also the analysis code, so that other people can actually retrace what was happening during data analysis. So with this, I would like to come to Cardinal. So Cardinal is developed in the lab of Olga Witek, and Kylie Bemis is the main developer of the software. Cardinal is unique in being free and open source, while at the same time providing a broad methodological toolkit with a special focus on statistical methods that help answering a lot of research questions. Cardinal supports all common types of data from all common mass spectrometers and also all common analytes. And Cardinal is implemented in R and Bioconductor because that's a programming language that is open source and allows reproducible research and is familiar to many mass spectrometry, bioinformatics, and mass spectrometry research. So from Bioconductor, Cardinal was downloaded more than 8,000 times since its release in 2015, with about 100 downloads every month, so unique downloads. Recently, Cardinal was also awarded to be an essential open source software for science from the John Zuckerberg Initiative. And below you can find several linked published methods about the software and statistical methods. So here are depicted some common data analysis steps for mass spectrometry imaging analysis. They are depicted here in a linear manner. However, in practice, they may be rather iterative. And the steps are MSI data and metadata import, visualization of any raw data or also statistical results, then preparation, meaning combining and filtering files and annotating spectra, pre-processing of the spectra, and then the statistical analysis itself. And finally, export of the processed MSI data and also the R code. So Cardinal covers all these analysis steps at a very broad depth. And in this representation, the dashed methods mean that these are methods that were either improved or newly implemented in the Cardinal 2 version. So there was a lot of restructuring also on the technical side of things. So for example, the R package method was used in order to enable for basically all of these methods an out-of-memory computation. So also larger files are now supported. I will come to that in more details later on. And now I would like to give you a glimpse into the functionalities for each of the mentioned analysis steps and provide some examples before I come to the case studies where we actually apply them to real data sets. So we start first with the data import. Here Cardinal has now full support for IMCML files. That means they can be either continuous or processed IMCML types, profile or centroided mode, and there's support for larger than memory files. Again, Cardinal is compatible with the typical data types coming from multi-DESI SIMS ionization and TOF, Orbitrap, FTICR mass analyzers, and is a broad software that does not focus on any specific analysis steps or analytes. So currently, most of the MS vendor software support an export of the open standard IMCML file. However, in case that's not possible for you, there's a nice IMCML converter developed by Alan Reyes, which you can use to obtain IMCML files. And Alan Reyes also established a very helpful IMCML validator tool that we highly recommend before starting to load the IMCML files into Cardinal, just to make sure that the actual IMCML file is a valid file. Cardinal offers a large variety of visualization options, which is also due to its implementation in R. It can easily adjust visualizations and generate visualizations that may not be as typical in common MSI analysis. Depicted here are the typical plots we do, so mass spectra of a single spectrum, average mass spectrum zoomed in, and overlaid mass spectra, and ion images in the raw state with a smoothing applied, with a contrast enhancing applied, or the overlay of multiple masses into a single ion image. For data preparation, that means that we can filter the data into any dimension and combine multiple data sets. However, here I would quickly like to highlight the spectra annotations. So first, it's possible to draw freehand areas into either ion image or like here in statistical results image in order to annotate certain characteristics to these selected spectra. On the other hand, it's also possible to load a complete data table into R and then merge this directly to the MSI data. And that's also a feature new to Cardinal 2, that the metadata is stored in a simple data frame, so in a normal table, but at the same time it's directly attached to the MSI data. So once it's attached there, there's no chance that you can somehow lose the annotations or confuse them. So that makes the analysis even more reliable and reproducible. For pre-processing, Cardinal offers very diverse methods. The main method listed here on the left-hand side. However, all of these methods can be customized to very specific niche applications and needs. Here's just a quick image of a baseline removal and a peak detection. I would like to spend a few seconds on this peak binning algorithm, which I think is relatively unique in Cardinal, but very powerful. So at this peak binning or peak integration step, we provide a list of detected masses that we obtain from a normal peak detection and pre-processing pipeline, and then go back into the normalized profile mode data. And for each spectrum, look at each location of the masses that are in this peak list for the peak, and then whatever is there in this tolerance window will be integrated, and from this area under the peak we obtain an intensity value. And the advantages are that with this method we are less reliable on the correctness of the peak detection algorithm, and we have less zero values, because often even if there's not a nice peak, there is some background noise that will then get integrated and will be used as an intensity value. So Cardinal has a strong focus on statistical analysis steps. There are three very common analysis approaches in mass spectrometry imaging. They are the discovery of classes by using unsupervised segmentation, class comparison using linear models to find differentially abundant analytes between classes, and class prediction that is done by supervised classification approaches. And for each of these main analysis steps, Cardinal provides several methods that have different strengths, and I just want to quickly highlight that the spatial shrunken centroids method was also developed by Kylie Bemis, and it has the advantage of taking into account the spatial dependence of the pixel at the one side, but also performing a feature selection so that in the end you don't only obtain the segments or the classifier, but you also know which are the features that actually define these classes or segments. And last, we have the export option. This is very important because here again, Cardinal exports the open standard IMCML files now also in all available modes. And this makes it compatible with other MSI software, for example, with Metaspace for analyte identification, and it facilitates upload to public data repository, and the IMCML files can be reused by everyone regardless which software they have. But to achieve reproducibility, it's also important to publish the actual analysis steps and scripts. So this in R can be either done by publishing R scripts or R Markdown files, which is shown here, where the R code is actually embedded between text and the results images that are produced. So new in Cardinal 2 is that the whole code base was restructured. And the reason for this was that when Cardinal was developed several years ago, it was not yet clear that the data will get so big and complex. So now with the restructuring, this really helps to be more computational efficient for large files, which is especially important because a disadvantage of the R programming language is that it is quite challenging to handle large files. So here, Kylie Bemis developed a meta R package that is now implemented in basically every cardinal analysis method, and this allows to compute larger than memory files, and it's very efficient because in the background there's no file conversion and no file copying going on. And with that, one can typically use a smaller computer than the size of computer memory than the size of the data set. And furthermore, also for the most computational demanding steps, multiple CPU support for all operating systems is available, which can help to speed up the analysis of large files. Before I will present with the case study, I just want to quickly highlight the work of Gordon Liu from Laura Sanchez's lab. They compared the performance of Cardinal with the Skills Lab software from Brukker on two different research questions on bacterial cultures of Pseudomonas. And here we have an example of unsupervised segmentation of these bacterial cultures and their secreted metabolites, and both softwares find these circular segments with the secreted analytes in the agar. The other question was to find differentially abundant masses that were known between two different conditions. Skills Lab found all the three masses that were known to be differentially abundant, while Cardinal could only find two because for pre-processing they had used an automatic window estimation step that unfortunately had a bug, which is now fixed. And so one mass was not existent in Cardinal after pre-processing and couldn't be included into this differential abundant analysis. So to sum up, Cardinal performs similarly to Skills Lab for these two analysis approaches. However, I would like to highlight that Cardinal is free to use, allows reproducible research, and comes with a rich methods portfolio that allow a sheer unlimited customization analysis for a broad range of research questions. And now we use Cardinal on three public data sets to show its capabilities to analyze data from different MSI modalities. So the first case study is from Andreas Römb and quite popular because it was, I think, the first data set that was published in Pride. It's a mouse splatter phospholipid imaging. And the aim here is to do unsupervised segmentation in order to find tissue-specific segments. The second case study is one of the largest data sets available from the Carsten Hopf lab, where exogenous peptides were measured in different pig organs by Malditoff, and the aim here is to do single ion segmentation and some basic statistics to measure the protease activity at different time points. The third case study is human bladder tumor cohort of 39 patients where tryptic peptides were imaged by Malditoff, and 2 different tissue types will be differentiated by supervised classification. So all these case studies have very different spectral properties and different experimental designs and different research questions, and therefore require different analysis approaches. So case study 1, I called it the high mass resolution segmentation because we would like to especially highlight that Cardinal is now able to fully support high mass resolution data sets. After performing a typical pre-processing analysis, we visualize some of these features of the data set, and in the data set there were 2 peaks that were only 0.05 MC apart, and this image actually shows that both peaks were picked will be used during peak picking and can thus be separated. And after pre-processing, the peaks show that they have a different spatial distribution in the mouse bladder. So Cardinal was successful in pre-processing the data set, and we also repeated this image from the publication where 3 different masses were overlaid. 1 mass corresponding to the urothelium, the red one to the lamina propria, and the blue one to the muscle layer of the mouse bladder tissue. But we wanted to know if there are more masses that are correlated with these tissue types, and therefore we performed unsupervised segmentation. We used the spatial Schwanken-Sandwich algorithm developed by Kylie Bemis, and we found several segments. Some of them were here the background in green and in yellow, but we also found tissue-specific segments, like in red the urothelium, in blue the muscle, and here in yellow the adventitia. And due to the underlying feature regularization in the spatial Schwanken-Sandwich method, we were actually able to obtain a subset of masses plotted here for each spectrum that are the most important masses for this spectrum. So with this approach, we found with an easy step several masses that describe different tissue types. However, we were a bit surprised that we didn't find a cluster corresponding to this lamina propria area here. So we performed an additional colocalization analysis. And in this analysis, we found that when we used this lamina propria specific MC feature from the original publication, there are actually not very good correlating analytes in the data set. So the next one had a correlation of only 0.5, and also from the ion images, we see that these are not very localized to the lamina propria, and this explains why we couldn't find it with our multivariate segmentation approach. But in Cardinal, there's also a univariate segmentation of single ions. So with this approach, this would be an opportunity also to find such clusters that are defined by only very few mass features. We have not applied it here, but we have applied the single ion segmentation in the next case study. So case study 2, we have a large-scale data set, and one of the aims is to perform single ion segmentation to obtain some annotation information that was unfortunately not provided with the published data set. So this data set by the lab of Carsten Hopf used different pig organ tissues, and they sprayed a peptide on it called substance P. And the idea was that in the tissues, the proteases are still active and then cleave the substance P peptide into smaller peptides that can then be visualized by mass spectrometry imaging. They also added some protease inhibitor spots, which should prevent the digestion, and they even included a control tissue for each tissue type and for each time point, meaning that this tissue was never digested, was never incubated, and without incubation, the proteases cannot get active and not digest substance P. And because this setup was done on 5 different organs and with 5 different incubation times, that this is the full amount of tissues. And this data set is 55 gigabyte large and is one of the largest publicly available data sets with around 50,000 mass features and more than 280,000 spectra. And we use this data set to show cardinal capabilities that are now improved in version 2 for large-scale data analysis. So we started to calculate a mean spectrum with using 1 CPU and found that this was about 1,200 seconds in time and took 8.5 gigabyte memory. And from the memory, we can see that the maximum memory consumption is just a small fraction of the size of the full data set. We repeated the same step with 2 CPU cores and found that then also the analysis time was decreased a lot, meaning that we can really speed up the analysis of such large files by using multiple CPUs. For pre-processing, this was done again only with 1 CPU, took nearly 1,000 seconds and around 10 gigabyte of maximum memory usage, which again is a fifth of the full data set size. And previously, we would have needed a much larger computer to run the analysis. And now in Cardinal 2, this is way easier to do on a normal computer or even laptop. The resulting dataset after pre-processing was quite small with 210 megabyte and 187 mass features. We then continued and annotated the spectra and filtered the spectra. So we only kept spectra of the spleen tissue and we only kept spectra that were actually digested. And this is an ion image of the substance P peptide. That's a peptide that was sprayed equally onto all tissues and then incubated. And we can see that only in the areas where the peptide inhibitor mix was spotted, substance P intensity is still a bit higher. And in the surrounding where the proteases were active and digested the substance P into smaller fragments. And the problem was that with this data set, the information was missing about which spectra actually were the ones that were spotted with this protease inhibitor mix. And therefore, we made use of this very unique single ion segmentation approach in Cardinal. And we used it here in order to segment the substance P distribution into 2 clusters. And we defined then the resulting cluster as a spectra that had the protease inhibitor in orange and spectra that had no inhibition and where the digestion could occur in blue. And then we wanted to repeat the analysis that from the original publication. And for that, we just calculated the mean intensity and the standard deviations for each of these clusters at each time point and normalized it to the non-digested non-digested tissue intensities of substance P, and obtained a similar curve as in the original publication, where we can see that in the areas without inhibition, the substance P concentration is decreasing with longer incubation times, while when we have inhibitors around, the substance P is not so quickly digested, and it takes longer to actually digest the peptide there. And last, in case study 3, we have a multiple replicate dataset on which we want to perform classification. The dataset had 39 patients and had very detailed annotation of tumor and stromal areas that are present in this tumor tissue course. And here we imported the MSI data and the metadata and added again this metadata directly to the MSI data, which allows us to later on have easy access to all this metadata information when we are doing the actual statistical analysis. After adding the metadata annotation, we combined the files and performed pre-processing for multi-TOF data. This is just an example how the R code looks like for these steps. And I would like to highlight the 2 new pre-processing methods, the mass or spectral alignment and the mass recalibration. So in this data set, we had quite some shifts in the mass direction between the spectra, also of the same data set. There were many shifts visible in the mass direction, and here are just 6 random spectra plotted, and we can see that after the mass alignment, the spectra are way better aligned than before. And also the data set contained several internal calibrant, and here the vertical line is the mass of angiotensin, which was used as one of the internal calibrants. And before recalibration, we see that the peaks are actually a bit off and not correct in their mass accuracy, but after recalibration, they fit very nicely on the position where they are supposed to be. We then performed classification again with the spatial Schwanken-Sandwich algorithm, and we obtain high accuracy, sensitivity, and specificity for the training and the test data. Here's an example output how the classification looks like for one of the tissue microarrays. And this is in comparison the annotation from the pathologist. So we can see that there's high overlap, even though some, here for example, some spectra are misclassified. And the advantage, as I said before, of the spatial Schwanken-Sandwich method is that it can shrink down or select the most important features. And the feature contribution to the classification is shown here. The higher the bar here, the more important was the feature contribution for this classification. And when we picked the masses that were most important for tumor classification here and stromal classification, this mass, we can actually see that the intensities are pretty perfectly aligned with the annotation shown here in this H&E image by a pathologist. And this shows that Cardinal can handle quite well multiple replicate data sets that have a lot of metadata, a lot of patients, and even subtissue information that is used for classification. However, the disadvantage here is that for these typical classification methods, we actually need really detailed annotation by a pathologist in order to have them perform optimal. And to overcome this issue, we developed a new classification method that works with only rough annotations. And this is of course good because pathologists are short on time and it takes quite a lot of time to do such sub-tissue classifications as seen here. But this data set was used as a exemplary data set to show that with sub-tissue annotations, the classification performs well, but also with rougher annotations. And this is because the algorithm takes into account also the dependency between the different mass features, and in the end, performs a bit of a semi-supervised classification and organizes the mismatching pixels itself and finds out to which class this belongs. And this currently works for 2 classes, but in the future we are working on it to make it available also for multiple classes. However, I have to say this is currently only available as Python libraries because the deep learning behind it is based on Python. But we will definitely also include it into Cardinal so that everyone can use the normal R environment to use this method for their analysis. And with this, I hope that you got interested in using Cardinal for your research, and I would just quickly like to highlight some resources. So the availability of Cardinal is typically via Bioconductor and then using RStudio. However, for more bioinformatics interested people, there's also Bioconda recipes and biocontainers available for download, and then all dependencies and versions are there and controlled. And most of the Cardinal functionalities are also embedded in the Galaxy bioinformatics data analysis platform, and within Galaxy the tools have a graphical user interface and they can be run on large public clouds for free, and they can be used in a modular way to build standardized workflows and run them on the samples. And to get started with Cardinal, there's a lot of information available already, and I would like to point you towards all this information. So on the main website, there are hands-on trainings via vignettes, starting from raw data, going to the statistical results, a lot of documentation about the methods. Every May, the VTech Lab is hosting the May Institute, and this includes also basic and intermediate R lectures, as well as Cardinal workshops, and previous meetings were recorded. So you can also look into the YouTube channel and watch these videos. Then we have a help forum if you have problems or questions with Cardinal, and there's also specific training material for Cardinal in Galaxy. And with this, I would like to thank the VTech Lab and especially Kylie Bemis, Dan Guo, Sai Lakim Sethi for their great work and for being great colleagues. And I would also like to acknowledge my colleagues at my home institution at the University Medical Center in Freiburg. And I would like to thank you for your attention, and I'm happy to take questions.
Thank you, Melanie, for that presentation. Very detailed and comprehensive, powerful piece of software that you've got there. Just thinking in terms of next steps and the future developments, are there established research groups that you work with in order to gather, run-throughs and case studies, or have you've got all you need in order to move things forward?
We are always interested in collaborations and obtaining state-of-the-art data sets to test existing algorithms, and especially to develop new algorithms. So if someone is interested, just contact us. We are more than happy to collaborate.
That's wonderful. Thank you. I'm just going to look at the chat just a minute. Okay, so how does Cardinal deal with intact protein peaks, mainly in the peak picking and the binning stage?
Not So you can just set the parameters as you want. So I've not yet analyzed full protein data, but you can set the windows and the signal to noise ratio accordingly. And yeah, so you have full control with all the parameters that you need. And if there's something you need that is not there, all the functions can be customized for very special needs.
Okay, thank you. I don't think we have any further questions, but thank you. And if you're happy for people to contact you for to provide data sets, that's wonderful going forward. Thank you for your presentation.
Sure. Thank you.
Thank you. So we're running perhaps a time, but if we're ready for Landry Blanc, please, from the University of Bordeaux. He's ready ahead of time. That's wonderful. So the sound is good. Shall we?
Yeah, can you hear me?
Yes, perhaps a touch louder.
A bit louder. Okay, let me try to go on
the
mic.
No, that will be fine. We'll be able to adjust that. Thank you. As it is. If you share your screen, and we'll see if we've got what we need.
Can you see?
That's wonderful. Thank you, Landry. Over to you.
Okay, thanks. So good morning, everyone. I would like to thank the organizer to let me present this current work we perform at CBMM in Bordeaux. I will split this presentation in two parts. First, I will talk about isotopic labeling to study metabolism of brain metastasis by mass spectrometry imaging, and in a second time, about a processing method to decipher this data. Okay. This work started for us in 2017 when we get involved in a collaboration with researchers from Harvard Medical School studying fatty acid synthesis dependency of metastases located in brain environment and resulting by a publication this year in Nature Cancer journal. Our collaborators study breast cancer metastases and especially the one located in the brain as they present a poor survival outcome and limited therapeutic option. In mouse model, they observed a large difference in relative abundance of metabolites between tumor growing in mammary fat pad or in brain. Moreover, by studying cancer cell gene expression, they identify a striking enrichment in gene associated with lipid metabolism for tumor growing in the brain. They confirmed this data by Western blot and found higher expression in brain metastasis of enzymes involved in fatty acid synthesis, like fatty acid synthase, stearoyl-CoA desaturase, or acetyl-CoA carboxylase. Altogether, this data suggests an adaptation of tumor cells into brain environment, leading to an interesting seed-specific dependency on fatty acid synthesis and a potential new target for pharmacotherapeutic strategy. So I will not go into all the detail of the aspect of this paper. I recommend you to have a look at it. I will focus on our collaboration for mass spectrometry imaging. So in order to study metastase metabolism, we decide to trace the fate of glucose by injecting glucose C13 into living mouse bearing brain tumor. The isotopic tracer is picked up by cells as regular glucose, and carbon C13 are incorporated into metabolite. We then collect in tissue from mice to analyze them by mass spectrometry imaging. So we perform a very classical MSI workflow acquisition. After tissue collection and cryosectioning, samples were sprayed with DEN matrix, and then MSI acquisition were performed on a MALDI-LTQ trap in negative mode at 60,000 mass resolution and 50 micron spatial one. On adjacent section, we also perform an immunohistological staining to identify tumor area. With MSI, we are able to show a C13 enrichment in some fatty acid in the brain tumor, like for palmitate or stearate, whereas in other location, it's not the case, and of course, in the neither in the non-dose mouse. This result show that in brain, glucose is uptaken by cancer cells and metabolized in fatty acid, and consequently reinforce the idea that brain tumor microenvironment contribute to increase lipogenesis in tumor cells. During this first round of analysis, we target specifically fatty acid labeling based on the initial data collected by our collaborator. We look for peak which could correspond to fatty acid isotope, like for palmitate with the M+2, M+4, or M+6. We also try to look for unexpected metabolite, which could have been labeled by C13. Unfortunately, due to the complexity of MSI spectrum, as there is no separation like in LCMS, we struggle to identify labeled metabolite with an untargeted way. And now comes my second part. After the publication of the Nature Cancer paper this year, we were very happy, but a bit upset as we were sure that this isotopic labeling dataset contained more valuable information. We decided to implement a new processing approach, offering another way to visualize MS data and helping to decipher isotopic labeling. This approach is using a variation of the 60-year-old Kendrick's mass defect analysis and is currently under review. So first of all, what is a Kendrick analysis? So it's a processing method which offers an alternative way to visualize MS data, like in this graph, where each dot corresponds to a peak in this spectrum. To do so, we use a basic mathematical formula. So for each peak of the MS spectrum, so for each M on Z, we calculate its Kendrick mass, and then the decimal part of this Kendrick's mass called the Kendrick's mass defect, or KMD. And then we generate the graph by plotting KMD in function of M on Z. In 1960s, by choosing the R variable equal to the mass of C2, so the minimal block of a lipid chain, Edward Kendrick obtained a plot where compounds with same substructure but different lipid chain are horizontally, like here for this fatty acid. Moreover, different level of insaturation are following another, like here, here, or here. This way to visualize MS data turned to be very useful to study complex mixture of petroleum derivatives, polymer, or simply lipids. In our case, to study isotopic labeling, we decided to use a variation by choosing another value of R. As we are looking for incorporation of carbon C13 instead of C12 into metabolites, we use a variation proposed by Nakamura and al. In 2019 by choosing R equal to the difference of mass between C13 and C12. By doing so, we are now able to reorientate the Kendrick's plot in a way that C13 isotope of the same compound are aligned horizontally, and when also CH2 increments or lipid chain and insaturation are still aligned but follow another. We can now compare the MS data of mouse brain tumor dosed or not with glucose C13 by zooming into the dot plot, like here for fatty acid area, and identify easily metabolite presenting an enrichment into their lipid profile, like the fatty acid we already identified into our old analysis. We can have a look into more detail into them, like enriched stearate and the non-enriched arachidonate. So we can see here the C13 enrichment of stearate in brain metastase up to 8 carbon compared to the non-dose mouse. And here it's the relative abundance. It's the red cross represents the theoretical natural abundance of stearate as isotope based on the occurrence of carbon C13 in nature. We can see in green the condition without glucose C13 following the theoretical trend, whereas in blue the dose mouse show an enrichment of a C13 enrichment for stearate. On the contrary, for arachidonate, we don't see a C13 enrichment as dosed and non-dosed follow the same trend. To go further, the Kendrick's analysis allows us to visualize easily complex mass range of MS dataset, like the phospholipid mass range, by looking for horizontal cluster present in the dot plot of mouse dosed with glucose C13 and absent in the non-dosed one. We can identify enriched metabolite, like some of these phospholipids, probably due to the incorporation of a labeled fatty acid chain into this phospholipid. Again, we could have a look on some of them, like the phosphoinositol with 34 carbon. We can clearly see the labeling of the phosphoinositol 3041 in the MS image or in the relative abundance, whereas it's not the case for the PI 42. This KMD analysis allows us to identify in negative mode 12 compounds labeled by glucose C13 in comparison to the 5 we are able to detect initially. Moreover, this approach allows us to identify metabolites where isotopic tracer seems not being incorporated, like linoleate or 20 carbon fatty acid, which in that case make perfectly sense, as these fatty acids are known in mammal to not be generated by lipogenesis from glucose, but have to be uptake through diet. As a take-home message, this Kendrick's analysis is not a way to improve the quality or the resolution of your MS data. It offers another perspective to visualize this data, helping, for example, to decipher isotopic labeling in metabolite, like in lipids. It's applicable to different tracers like carbon C13, deuterium, or nitrogen 15, just by adjusting the R variable in the formula. Moreover, it does not require advanced computational capabilities, as a basic Excel spreadsheet is enough to perform this analysis. Finally, I would like to thank the collaborator of this project from Harvard Medical School, CDI New Jersey, and University of Texas, as well as the colleague from the team in Bordeaux. And I also encourage you to have a look on other work performed in our team in poster 22. And thank you for your attention.
Thank you, Landry. Thank you very much. That's a really nice picture, that isotopic labelling and showing those clusters. It's really beautiful to see that. I'm going to wait just for a minute to see if any questions come through, because we're a little bit ahead of time. So I'm just going to check over on the chat. Okay. Do you, what are the next steps, Landry, employing this same process or this same method? Have you got another study that you're looking to apply this to?
So we would like to perform more study in strong collaboration with the biologists, because this, of course, this kind of isotopic labeling strategy is very heavy to implement, because it implies a lot of people. We could be also very interesting to be able to reprocess this data set, because unfortunately I move into overseas now, but especially in positive mode to see storage lipid, like for like in triglyceride, and also may probably use another instrument, because the LTQ-RB trap is limited to up to 60,000 resolution. It could be more interesting to resolve the isotopic labeling to use a higher resolution instrument.
Thank you. Yes, that's very informative. Thank you. We do have our next speaker now ready. We were going to move to a very short break, but I believe our next speaker is ready. So thank you, Landry, for your
Thanks
story, and congratulations with the publication.
I hope, cross finger. Thanks.
So if we can move now to Olga. So Olga Vendenskaya from Spectra Swiss. My apologies, Olga, with the pronunciation there. Over to you, please, for your presentation. Thank you. We haven't got any audio just now. So if you check down on the towards the bottom of your screen, you should be able to check the audio, turn that on.
How about now? Do you hear me now?
Yes.
Yes. Oh, wonderful.
Yes, we do.
Okay, let me then just try to share the screen. How do I do it? Like last previous speaker was also a bit stressed, and I'm
So with the mouse down at the bottom of the screen, can you see the option to share?
Mm-hmm. This one. No.
But we are running ahead of time, so this has put the pressure on you.
Exactly.
So don't worry too much. We're not going anywhere.
Do you see my screen now?
Not yet.
Share the screen. Should be able to do it now.
Yes, we didn't have a chance to check this with you because we're ahead of time, aren't we? So.
Aha, now. And then you should be able to see my screen now, right?
Yes. So if you go onto
wonderful.
presentation mode, that
yes,
should
exactly.
be it. Yep.
My apologies.
you.
That's good. We were ahead of time.
No problem. Over to you. Thank you, Olga.
Thank you so much, and thank you so much for a wonderful introduction. My name is Oля Videnskaya, and I have recently moved to Lausanne and started to work with SpectraSwiss. And today I'm very, very excited to be given the opportunity to present our new software called Mosaic. Mosaic is a memory-efficient multi-vendor software for big data analysis in mass spectrometry imaging. I would like to start my talk with giving you a historical perspective. So sometimes in 2017, when I was just about to defend my PhD thesis, these wonderful guys you see here on the picture on the left, they installed a booster hardware to improve MSI on FT-ICR MS in the lab of Ron Heron. And that's how the need to process large DESI MS datasets emerged. And this is exactly how the customer needs-driven pre-Mosaic software version was born. So this data was later on published in 2019 in Scientific Reports. Further, in 2019, there was a need for a software application to apply to NanoDESI Orbitrap MSI to match the needs of lab of Yulia Laskin for DESI MSI. And this data was also later on published in 2020 in IGMS. Finally, the software application was extended to MALDI Orbitraps to support the processing of large MALDI MS datasets acquired in LTQ Orbitraps in the lab of Franklin Leach, and this data was presented by us in 2020 in ISMS Reboot. So all these achievements, they were followed by the further developments and the launch of Mosaic software in 2021. Since historically, Mosaic was developed for the FTMS data, it offers the support of a full cycle of FTMS images. So we schematically here divided the full cycle on two main parts, data processing and data analysis. And I think it's important to show it here because as I realized listening to all these talks today, we've got different definitions of data processing and data analysis, and this is how we see it. So the data processing starts with data acquisition, is followed by time domain transit acquisition and absorption mode or AFT mass spectra acquisition, and then followed by peak peaking. And then data analysis is the individual images plotting, image overlay plotting, statistics, and knowledge acquisition and knowledge extraction. I would like to briefly explain you the FTMS data types. Since the type which contains the least amount of information is the magnitude mode MFT centroided data. Here you can see it on the left bottom corner. And further, the information content grows from magnitude FT to enhanced, EFT, and to AFT modes. This way, and it also grows from centroided to reduced profile to full profile data. So the data type which contains the most information is AFT full profile and is called unreduced data. Mosaic software supports all the FTMS data types, but not only them. So now we've got the support for, as I already mentioned, not only FTMS data, but basically for the MSI instruments and the ion source of all types. When it comes to the ion source, Mosaic supports both DESI and MALDI from multiple vendors. And regarding the mass analyzers, there is a support for orbit traps and MRMS and FTICRs, historically, of course, with the possibility to add FTMS boosters. And it's now extended to MRT, MSI, TOFs, and triple quads MSI with their particular file types. There is a possibility to perform the direct data upload from Agilent, Brukker, Thermo, and Waters, and the indirect data read is happening through the ProteoWizard. And Mosaic performs the comprehensive data processing, some data analysis, and has the possibility to process and store the files and projects. Further annotation when needed and in-depth analysis, case when again it's needed, can be performed with some built-in capabilities and some third-party software tools, such as Metaspace, for example. So our main drivers in Mosaic developments were the following. First, to optimize data processing to extract the most information from MSI datasets of any size, and to maximize quality and confidence in the MSI data. Then the second thing, we wanted to support the memory-efficient data processing for on-the-fly data processing and instant visualization functionality. And third, we wanted to provide the users with the basic data analysis, of course, capabilities and output data in IMZML file format for further potential use with the already existing wonderful tools which we've heard about today, and more of them as well. The main features we aimed for this software to have were the following. So we wanted to develop the processing for time domain transients and full profile mass spectra and FTMS. We aimed for the software to be a vendor neutral and multi-platform, to be able to process the data from FTMS, MRT, TOFs, and triple quads, supporting both MALDI and DESI ion sources, including the ones from niche manufacturers such as Mastec, Spectroglyph, and Transmit. We aimed it to be developed in Python programming language, so it could support HDF file-based high-performance architecture for memory-efficient data processing, as it's done in the modern softwares which are developed these days as well. And we also aimed to have a comprehensive yet ergonomic graphical user interface, and we strongly believe this is one of the features which is really essential. Well, now, when we're clear with the objectives, I will show you what we have achieved so far. Here is an overview of GUI when one works with the individual image. For example, here you see the image, which has the interactive pixels. You can see that the ROI selection is also here. You can see on the left processing parameters set up, and on the left bottom you see the selection of MS or M over Z targets. On the right you see the mass spectrum. You can also plot the mass accuracy, and you can also plot the intensity distribution. And here in this case you see the mass overcharge tolerance window, then information about the average scans of ROIs here, and the targeted mass itself. So this is the individual image, and we also have an option to work with the multiple experiments, including those acquired by multiple experiments from various vendors. So the same list of experiments and the list of selection of mass to charge targets here, but here you see at the center of the figure, you see the multiple experiment view. And so the upper one was acquired by FTICR, the middle one by TOF, and the lower one by Orbitrap ELID. And so you can see here the interactive mass spectra, the adjustable and customizable color maps, and the possibility to perform the comparative analysis of spectra. I also have prepared an overview of a couple of selected features. First of all, as we have talked and we've seen other people talking about binning, I also want to point out that in our case, we chose to use no binning approach. And we do process without mass scale binning. And this allows us to work without data loss, to keep the peak shape, and to avoid the time and resources consuming recalculations in case if the binning parameters were not chosen optimally initially. The picking in Mosaic is done with a standard 3-point interpolation procedure. Another feature I wanted to point out is the possibility to colocalize and overlay of multimode images. Here you see the possibility also to adjust the image transparency, to add color functions, to check the customizable and adjustable color maps. And here you see on the left the overlay of selected ions with the distribution with the optical image. This, of course, is allowed for the memory-efficient processing of multiple experiments. The next one is the work with ROI. And first, I'd like to point out that there's a possibility to perform not only manual selection, but also an automatic selection of region of interest. And there's a possibility to perform a subsequent geometry handling. The follow-up analysis of ROIs can be performed comparing mass spectra from multiple ROIs for the same or different data sets. A feature which we are particularly very happy about is the possibility to perform an averaging of unreduced data. So if you remember a couple of slides ago, I showed you the possibility, the AFTMS data types. And so we can work even with the unreduced data. Here is which we're also particularly very happy with. This data was obtained by Q Executive HF. And so let's have a look at the ROIs and perform the spectral averaging. So on the left, here you see the result of the reduced data processing. And on the right, you see the result of unreduced data processing. On the left panel, the A+1 isotopolog is missing, while on the right panel, it can be seen here, especially properly seen on the 25 pixel averaging. So this allows for a more reliable identification of the features. Another cool feature Mosaic has is the possibility to perform the mass recalibration with the several different algorithms. So here's an example from the Lumos instrument, multi-source from our collaborators and partners from MASTEC, which was obtained at the resolution of 120K and AGC is disabled. You see here the mass accuracy distribution prior to the recalibration. The options which are available for recalibrations are the linear option, can be performed with a single log mass or with the multiple log masses, and alternatively, the iterative nonlinear recalibration can be performed using multiple log masses. Here you can see the visualization of the mass accuracy distribution before and after recalibration. So before you see the standard deviation is 1 and 1.6 ppm, while at the after recalibration, the standard deviation is only 0.2 ppm. And finally, I'd like to show you a couple of extra examples of non-FTMS application of Mosaic. This is the data from the very brand new MRT instrument from Waters. Impressively, this data was acquired on DAISY ion source at 2 resolution. Here are the images of selected doublets on the left, and you see the mass accuracy. So here is in the middle, there's a mass spectral peaks, and at the right, there is mass accuracy distribution. The isobaric compounds differ in each case only by 9 millidalton each. And we were very, very excited to see the excellent mass accuracy on a very wide mass range at a very high resolution. And recently, the use of triple quad MSI for MSI has received a particular attention due to its sensitivity and world availability. Following our collaboration with MASTEC, Mosaic now supports triple quadrupole MSI data processing. A particular feature of this software is the possibility to work with the user-specified transitions enabled by selected reaction monitoring filter. And I would like to just conclude my talk and underline that Mosaic is a vendor-neutral MSI software that heavily focuses on data processing. that we also support the unreduced data, and this is beneficial for applications requiring enhanced sensitivity, for example, for metabolomics or peptidomics. Mosaic provides basic data analysis capabilities and outputs data in IMZML format for integration with the third-party data analysis software tools, and it is a software package which is available for evaluation and benchmarking, and we're highly welcoming any input. And with all the software development, it's work in progress currently and will always be the work in progress. So we're very happy to hear your feedback. And with this, I'd like to thank you all for the wonderful opportunity for us to present the data, and to thank our collaborators, partners, and customers. Thank you so much.
Thank you, Olga. Thank you very much for your presentation. I'm just going to give a reminder to our delegates to put some questions in the chat, because we do have a delay coming back to the studio. So if I can just say to you, Olga, some of what I guess were screenshots of the user interface. They looked really, really nice, actually, the layout of the platform. And what I was going to comment on was the overlay of sort of the ion images with the optical images, which is increasingly becoming important. But I wondered if you'd got any developments to sort of generate 3D images at all. And I guess this question I should have put to the previous speakers as well. Is there any developments in that area? Because I think that's what we're starting to require.
Thank you so much for this question. It's really important, and we're working on it. If we're talking about the code and the code implementation, that's pretty much there already, but we're just working on it to be available for users who could use the graphical user interface as well.
Okay,
something which takes time.
Yes, thank you. So I have, again, with the delay, I've got no questions come through in the chat. Is there any additional comments you'd like to make with regard to presentation or future developments that you're looking at?
Well, we're always happy to hear the feedback. So basically, we're customer oriented, and if you see that there's something missing in our software and you see that we can benefit and other users can benefit from it, we'll be very, very happy to implement it.
Thank you. That's wonderful. I think as we're 10 minutes ahead of time for our next speaker, and while we I just wonder if perhaps either we'll have a bit of a break, or if we go to Weiwei. So I'm looking at the schedule, and it looks we do have a break after. So 10 minutes ahead, but we will, I think, bring up the next speaker. Thank you, Olga. And if we can bring up Weiwei Zhu.
Hello.
Hello. Hello. How are you?
Very
yes,
good. How are you?
We've rushed you ahead into this ahead of time, which was very different to the session I chaired yesterday, actually. So you're at the National Physical Laboratory, Weiwei, and I look forward to hearing your presentation. If you want to bring your screen up, we'll let you know if there's any issues.
Yes. Can you see my slides? No?
Yes, we can see them. Just if you go into presentation mode, we'll be all set. Thank you. Thank you very much.
This. Yeah, great. Hello, everyone. My name is Weiwei. I am from the National Physical Laboratory in the UK. I would like to thank the Ourcon for giving me this opportunity. And today I'm going to present our recent work on the synthetic and benchmark data for mass spectrometry imaging. We know that the spatial segmentation is a common image data analysis practice, which by using clustering methods aims to partition the imaging pixels into different groups based on the spectral similarities between them. For example, the k-means clustering is one of the most popular algorithms used in MSI data, which can distinguish between the anatomies within, for example, mouse brain tissue, and distinguish the tumor margin and even the intratumor heterogeneity. The k-means clustering relies on the definition of the distance metric. For example, two approaches are shown here, the Euclidean and cosine distance, which is used to find the minimized intra-cluster sum of the distance of each point in the cluster to its cluster center, as illustrated here in this typical iterative process. It is noted that any normalization strategies applied to the data will have a significant effect on the clustering result, which often helps to reduce but not remove the non-biological variances that inevitably introduced at some point during the experiment, such as sample preparation or laser instability. So in order to evaluate the success of a clustering method, we come into a reality that a known ground truth is the gold standard starting point, which is unfortunately not available due to the biological nature of most MSI samples. Previously, we made use of the fact that the k-means clustering assumes that the data within each cluster are normally distributed, and we perform multivariate normality test and use this multivariate normal model derived from the reference data, for example, the one shown here, as a basis for generating synthetic data through resampling from a known multivariate normally distributed data with the same mean and covariance of this experimental reference data. This is based on the assumption that the data from the population are derived from a known probability distribution function, if such function can actually describe the data. We found out that when comparing the synthetic and reference spectra, they show high degree of spectral similarities and features such as the isotope ratios and fragments. We then use the PCA technique to a combined real and synthetic data to determine if the statistical modeling process that we introduced cause any additional observable variance or not. The result shown here suggested that there's no principal component scores that were found to separate the synthetic from the real data. As you can see, the clustering are pretty much similar between them, which means that the difference between the two data within this combined data is smaller than that between different anatomies and noises. We have also used this approach to generate synthetic data to help evaluate new clustering methods such as graph-based or two-phase algorithms. It is not only of interest to achieve high reliability or accuracy when performing this clustering on the data, but also the fact that the size of MSI data has been continually increasing, both in terms of the number of pixels and the number of spectral channels in each pixel. When trying to evaluate the validity of these algorithms, we need to generate image datasets in a controlled manner in terms of its size. It's very unpractical and time-consuming to acquire experimental datasets with all different sizes and remain the size as the only control variable. For example, an image data that consists of 187,452 pixels required about 36 hours experimental time, but only takes 5 minutes to generate synthetically on the PC with 4 cores and 64 gigabyte RAM. In a not so recent paper, we generated a set of synthetic datasets with varying number of pixels from 3,000 to 300,000. And when comparing with the conventional k-means clustering methods, the new two-phase k-means methods takes only about 1/3 of the time, computational time, at all pixel sizes, as shown here. And the accuracy is also not deviated from that of the conventional k-means methods at all pixel sizes. So as you can see, without the help of this a series of synthetically generated datasets, it would take enormous amount of time and effort experimentally and not even be able to come to this very basic conclusion regarding the accuracy of the clustering methods. In a slightly broader perspective, synthetic data can be further generalized as a simulation-based approach to help achieve many other data-related ambitions, such as data fusion of MSI data in different modalities. Most of the time, when we are just looking at our newly acquired batch of image datasets, we might often find out that there was some sort of clear difference in signal intensity across the tissue, which is not the inherent property of the tissue, as shown here from one of the publications from our group a few years ago. There might be variation of the MZ values of ions across many pixels, which is again not the inherent property of the original tissue. Again, can be seen from several other publications. To a much larger extent, there is often a batch effect that basically introduces systematic source of variation at different levels to your raw data. A typical example is the day effect, as can be seen here from one of our unpublished data, where the instrumental source of variation causes the drift of the signal intensity over time. Of course, there are maybe many other sorts of artificial biases that may be introduced at some stage of performing an experiment, which severely hinders the objective analysis and processing of the data. So with all these challenges and demands, we are looking for a way to be able to generate synthetic data and benchmark data in a controlled manner that can allow us to perform, for example, many of these tasks listed here on the right-hand side. Hence, we proposed the corresponding functionalities on the left-hand side that can actualize each of the tasks. For example, by creating regions on a real image or from the ground up with defined molecular identity and distributions of sample molecules, matrix, or adducts, we can establish a known ground truth image dataset, however simple. Then the results of any segmentation and classification algorithm done to this dataset can be directly related back to the known clustering results that we created in the first place. Another example is that once we have created our clean base reference data, we can purposely introduce signal intensity gradient in a controlled manner. For example, here, a linearly reduced intensity by 75% from left to right. And then we can evaluate the pre-processing techniques such as normalization methods. Here is an example of the evaluation of the normalization techniques. The top row is the base data without any introduced artificial biases, and the bottom row is the same data but with the intensity gradient from left to right by 75% reduction. When apply L2 and the median normalization methods to the raw intensity gradient dataset, it does not return an image dataset that is comparable to the raw base data, but they are similar to those that normalized with the same methods from the raw base data, suggesting that for data with intensity gradient, normalization methods offers a starting point comparable to that of the base data. And then if you look at the clustering methods, we found out that if we apply k-means clustering to the gradient data directly, there is a significant asymmetrical features of the clusters compared to the result, the normalized result from the base data, presumably from the fact that the one-directional intensity gradient introduced in the first place. However, if we use L2 normalization methods to pre-process the gradient raw data first, and then apply k-means clustering, it can reproduce most of the clusters that were found in the results of the base data. As you can see here, apart from some of the slightly different color scheme, the clustering is essentially very similar. But if we use median normalization methods, not only the tissue is misclustered, the background matrix is also clustered into many artificial clusters. We also then apply the same approach to look at TSNE clustering method and have seen more or less the same result, where the L2 normalization methods seems to be able to rectify the miscluster asymmetrical features, while the median normalization methods fails. Please note that we're not trying to make any definite or solid suggestions on normalization methods at this point, but only to show that how the synthetic data can help evaluate them. Another purposely introduced artificial biases is spectral shift. We first create a clean original base data, and then added either a jittered linear shift or a 1D Brownian shift to all pixels, and then obtain a spectral shifted image data. If you look at, for example, the data with 1D Brownian shift here, that we introduced a maximum of 30 ppm random shift to each peak in all pixels, the amount of shifted mass can be seen from this offset figure in the bottom row. And then we can apply different recalibration methods. For example, give a plus minus 30 ppm window to each of the integrated peak and trying to find if they can match the original peak in the base data. And then use the offset figure to verify that. So with the help of a paired original and shifted mass shifted image dataset, it becomes possible to verify different recalibration methods in a systematic way. With all this in mind, we formulated these functionalities into MATLAB functions and classes in the object-oriented programming approach, and made use of spectral analysis, which is a very popular MSI analysis and processing tool that were developed by Reyes et al. Many years ago, and then program everything into a standalone toolbox. Users can normally start from one of the three starting points, namely to create from a real image dataset in a hybrid fashion, or create artificial regions, or load in existing artificial regions. And then the standard workflows is pretty much similar between them, which basically involves all the functions that were shown in previous slides, such as add molecular peaks, add variances, add combinations and permutations. The final generated data can be exported into the standard IML format, which is then readily loaded into any of this open source or proprietary software. It is worth emphasizing that any of the base benchmark data that I've shown here are at a very basic and simple level, essentially describable by deterministic physical functions, and it's not intended here to mimic any amount of the complexity of a real biological sample or the total combined source of variation from instruments or sample preparation. The current approach is to offer a way to make objective comparisons of on the same baseline, on the same isolated source of variation, which is often not easily manageable through experimental effort. But on the other hand, we anticipate using this synthetic data generation approach as a way to enable us to explore and build up the introduction of different sources of variation in a systematic way. For example, we can gradually introduce non-biological variances as per the metadata of the instruments to specifically reflect them as if acquired them in a real experiment. We can also introduce semi-theoretical or even first principle methods to better describe the heterogeneity of the molecular samples. The idea is really to open like a playground for researchers in MSI community to use it and provide feedback to help advance it. Therefore, we have also developed this toolbox into a GUI interface so that users can follow this typical workflow to generate the synthetic data for their very particular research questions. At the moment, it is still at internal testing phase, but we are looking forward to releasing it in the near future. So to sum up, we have developed an open source simulation-based synthetic data generation approach and a UI toolbox to help providing a starting point to perform various algorithm comparison objectively. We can systematically create regions and molecular distributions within them, and we can also programmatically and purposely introduce artificial biases to the data in a way to replicate some of the non-biological source of variations in a controlled manner. We have so far used it to help remedy some of the variations in our data analysis, and we provide more results soon. So this is the end of my talk. I would like to thank my colleagues at NICE MSI, National Physical Laboratory, Helen from Marburg, and of course our generous funding institutes. Thank you very much for your attention.
Thank you, Wei Wei. Thank you. Excellent presentation. Thank you to see what you're doing there. We've, as I've said, I think we've got quite a delay with the questions coming through. So I've just got one question that's come through at the moment from my colleague Malcolm. In terms of generating the test data sets, you're using rat brain as the standard sample. What are your thoughts on whether that is a good sample?
We first just use generate data from ground up, basically create the regions on completely artificial data sets. We can use, like basically, if you load in a mouse brain tissue, you can add in the regions to as if you, for example, the matrix deposition or the distribution of certain molecules of the adducts. But at the moment, we're only focusing on either create from ground up of the data, or we generate a data with the introduced artificial biases for evaluation of the methods.
Okay, thank you. That's very comprehensive. Thank you. We've got no more questions coming through. I'm just going to ponder for a moment. But I think what this will mean is moving to an early break. So again, thank you, Wei Wei, for your presentation, and to all of the speakers in this session. And we'll be breaking until UK time, 10 to 4. So it's time for refreshments. And then everyone to come back for the final session, 8, multimodal imaging. So thank you very much for your time, and it's goodbye from me.
Good afternoon, everybody. Good evening to some people and good morning to others. Welcome back to session 8 of Ourcon 2021, which is concerned with multimodal imaging. Before we start, could I just make a couple of requests? So the first request again concerns the use of the chat function for questions. In the studio here, we are about 2 minutes ahead of what you're seeing out there on the internet. So if you are going to put a question in, can you put it in during the talk rather than waiting to the end, because it's not appearing on the laptop on the screen here until after the next talk sometimes has started. The second request is I've had a virtual health and safety avatar complaining at me, and he's asking if people could move their avatars away from the door. It's stopping people getting into the lecture theatre, and it is apparently a virtual health and safety hazard in that you're blocking the fire exit. Okay, and on that note, we'll go over to our keynote speaker for this session. It was Benjamin Bluff from Maastricht, who's going to set the tone for the rest of the session. Are you there, Benjamin? Can you hear me? Okay.
Yes, I can hear you, but I don't know if we have a 2 minutes delay. I don't know.
No, not that. Don't worry about the delay. It's just affecting the people asking questions. Yeah, you and I are talking in real time.
We are in real time. Okay,
yeah, we can talk in real time, but people out there don't hear us talking until 2 minutes late after we've had this conversation. It's slightly odd, yes, but that's the way it works. Okay, I can see your slides are working. If you just want to put it on the slideshow, and then we're over to you.
Okay. Now, does it work, Malcolm?
I can. It's the slides are there, but you're on. You're still on. You're not on presentation mode at the moment.
I am, but
not on what you're sharing. You're not.
Okay.
Yeah, if you you're on that screen. So if you put that screen that your mouse is moving into presentation mode, we'll be in.
Yeah, I'm doing so. Still not.
It's not changed what in terms of what we're seeing. I can see your mouse moving around. Yep.
Yeah.
Just want to unshare and share again, see if that works. Oh no, yeah, you're definitely on that screen. I've seen you activate it. So try putting that one in presentation mode.
So I just
Hi, right, I can see you now.
Yeah.
Nice to see you. Ages since we've met.
Now?
Still, the slides are there, but just not in presentation mode.
I am in presentation mode, so just a second. I'm really sorry for this.
No problem. There's no rush. I think it's taken a couple of goes to get a few of the talks up and running. So just take your time.
So what I will do, just a second.
Hello again.
Yeah, again me. Okay. Now?
Slideshow, yes. Presentation mode, no. So when you go into presentation mode, is it going into presentation mode on your computer? Does it look like it is?
Yes, exactly. That's how it is.
Okay.
Yeah. Then I just give it like that because I make it as big as possible. And
I think in order to make progress, that's a really good idea. Yeah.
Yeah.
As long as the slides advance, just try advancing the slide to number 2. In that mode, just go to slide number 2 and just check that they advance as we're looking at it now.
I will do the following. Tell me now.
Oh, perfect. We're there.
All right.
Full screen.
Good. Sorry for all the fuss, for all the fuss, people. Okay, so thanks, Malcolm, and the Ourcon organization for inviting me to kick off this session on multimodal imaging. And I also gave the title to my talk like that, but with a slight subtitle, namely the importance of image processing and analysis for multimodal imaging and mass spectrometry imaging. And as you all know, mass spectrometry imaging, I must say there's something is going on with the slides. Let's see if that's if it's also a problem for you guys, but I see only 2/3 of my slides, Malcolm. So mass spectrometry imaging can make very nice images, as you all know, and we can see here the visualization of several lipid markers, lipids in a mouse intestine sample. And if you look at the sample, you can clearly guess, yeah, that's a mouse intestinal sample. However, if I had to show you these type of MSI images, and this is another MSAT species, this is another MSAT species, so without further context, it would be very difficult to interpret the visualizations provided by these MSI data. And in order to be able to interpret it, you need to contextualize these MSI visualizations. And with the histology in the background and registered to the MSI data, we are now able to see that one of the first MSAT species actually co-localized with the tumor cells in the sample, the second one with the blood vessels, and the third one with the infiltrating lymphocytes. So this is very crucial. So the combination of imaging data sets, also then under the term multimodal data sets and mass spectrometry imaging, is very important in many projects to really exploit fully the information provided by the mass spectrometry imaging data set. Due to the ex vivo character of mass spectrometry imaging has been mainly be integrated with microscopy, namely the optical microscopy of the H&E state tissue section. Then also I will talk about how to integrate it with laser microdissection. This also been integrated with immunohistochemistry data. And with multiparametric MSI data, we'll just in a few seconds explain what that means, and also in vivo modalities. Now my question to you, Malcolm, can you see the full slide? For example, do you see in vivo or just in, Malcolm? I don't know, okay. As a small example, Katharina Huber in 2014 has shown that you can integrate mass spectrometry imaging nicely with immunohistochemistry for the detection of the drug sorafenib in sarcoma xenograft mouse models. And combining that with digital image analysis, they were able to quantify the intensity of the drug with respect to the distance to the next blood vessel, as indicated by the immunohistochemistry data, and therefore you could show that the drug intensity depends definitely on the distance from those blood vessels as an effect of diffusion efficiency. What multiparametric MSI means has been shown by Heath Patterson just 2 years ago, where he integrated different IMS datasets from the same sample, so usually based on consecutive sections, based on image registration, where every of those experiments has been done with a different MSI modality or with a different sample preparation, so that you can get a lot of different type of MSI data on a per pixel level. For example, it was integrated MSI of proteins with MSI of lipids with different polarities and LDI, all that on a pixel level, and therefore image registration is very important. And finally, to contextualize also the mass spectrometry imaging data with in vivo imaging, many studies have integrated or have already shown that you can integrate mass spectrometry imaging data with MRI or CT data, as recently shown by Walid Abdelmullah, where MSI data of showing the drug distribution of erlotinib was contextualized in the volumetric information as shown by MRI. Now, I want to mainly focus, my work is mainly focused, and the presentation is therefore mainly focused on microscopic data, which is integrated with mass spectrometry imaging. And as a first example about the complementarity of multimodal and or let's say potential of multimodal data sets, we'd like to introduce that study which we recently published. that is about using both modalities, MSI and H&E, for tissue classification and prognosis in Barrett's esophagus. Just one explanation on Barrett's esophagus. It's a precancerous condition of the esophagus that develops over several steps. So Barrett's esophagus without dysplasia, then over low-grade dysplasia, over high-grade dysplasia to adenocarcinoma. And obviously, every of those steps is associated with a certain probability of ultimately developing cancer, where the higher stage you are, the more likely it is, obviously, that you will develop adenocarcinoma. However, the biggest step is actually from low-grade dysplasia to high-grade dysplasia, from 10 to 40% chance developing ultimately adenocarcinoma. That's why a correct assessment of the prognosis of patients with low-grade dysplasia, if they will further develop or progress, is very important. In order to investigate that, we disposed of 57 samples, which were provided by the Amsterdam University Medical Center, Department of Pathology, under the guidance of Professor Silber Meyer, and of which I hope you can see that. I don't know really how the slides look on your screen, but so the 28 low-grade dysplasia samples were separated into 14 samples, which had a follow-up of being stable after 2 years, and 14 samples which are progressing towards a higher stage. And what our interest was, as we have this multimodal data set of MSI and H&E, where MSI was performed on a peptide level at a spatial resolution of 50 micrometer, we wanted to see if we can integrate this data in such a way that we can address different questions in this task of Barrett's esophagus classification of prognosis. So as you can see here, this is just an overview of what data we have actually available. So I said we have mass spectrometry imaging data at a spatial resolution of 50 micrometer. We have also the corresponding H&E data co-registered to the MSI data. And we decided for the machine learning approaches to keep the H&E tile size the same size as the MSI pixel size. And on top of that, we obviously also had the tissue annotation levels as provided by the pathologist on a quite detailed level. Overall, as said, we had 57 patients and almost 150,000 annotated tiles. I want to spare you the details of the machine learning process, but I want to stress again that because this was done in collaboration with the local Department of Precision Medicine, which is specialized on deep learning and advanced machine learning, that let's say these approaches of these advanced machine learning and deep learning approaches have been applied to MSI and H&E separately with state-of-the-art approaches. Yeah, and for us it was just interesting to see which modality can actually perform better at what time. So first of all, when we look at the prediction of the actual tissue type, so if it's stroma or epithelial tissue, we can see if we look at the confusion matrices here. So I hope you can see the second part of the confusion matrices. I really don't know. That MSI does not perform as well as H&E to be able to predict the tissue type. So H&E based classifier is by far better than MSI in predicting the tissue type. However, if we look at predicting the dysplastic grade of the tissue, you can see that MSI starts outperforming the H&E classifier. Ultimately, we were interested in classifying a patient on if the patient is likely to be progressed to a higher stage. And that can only be done on a patient level. That means not on a pixel or tile level anymore. That means at the end of the classifier has to make a decision if the patient as a whole will progress or not. And therefore there is an element in the machine learning that provides a label to the patient based on just the majority of all pixels belong to either progressive or non-progressive. Ultimately, what we also found here is that MSI outperforms H&E in predicting the progression of the patients to a higher stage, with values over 70% sensitivity for MSI and lower than 50% for H&E. That corresponds more or less to what is also known from the literature when we look at what pathologists can do with their prediction on H&E basis. So ultimately, we can then see how this multimodal data set can actually be translated based on its capabilities to clinical tasks. So in this particular case, a single tile or MSI pixel would be classified by H&E based to the fact if it's what type of tissue it is, either glandular or stroma. If then the MSI classifier would be applied if it's low grade or high grade dysplasia, and then also based if it was low grade, the MSI classifier would tell you if it is likely to progress or not. So this very nicely shows the complementarity of these two modalities for these clinical tasks. Now, this study was performed at 50 micrometer resolution, and as, let's say, especially MALDI MSI is moving more and more routinely to a single cell level in terms of spatial resolutions, we really need to match the annotations accordingly. So while the pathologist in the previous study just shown made the annotations very detailed, but the resolution was 50 micrometers, so the pathologist would not have to do the annotations on a single cell level. However, if we move single cell, the annotations have to be done also single cell. So here you can see just how the annotation accuracy has grown over the last years from 2010 to 2020 or 21. So this was sufficient to annotate in 2010. Then with an increasing spatial resolution, this demanded higher detail level of annotation, and now we have reached really single cell level. But as said, the pathologist cannot do that and just in a manageable time. So you need again image analysis and processing tools as provided, for example, by QPath that help you to annotate tissues on a single cell level. So in QPath, for example, has this very nice cell detection algorithm, which can detect the cells in your H&E image. Furthermore, it provides you then, once it has detected the cells, the morphometric characteristics of those cells. And these can then be used to classify those cells based on these characteristics with these with classifiers that you train. And that allows in a semi-automated fashion to quickly annotate tissues in such a way that you have single cell annotations. And these can also be nicely now imported into MSI software. There are import tools for several of the most common MSI software packages with a graphical user interface, such as Skills or LipoStar. And this allows you now, at least theoretically, and I stress theoretically, to overlay the MSI images with the single cell annotations. Why do I say theoretically? Because there is a limitation currently, which is the registration error. And as you can see, this is an example that shows you that we have single cell annotations, but there's an offset to the MSI image, which is based on the registration of these two imaging modalities. And this is because the current state of the art of image registration is basically, in most of these graphic user interfaces, restricted to very simple registration methods, which work interactively, made with 2-point registration, and not on a sufficient detail level so that you can minimize their error in such a way that it is sufficient for single cell mass spectrometry imaging. And therefore, advanced image registration workflows are necessary. It's depicted here, namely that you choose I hope, sorry, something happened. So that you choose, and now I can see my own screen better, so that you choose features from both modalities in such a way that you can use automated registration approaches to optimize the registration. This is, however, quite tedious and requires really image processing and analysis expertise. But this way, currently, it's possible to achieve registration errors that are as low as so below 1 pixel size in order to provide single cell imaging and single cell assignments. So once this can be achieved, this has been achieved, I want to show you what is then possible. You then have the H&E data with single cells, annotations, and also the morphometric characteristics of those cells. You can then correlate these morphometric characteristics with the molecular profiles as shown here as principal components of the MSI data. And you can also, having the spatial coordinates of the single cells, for example, start making comparisons. How do actually the molecular profiles differ from each other of cells that are either close, like these blue cells, or distant to the actual cancer cells? This is a diffuse type gastric cancer sample. But I want to show you what is then starting to get possible. One of the drawbacks is, as we're moving towards single cell and higher spatial resolutions, is that we are actually losing sensitivity. This has already been known and shown, like in 2007, and the higher the spatial resolution, the lower the intensities and the material we can actually extract from a very single pixel in mass spectrometry imaging, together with the fact that it has always been like that, that due to the lack of chromatographic separation, that we detect mostly the high abundant and ionization of molecules, as shown here for protein mass spectrometry imaging. So this together limits, of course, how deep we can go with mass spectrometry imaging. And therefore, we established the spatial omics approach. Maybe we heard that this noon, and the presentation by Janina Oeten from Brukker, where mass spectrometry imaging is used to highlight areas in the tissue that are then cut out by an LMD, and then these are then further processed, submitted to classical microproteomics or other omics pipelines. So one important thing here as well, coming back to the subtitle of my talk, is that registration is really at the core of the spatial omics workflow. Registration means image registration. Yeah, so in this workflow that we established, you need to perform three registration steps until you end up actually at the LMD live image, where you transfer the information from mass spectrometry imaging to the LMD. And if you do that very carefully and accurately, you can, at least we could achieve a registration area of 30 micrometer, which is really, for most applications, sufficient. Of course, not for single cell applications. But it helps overcoming one of these limitations of mass spectrometry imaging. Also here, coming back also to my subtitle, image processing is important, because when you perform, for example, a clustering in mass spectrometry imaging, it is always like it has the salt and pepper as look and feel. And in order to make it really practical for this LMD to cut out different segments, you need to pre-process with image processing. That can be rather through smoothing or removing of small objects and filling those holes, and also ultimately to detect a boundary, because you can only provide polygons to the LMD. And then once you have established this workflow, you can then nicely provide these polygons for dissection to the LMD. After that, because the regions can be very small, you need also ultra-sensitive proteomics approaches. But we're lucky to establish in our lab, where we can, even from very small areas like 0.1 square millimeters, extract and get data of over 2,000 proteins. What could be an application look like? So we applied it for the investigation of intertumor heterogeneity, where mass spectrometry imaging is used to segment out within the tumor molecular distinct tumor subpopulations, which can then be cut out by the LMD, as you can see here, and then analyzed by this microproteomics approach, which then you can do all types of biological pathway analysis or gene ontology enrichment analysis, and start comparing those segments on a more biological and meaningful basis. So ultimately, my time, I think, is over. I want to show you that mass spectrometry imaging and microscopic imaging complement each other. That was with the study on Barrett's esophagus. Every modality has its strength, and moreover, you need the one to understand the other. Then single-cell mass spectrometry imaging requires single-cell annotations. I hope I could also show you that. And that an accurate image registration is crucial for almost all multimodal mass spectrometry imaging approaches. And because this is so crucial, I want to use this this moment to announce a small initiative that Helen Race from the University of Marburg and me are initiating, namely We call it Mass Spectrometry Imaging & Co. And there will be the kickoff meeting just next week at the same time in Zoom. So you can make maybe a screenshot of this. And if you would like to join, it is about developing new image alignment tools for multimodal mass spectrometry imaging and everything that is related to that, maybe new data formats for mass spectrometry imaging and so forth. So if you are interested, please join our kickoff meeting next week. At 16:00 Central European Time. If there's anything, you can still contact me. I can send you the details, but they are also on the website of Comulis, which funds this initiative. Okay, thanks so much, and I'm happy now to answer the questions in the chat.
Thank you, Benjamin. If you'd like to stop sharing your slides now, so we can speak to you. So that was an absolutely stunning talk, and the images got better and better, and I think there are questions on the chat, but I'll ask the question that must be on everyone's lips who's been watching it. How did you persuade it to go to full screen from half screen? So we were seeing half of your slide as you feared for quite a lot, and then suddenly...
You went, oh my God, this is really...
How did you fix it? I think everyone wants to know, just in case it ever happens to them.
Was it fixed during?
Your screen share suddenly went off, and then when you went back in, as if by magic, we had the full screen. We don't know.
I constantly was getting some emails, and I thought it was maybe you telling me something. So I quickly.
It was everybody. Yeah.
I went outside, and then I went back again inside.
And then it worked. So as someone said, it was the turn it off and on solution. Yes. Always a good idea. Okay, don't worry. Yeah, it was good. It was excellent talk. So there are lots of questions. Yeah. So let me get on to the proper questions from the chat. So the first question comes from Francesco Greco, and he's asking, how does our path perform in automatic cell recognition mode if there are laser marks on the sample?
That depends on how intense those laser marks are. Ultimately, the cell detection is based on the nuclei, and it's very unlikely that the laser will make holes that will look like cell nuclei. But that I cannot speak for every MS system how these burned holes look like. But as long as they don't look like nuclei under the H&E stain, so usually blue, with a certain size, then I don't see that would result in a problem. But I cannot.
Okay, thank you. So you were talking about proteomics, and this is from Martina. So talking about proteomics and laser microcapture detection. How is it for other analyte types, and what spatial resolution were you using for your analysis?
The spatial resolution, that's 50 micrometers. That was 50 micrometers. And we many times got the question about how does this, with a more dense ablation of the laser, would be detrimental to certain molecular classes. And we have not investigated that to the, let's say, fully, but I can, from maybe a parallel. So the laser marker is for, if you put your slide on an ITO slide, if you put your section on an ITO slide or a glass slide, it needs to be ablated by the LND anyhow. So the tissue is burned from the slide anyhow. So, and there it has been shown that it doesn't have such a big impact, the laser, on on proteomics at least. So it is another laser, but it is by far more dense laser because it has to burn away the tissue. So, and there it has been shown it has limited impact on proteomics. For other molecular classes, I can't tell.
You can't say. Okay, thank you. Let me just see this. No, I was just probably got time for one last question, and it's from me, and I think it probably It isn't something you covered in your talk, but you were talking about some registration and correlation with in vivo MRI type imaging. I just wondered how registration is being, co-registration of the images is being done for that particular experiment.
Yeah, so that has not been done by me, but that was done mainly by Walid Abdelmullah. And this is the approaches are very advanced image registration. Yeah, so this is the point that I wanted to make here is that we need really advanced image registration and really expert to do that. Yeah, so this has all been done. It would be now too technical, but it's all really advanced non-linear image registration techniques which are not available in any MSI package.
Yeah.
So,
but it sounds like the future. Or is it the future, or is the future here now, and it's happening in other labs? Yeah.
What is it? You mean the integration within vivo?
Within vivo, yeah. How close are we?
I think that my personal opinion is that will be very niche application. Yeah. The future will be certainly, because we are doing that since many years, the integration with H&E or other, and that is the future, just on a by far more accurate basis. That's for sure. That will be for sure. But with in vivo, that requires special collaborations, and will still be a niche application. I have to see that.
It's good. Thank you very much, Benjamin, and thank you for fielding the questions and for battling with the technology so manfully during your talk.
Yeah, I'm really, really sorry, and it's really awesome to see you there. It's really spooky. Next time, I really hope to be there in person.
Yes, same here. It'd be nice to see you again. Yep. Thank you very much. So
great. Do I disconnect then, or how does it work?
No, you'll be thrown off. Yep. So if we could move on to our next speaker, please, producers. And so our next speaker, good afternoon, Florian.
Yeah, good afternoon.
Can you hear me? Yes. So our next speaker is Florian Glerard. Do you want to try sharing your slides, please, Florian?
Yes, I'll try.
So right at the bottom of the screen. Yeah, great, we're in. So when you're ready, take it away. We've lost your audio now, Florian. We can't hear you. If you can see me, we can't hear you. We could hear you, but now we've lost you. Have you accidentally muted yourself? Okay, so maybe try coming out of the slide share. Just see if we can talk to you again. We'll try the turning it off and back on again trick.
Hey, can you hear me?
We can hear you now, yes. So we lost your audio when you went into slide share.
Okay, I'll try again. Hope that works. Okay, can you hear me?
Yeah, that's good. We're good to go now. Yep.
Perfect. Okay, so thank you for the nice introduction, and thanks to the organizing committee for having the opportunity to talk here today. I'm going to talk about fusion of three-dimensional mass spectrometry and magnetic resonance images with an application to the wheat grain. So in this project, which I started at INRAE of Nantes, we studied the wheat grain development, and more specifically the molecules that are involved in this process. Cell walls play a major role in this. They have specific mechanical properties which allows for cell growth and differentiation. They also allow to regulate the water and nutrient intake inside the cells. And chemically speaking, the cell walls consist in layered polymers involving, among other molecules, cellulose and hemicellulose. In this project, we are interested in arabinoxylans, which are a type of hemicellulose which are found in the wheat grain. And they consist chemically in a chain of xylose, onto which arabinosyl residues in green here can branch. And the degree of arabinosyl substitution directly impacts the physical-chemical properties of the polysaccharide. On another note, in our group, we highlighted the gradient of spatial distribution of highly substituted arabinoxylan in the wheat grain. And this spatial distribution is similar to that of water. So the goal was to find objective quantification, to find automatically spatial correlations between molecules and water. And to that end, we had two images at our disposal. The first one was MRI, where images enclose the water distribution. So you have one example here on the left. And the second type is MSI, where you have hyperspectral image, and you have one ion image here on the right. So the purpose was to perform automatic image fusion in order to find the molecules in MSI which correlate with the water distribution in MRI. But there are several problems for this because the images are different in nature, and also the acquisition are different. So about the acquisition workflow, we can work on the same grain. We start with MRI. We can acquire the whole grain, so that's simple. Then we use the same grain, we process it, we obtain different slices, on which we use an enzyme to look at the molecules of interest. Here it's arabinoxylans. And then we spray the matrix onto the sample, and we can then send this sample to the acquisition device. Here it's a MALDI-TOF. So we get an MS image, but from an image analysis standpoint, there are other problems for the joint analysis of these images. For MRI, we start in a format that's raw, but there are several converters that allow us to work with NIfTI and TIFF. At the end of the acquisition process, we obtain a three-dimensional image along with a time dimension, so 3D plus time image, that is directly dependent on the acquisition sequence of the MRI. Here it's multi-echo sequence, and you have here different echoes for the MRI. The voxel size is 50 by 50 by 500 microns. And by contrast, for the MALDI-TOF images, we have a format of Brukker FID at the start, which we can convert to IMZ-ML thanks to various tools, including the one from Alexandrov's teams called PyMZ-ML. And at the end of the acquisition process, we obtain a data cube where we have a spectral dimension with M/Z ratios. The pixel size is 25 by 25 with a 90 micron between slices. There are other issues because we are studying several slices inside the sample. And for MS images, we have two types of problems. The first one is the intensity inhomogeneities inside the sample. And the second one is the fact that they can have different orientations. That is very apparent here. For three same slices inside the sample, for MRI, the intensities and orientations are consistent, but for MSI, that is not the case. So we have to handle these problems, and we need reproducible and generic computation methods in order to perform the image fusion. So to achieve that, we proposed a 3D workflow that is based on a previous workflow in 2D that was published in BMC Bioinformatics, and that is also available online as a software. The software is called Esmeralda. It's completely available online and open source, but I will get back to that in my last slide. So about the 2D workflow, I won't go into too much detail here, but to recapitulate the steps, we start from the raw images, we process them, we obtain a shape that is representative of the sample, of the wheat grain, so as to align them in a registration step. Then, once both images are aligned, we can find the spatial correlations, and that is performed by non-negative matrix factorization by NMF. So in the processing steps, there are various steps, including normalization, peak picking, and peak alignment of the sample for MSI. In this talk, I will focus about the 3D aspects of the workflow. So what steps are changing in the three-dimensional case, so that are registration, statistical analysis, and visualization of the images. So to start off with registration, the registration methods that we use are completely automatic. They do not require user input. And the algorithms behind this process are defined by 3 components. The first one is the geometrical transformation. It can be rigid or affine. So those are transformations that involve rotation, scaling, and translation. And deformable transformations can account for local deformations inside the sample. So the purpose of this algorithm is to find the perfect transformation mapping the target image onto a reference image. So in our case, we want to map the MS image onto the MR image. In order to do that, we need to, the algorithm proceeds by several steps, several iterations. They modify slightly the parameters of the geometrical transformation, and they check whether this transformation is better or not than at the previous step. In order to compare the result of the transformation, we have a similarity metric, which estimates the similarity between the reference and target images. It can be based on intensity or the shape of the sample. And then we have the optimization algorithm, which finds the minimum of the metric and find when to stop. So here I will show the example at the start of the iterations, and over time, the transformation parameters will get better and better and stabilize and converge towards something that is much closer to the MR image. The main problem in this kind of algorithm is to select the appropriate similarity metric. In our case, as I've said previously, we have intensity in homogeneities inside the MS image, so we cannot use intensity-based metrics. Instead, we use geometric-based metric. In this case, we convert the images to the distance transformation of these images. And distance transformation maps each pixel of an object to its distance to the border. So from both these images, MR and MS, which have different intensities, we get those kinds of images which are much more comparable. Then the registration approach proceeds in 2 steps. First one is to register each pair of 2D slices independently. After the registration of these images by affine transformation, we get a 3D volume, which we can then register by a deformable transformation in 3 dimensions. So after affine registration, we need deformable registration because certain areas in the sample are not properly aligned with the MR image. That is very apparent here in this region. And the difference can come from the the sample preparation done in MSI. So the cutting, the enzyme that we use, and also the matrix spraying. So about the deformable registration, we use variational methods where displacements are modeled by a vector field. And this vector field is guided by 2 forces. First type of force is external forces. So that is the similarity metric. And the second type of force is a regularization term, which allows smoothness of the vector field locally in order to avoid artifacts, local artifacts of the vector field. So over time, we have several iterations, and once again, it will converge towards something stable. And we can see here in this case, the part that I highlighted before, it's stretched so as to match its counterpart inside the MR image. About the statistical analysis, it's done iteratively here on each pair of corresponding 2D slices. It can be done also in 3D, but since we have inhomogeneous intensities, it made more sense here to work on each slice separately. So the principle is to start off by dimension reduction of the MS image by non-negative matrix factorization. Once we have a reduced space of the MS image, we project the MR image onto the reduced space. And then we select the ion images in this space, which are the closest to the MR projection. But the problem is at this stage that we've performed the registration on the shape of the objects. So similar regions in terms of intensities might not be perfectly aligned. So we try to account for this problem by using the information given by the NMF. And the purpose is to register each component image coming from decomposition by the NMF of the MS image. So each component image encloses a particular interesting region of inside the sample. And we want to register those component images individually onto the MR image. So after registration, we obtain something that is aligned with the MR image. And then we use these component images to create a new subspace onto which we project the MR image. And then the spatial correlations are established into a merged space by proximity, by closeness with respect to the MR image. Okay, so that concludes the workflow, description of the workflow, and I will now present some results. So about the registration, each step has been carefully validated. Here, I do not show all results, only on one example. So we have the MR image here on the left. We have the MS image for different parameters of the registration. So if we choose a classical registration based on the intensity, we obtain a registration that is not satisfactory because we have the fact that the intensities are inhomogeneous. Instead, if we use the distance transformation, we get a registration that is properly aligned with the MR image, but still has local deformations that are not yet accounted for, but that we correct then during the deformable registration step. So we obtain up to 83% shared pixels between the two. And what subsides can be small areas where there was no signal inside the MS image. So after registration, we can perform the correlative analysis, the statistical analysis, and we want to compare, to find, sorry, the MS, the ion images which correlate with the MR image. And in this case, we found two types of arabinoxylines which correlated with the MR image. The first one is highly substituted arabinoxylines, which are mainly located in a region called the crease of the wheat grain, the centered region here of the wheat grain. And another family of arabinoxylines are acetylated arabinoxylines, which are distributed mainly in the posterior part of the wheat grain. So this is very interesting because this is the first study that shows objectively the distribution of such species and the correlation with water. So in conclusion, we have proposed a workflow which is suited for 3D MS images. All the tools are available online in a library called Esmeralda. It's completely open source. It's written in Python 3, supports IMZML, and is available at this address. So it's still an ongoing project here at my new lab in CBMN of Bordeaux. And it will be extended, most notably with the new GUI in the coming future. Another prospect for the workflow is to work on a 3D analysis of the sample, which is not complete yet, since it's done iteratively on each 2D slice. With that, I would like to thank my former team at INRAE, and in particular David Legland, Mathieu Fanuel, Loïc Foucault, Helen Rognou, and Bastien Arnaud. Thank you for your attention, and I'm happy to answer your questions.
Thank you, Florent. That was absolutely fascinating talk in a really difficult problem. I think we've just got time for one question, and it's to do with when the registration software is needing to actually stretch or rotate images because of changes in the shape of the tissue during probably the sample preparation procedure or sectioning. In terms in terms of actually the decision as to whether that stretching or rotating is maintaining a true representation of the original shape, how is that done? Is that subjective or is that done in the software?
So that's validated quantitatively. We have some kind of measures, the mutual information that allows us to know if the intensities inside the registered image is the same as the original image. So that's what we use to measure this criterion objectively.
Fantastic. Okay, thank you for that. That's I suppose you're doing the one last quick question. If I'm sure the producer will let me off. He's telling me in my ear that we are over time now. Don't worry. Yep, we enjoyed ourselves. So how about you're doing this work on essentially the weak grains, which obviously are ex vivo situation. The question that I asked Benjamin, is your software applicable to the in vivo situation if you had MR data from a living system?
Yeah. Yes. So right now it's mostly in vivo, and there are some assumptions that are made from this. That the fact we observe a sample that is delimited, that we have a black background that delimits the shape of the object. If we have images where we don't have this prerequisite, it might be harder to use the algorithms that I use for registration. But right now at my new lab, we're working to extend these methods. So yeah, it's a work in progress.
Fantastic. Thank you very much. I'm sure, again, if people want to speak to Florent directly, I'm sure he'll be happy to answer any other questions if you put a message to him directly on the chat. So yeah, thanks very much.
Thank you.
Okay, so our final speaker of this session is Lucy Flint. Good afternoon, Lucy.
Hello, good afternoon.
Great, we can hear you loud and clear. So if you'd like to share your slides, we'll just check that they work okay.
Goes into slideshow.
Yep, that's perfect. So take it away whenever you're ready, Lucy.
Great, thank you very much, and thank you everyone for staying for the last session. I know it's the end of the conference, so I really appreciate it. I'm going to talk to you about the development of a multimodal imaging strategy to analyze 3D cell culture models for the purposes of preclinical biopharmaceutical testing. So a bit of background, 3D cell culture have emerged as biologically relevant models, and they are representative of in vivo tumors and diseases, and therefore they are quite valuable for early stage drug development. Likewise, biopharmaceuticals have become of quite high interest due to their high specificity and selectivity, and therefore there's an increasing demand to be able to analyze them specifically through imaging techniques, and that's where my whole project stemmed from. So just a brief overview of the imaging workflow. So the 3D cell culture model that we used in this study is termed an aggregoid. So this was developed by the Sheffield Hallam Research Group. And basically an aggregoid is the aggregation of tumor spheroids to create a larger tissue construct, typically about 1 millimeter in diameter. But it means it's got a lot more heterogeneity from all the individual spheroids, and it's quite morphologically representative of an in vivo tumor. So once this aggregoids have been cultured and treated with the biopharmaceuticals, the imaging workflow is as simple as all the rest of them, with embedding them in an embedding media, flash freezing, sectioning, and then preparing the tissues for their appropriate analysis. So the 4 modalities that I used in my study was DESI, MALDI, LAICP, and IMC. So if you attended the 24-hour MSI meeting last year, I did a brief, very short talk on the characterization of a HCC 827 lung adenocarcinoma aggregoid. So by employing DESI, IMC, and LAICP, I was able to detect metabolites, proteins, and metal isotopes that were endogenous to the aggregoid model, and it gave us a nice, clear characterization of the tumor microenvironment. So, for example, the intermediates of a TCA cycle, such as citrate, were detected within the outer region of the aggregoid, indicating an area of proliferation. As alternatively, you have lactate from the glycolysis reaction that was more detectable within the core, indicating a region of hypoxia. This was complemented with the IMC data with the protein expressions, where Ki67, a protein, a proliferation marker, was identified in the outer region, and the proxy hypoxia marker, GLUT1, was expressed more predominantly within the core. Then again, looking at the metal isotopes, we could see these distinct regions with an outer region and inner core by looking at magnesium, zinc, and the copper, giving a clear identification of a cellular metabolism and metabolic debris in the core. So with that in mind, we decided to kind of use the same strategy in order to characterize other aggregoid models of osteosarcoma. So MG63 aggregoid, which is a fibroblastic cell type, and SAOS2, which is an epithelial-like cell type. And we also compared these to clinical patient tissues, one metastatic and one not metastatic. So here from the DESI data, we did an unsupervised spatial segmentation using skills analysis. And initially, we were looking for any similarities between the aggregoid models and the clinical tissue. So a perfect example of that was looking at fatty acids, specifically of the arachidonic acid metabolism pathway. So if you look at the metastatic patient here, we can see almost like a clear pathway of Fa18-2, which is linoleic acid, down to Fa24, which is otherwise known as arachidonic acid. You can see there was more high detectable levels within of arachidonic acid compared to linoleic acid, suggesting quite a high active metabolism. Alternatively, from the non-metastatic patient tissue, we could see almost the contrast, where Fa18-2 had high detectable levels in comparison to the Fa20. When looking at the osteosarcoma models, it's clear to see that they followed a similar type of active metabolism to the metastatic patient, with quite high signal intensity within the MG63 of the Fa24. And interestingly, the SAOS2 model showed more detectable levels of this intermediate Fa18-1, but still showing that kind of metabolism from linoleic acid down to arachidonic acid. To see if there was any specific metabolites that corresponded to a specific aggregoid model, we looked a little bit closer. So, for example, ceramide species were detectable more within the SAOS2 model, specifically within the core of the aggregoids. Interestingly, we were able to detect the ceramide species within certain regions of the metastatic patient tissue. And by looking at the pathology report, these areas of the tissue actually corresponded to bone mineralization and differentiation. So potentially suggesting that the SOS2 model has these characteristics of bone mineralization as well. Then looking at the MG63 model, we were actually detected quite a lot of phospholipids, PS38.4 and PI38.4. And again, these were detected in the metastatic patient tissue and slightly within the non-metastatic here. And according to the pathology report, these were areas of solid tumor regions. And interestingly, these two phospholipids have been associated with high metastatic cancer of breast cancer. So if the phospholipids have that same kind of role in osteosarcoma, it's potentially showing that the MG63 has these metastatic characteristics, potentially. Then moving on to the complementary techniques. So imaging mass cytometry, looking at the protein expressions. So straight away, you can see the differences between the phenotypes of the MG63 and the SAOS2. With the SAOS2 for vimentin and collagen, which are clearly show like the differentiation status of the cells, we can see them more within the outer region of the SAOS2 model. However, they are distributed across the whole MG63. The most interesting one was actually the distribution of Ki67 and GLUT1. So likewise with the HCC827 aggregoid model, you can see the SAOS2 show distinct regions of upliftive outer region with the Ki67 expression and a large hypoxic region with the GLUT1 expression. In contrast with the MG63, it shows that there is a homogeneous distribution of both protein markers, suggesting that the GLUT1 gene is actually constantly switched on within the MG63 cell type, regardless of the environment in which it's in. So even in a surplus of oxygen, the GLUT1 is still switched on. And this kind of gives an indication of potentially a tumor that's relevant in quite hypoxic areas. Then looking at the metabolite, the metal isotopes, so magnesium and zinc, again, we were able to see some really high detectable levels within the MG63 compared to the SOS2 where there was just this thin rim, suggesting again that there's a higher activity of energy metabolism within the MG63 model compared to the SOS2, and this also gives an indication of that differentiation status. So now that we've characterized the different models, we kind of wanted to use that as a reference to see what metabolites have changed in response to biological treatment. So for this strategy for determining the biological response, I employed a multivariate analysis strategy in combination with DESI MSI, and I looked at 3 biopharmaceuticals. So for the osteosarcoma aggregoids, I looked at TRAIL and anti-DR5, which are clinical trial drugs that induce apoptosis, and HCC827 was looking at cetuximab treatment, which is an FDA-approved drug, and it targets the epidermal growth factor receptor. So as you can see, there were quite a few metabolites that were significantly upregulated or downregulated in response to treatment. And that's kind of how the data looks like. So we have separation between the controls and the treatments, using the loading plots to find these specific MZ values and metabolites. And as you can see from the images, there was clear differences in the metabolite distributions. However, although that looks quite nice from the naked eye, unfortunately, that was kind of the limited response. These metabolites that were detected were pretty much it. So potentially this is due to a couple of variables, whether we just need more biological repeats, this is N = 3, or a longer incubation time of the drug. But it also could lead to biological resistance, which is commonly found in 3D models, animal models, and obviously in clinical trials. So potentially by growing the cells in 3D, we could be seeing a mechanistic resistance, whereby genes and protein expressions have been switched on or off and therefore inhibit and impact the specific pathways that these drugs target. And that would require a lot more studies to understand the roles behind those. Alternatively, we could be seeing a delivery issue. So if the drug cannot penetrate through the tissue, or an insufficient amount of drug has reached the target. Therefore, we would be unable to detect enough changes within the metabolites. And that's kind of where the next part of my study led on to. So to kind of understand more about how the biopharmaceuticals work, where they are distributed within the tissue, it's important to be able to detect them. However, with biopharmaceuticals, they are much larger, complex molecules compared to small therapeutics, and therefore they are harder to ionize with low efficiencies. So, for example, I was looking at cetuximab, and cetuximab is 150 kilodalton in size, so very large. I was wanting to be able to detect this, and as we all know, a way of detecting proteins is by looking at their tryptic peptides, following a bottom-up MALDI MSI proteomics approach. So following this, there's a lot of different variables, trypsin concentration, tissue washing to remove any lipids and salts, and also incubation methods to find the optimum enzymatic activity. I also looked at trypsin deposition. So this is applying the trypsin onto the tissue, which is very important, obviously, for the imaging spatial information. The conventional spray coat method unfortunately didn't really work for my aggregoid because, as you can see, for endogenous proteins, peptides, for histones and actin, I had very small signal and actually quite a lot of delocalization. However, if you looked at the rat brain section, which was performed in the same experiment, the digest obviously did work because we could get some really nice detail of the cerebellum. Then moving on, I also tried a microspotting technique. So because the aggregoids are a millimeter in diameter, we thought that potentially by microspotting about 50 nanoliters of the trypsin over the tissue would help increase the sensitivity. So as you can see, it did increase the sensitivity. However, it completely lost the spatial information, and instead we got these lovely water ring marks. So not ideal for imaging studies. So there's still ongoing optimization that is required there. However, we wanted just to see if we could detect cetuximab in the first place anyway. So by compromising the spatial resolution, the spatial information, we decided to go ahead with the microspotting technique with the optimum parameters. So, like I say, we were looking for cetuximab tryptic peptides. So because cetuximab is a monoclonal antibody, it actually contains these complementary determining regions, which are unique to the antibody, and therefore they can be used as reference peaks for further validation of what we were detecting. So this is very preliminary work, but I was able to detect some signal using the microspotting technique, specifically of this MZ1788, which was containing a CDR region. However, this was with quite a high concentration of 150 microgram per mil, which is about 1 micromolar, and we were getting very, very low signal. So again, still really need to improve on that sensitivity, but also need to improve on the spatial resolution to get that information of the drug distribution. Alternatively, by following the multimodal imaging approach, we had the idea of using a metal detection, so more of a targeted approach to detect cetuximab. So based on the principles of IMC, where you have an antibody with a lanthanide species that targets the protein, we thought we could do the same with the cetuximab. So initially, I just tested to see if we could detect europium within the solution after the conjugation. And then also seeing through a routine antibody staining technique of tissue washing, whether we could still detect this europium cetuximab, and we weren't just detecting excess europium. So very nicely, as you can see, by detection of LA-ICP-MSI, we were actually able to detect some europium within the treated samples compared to the control. So this gave us an indication that the cetuximab is more localized within the core of the aggregoid. And because we know about the tumor microenvironment of this lung adenocarcinoma aggregoid model that I spoke about earlier, we could tell that this is more within the hypoxic region. And it kind of gave us an indication that the EGFR expression could be associated with hypoxia. So with that, just a brief overview, just to conclude, we were able to characterize 3 different models of aggregoid models of cancer, showing that the need for characterizing and phenotyping them by using different techniques to be able to understand more about the environments within these tissues. And then we use that multimodal imaging approach to actually spatially resolve any metabolite changes in response to biological treatment, and we've made some progress in actually being able to detect the drug itself. So I'd just like to say thank you to Sheffield Hallam and my supervisory team, especially Malcolm, because I've now finished my PhD, and then all the team at AstraZeneca as well for their collaboration, and thank you all very much for listening.
Thank you, Lucy. Absolutely excellent talk, as usual. So we do have a question for you in the chat, and then I'd like to ask you a question. Yep. So the question in the chat that's come up is about embedding mediums. So to embed such small objects as your spheroids, how are you going about that?
Yeah, so it was quite a delicate technique, and it's quite something that you have to practice. So because they are so small, you can't typically use like tweezers or spatula or anything, because you will just break the tissue itself. So the technique that I was using was actually using a pet tip, cutting off the tip so then it's larger for then you to pipette upwards, and then delicately pipette into the embedding media. And then I would always wash it with PBS, so I would try and remove any PBS before freezing as well. So it's definitely a delicate technique.
And in terms of the actual medium itself, what medium were you using for embedding?
Yes, so it's an embedding medium that was designed by AstraZeneca. So it's HMPC and PVP. So it's kind of liquid at cold temperatures, which is much better than like gelatin or CMC, because you don't run the risk of having heat destruction of the samples, and it's clear as well, so you can clearly see your tissues.
Great, and I think Greg mentioned that in his presentation yesterday. Yeah. Okay, so a second question from Martina. Do you verify the results from cell culture in xenografts? So because the actual tumours themselves are vascularized, unlike the aggregoid models.
We haven't really. The only kind of what was the question? Sorry to.
So it's about trying. So you moved on to the trying to compare the 3D cell culture model with ex vivo human tissue.
Yeah.
So there possibly could have been an intermediate step looking at xenografts.
Definitely.
Yeah.
I guess we kind of jumped the gun a little bit just to compare straight to clinical tissue. But yeah, that would be another step to compare with xenografts as well. The whole point, though, however, of this project was to try and avoid any kind of animal usage. So yeah, we kind of just looked at clinical tissue straight away. But yeah, it's an ongoing thing. You could definitely compare it to all these different types of models to see how they compare.
Yeah, to actually do the full validation. Yeah, exactly. Okay, and the final question is just from me. You were talking about you talked about the idea of the cells showing metastatic potential. If I'm right, yeah. So, and is that a known phenomena that a certain cell line would show more metastatic potential than others?
I think it's just, I suppose it depends really. You wouldn't necessarily say that the cell line itself is like metastatic. It's just more, it could exhibit certain characteristics of it. But yeah, I think you have to be careful with the fact that it's like if it is metastatic, because I know that the MG63 cell line isn't classed as the metastatic cell line, but it does have, you can kind of manipulate it to become a metastatic cell line.
Thank you. I think we've got one more. So just time for one more. How many days did you let the spheroids grow for?
Yes, so the spheroids initially grew for about 11 to 12 days, basically just to make sure that there was a sufficient amount that were up to about 100 microns in diameter. And then from that, you took it out of the alginate scaffold and you cultured them in the 96 well plate for 7 days to grow into the aggregoid. So it's about 3 weeks altogether.
3 weeks. Excellent. Thank you very much, Lucy.
Thank you.
Good luck in your new job.
Thanks.
Okay, so everybody, we are just about at the end. So I'll ask Liam to join us now. Hi, Liam. I think we're going to come over.
Hi, Malcolm.
Hi. So I think we're over to, do you have you been, before I ask you to talk about Ourcon 2022, can I check, have you been emailed to the prize winners?
Have I been emailed? I have not seen anything.
No, okay. Right. So while you are talking about Ourcon 22, I shall get in touch with the judges and make sure that we do know who's won the prizes for the prizes at the end. Okay.
Okay, please do that quickly because I don't have that much to say.
No problem.
Okay. So Ourcon 2022, we are pleased to announce, will take place in Japan in November next year, 2022. It will be in Kyoto, organized by Shuichi Shima. The dates that are planned is November 16th to November 18th. This is a Wednesday to a Friday, so to allow people then to also use the opportunity to visit the local sites, the tourist attractions. The location is not yet confirmed. We have a number of possibilities and we're currently chasing up the different options. Shu is currently chasing up the different options. And as soon as that is confirmed, then we will also confirm the provisional dates of November 16th to the 18th. But of course, we will announce this and promote it very, very actively. The meeting for 2023, well, this is going to be organized in very close collaboration with IMSS because we're in the process of trying to unify the societies, so the Mass Spectrometry Imaging Society and the Imaging Mass Spectrometry Society as a new society. And that meeting will actually probably not be called Ourcon, but it will be called the name of the new society meeting, and we'll continue the Ourcon name as a European-focused meetings on mass spectrometry imaging. So there will be a change in the how we organize these meetings, but the nature of the meetings will continue. Mass spectrometry imaging, very friendly, very open. We've been very active in trying to foster as much as an open and collegiate atmosphere as possible, and I sincerely thank you all for maintaining that here, even under these virtual circumstances. Malcolm, do we have the names of the prize winners?
Yes, we do.
Perfect.
So I think in terms of our virtual world, given that I can't actually show you my phone, I think I'm probably going to have to announce them, aren't I? So
yes.
Yep. So the winner of the prize for the best poster by researcher, this is the Mass Spec Imaging Society prize, is Dennis Roucherong. That was poster number 58. And from the oral presentations, the winner of the prize for the best oral presentation from a young researcher is Nuria Sleekhouse. Congratulations to you both. The oral presentation prize is, so both of these will be in the form of vouchers. So there'll be a voucher from the Mass Spec Imaging Society going out to Denise, and we're going to be sending a book token because this prize was sponsored by Springer. So there will be a €250 book token coming to Nuria, and both of those will come to you by email shortly. Congratulations to you both. Very well deserved. Very well deserved, both of you. So if we can go back to Liam as well, I think we're about there, Liam.
Yes, thank you. So I'm going to take this opportunity to thank Malcolm for organizing at a really quite short notice a splendid virtual Ourcon conference. It has actually been one of the best virtual meetings I've attended. So on behalf of everyone, I'm going to give Malcolm and his people a round of applause.
Thank you very much, Liam. I hope everyone's enjoyed it. If you check my Twitter feed, you can see what this studio looks like in reality. That you might find that interesting. Yeah.
Okay. Now we do naturally hope to see everyone at the next Ourcon meeting in Japan, and if you are not able to make it there, we hope to see you at the workshop that we will organize as soon as the sort of national and institutional travel restrictions are lifted. We have been, we've refrained from organizing a meeting until we actually know the travel situation. And the reason for that is so we don't have to cancel again or to change from an in-person to a virtual meeting. So the virtual meeting has been fantastic, but there is a strong desire amongst people now for an in-person meeting. So as soon as we know that people will be able to travel, we will quickly basically organize an in-person meeting. Actually, this is perhaps my last activity as the president of MSIS. So when I say we, I mean, well, the other people who are remaining in the MSIS board, a royal we, perhaps. So thank you again, everyone, for your active participation, and I hope we can see each other again soon.
Thank you. Goodbye, everyone, and thank you very much, Liam. Thank you for attending, and see you all soon.